All right, everybody. Check, check. Welcome back. Good morning for our final day here today. Again, I hope everybody had a good time yesterday on their field trip. Another beautiful day out there today. Before we get started, though, I might turn the mic over to Melanie Woolover to give kind of an update on one of our friends, Eric Rominger, and I'm going to be passing around a card for Eric. So Melanie's going to fill you in on what's going on there.
Sorry, I've messed it up already. So I've told this story several times, but I guess everybody probably needs to know. Eric Rominger ended up in a mule accident, and I think it's been about 2 weeks ago, and it really isn't funny. But he called me from the ICU room, which is astounding, to tell me that he'd been in a mule wreck. He was coming out with his cougar person that maybe the cougar gods were after him, actually. Following behind the cougar guy on a mule, and the cougar guy looked back and he was in a heap on the ground. So he walked out 2 miles. They took him to the Socorro Hospital. It was snowing terribly. They think that the mule broke through the snow and landed on a piece of PVC, and somehow Eric has amnesia, so he does not remember. He doesn't know what happened. Nobody knows what's happened, but anyway, he ended up at the Socorro Hospital and airlifted the next day to Albuquerque, and he has 7 broken ribs that are broken off and in pieces, too near his spine to do surgery, a broken vertebrae, let's see, a punctured lung, broken scapula, broken sternum, and head contusions, and he was unconscious for 5 minutes and doesn't remember anything. So at the very least, I'm thinking concussion. So he is now home. He called last week before I left. So he's home now and recovering, and thinks he's going to be in the field in 2 weeks, to which, yeah. So anyway, so he's pretty broken up. So you might, if your buddies send him a note, I think he's going to be bored to death. So you might want to call or send him a note. And oh, and we have a card. Mike, where's Mike Cox? There he is. Mike got a card for us, and we're going to pass it around. So if you know Eric or don't and want to sign, please do.
And has anybody sent him like a stuffed mountain lion toy? That might, yeah. So that could maybe go with the card. Absolutely. All right. So just a slight change of plans here. Peach Van Wyck won't be your moderator this morning. That will be Jessica Jennings Gaines, who is a wildlife disease specialist for the Wyoming Game and Fish Department in Laramie and in our wildlife health lab, who's been a critical component of all of our disease work statewide for many years, especially our bighorn sheep disease surveillance work. So with that, I'll turn it over to Jessica.
Good morning, everyone. This morning we move into disease and parasites, which is more of my purview. And we're going to have 5 talks this morning. So starting off, we have Christopher McGlover, who is currently a PhD student at the University of Wyoming in the lab right next to ours. Chris has done a lot of education between Minnesota, Montana, and Iowa. He has worked on bighorn sheep in Nebraska, Nevada, and Montana as well, and spent a lot of his young life working on bighorn sheep. His PhD is working on strain typing some of these bugs that we're getting out of these bighorn sheep, and doing some microbiome work. And he's going to talk about that today.
Thank you, Jessica. And thank you guys for all being here today, especially the committee that's put this together. It's been an awesome event. So today, I would like to talk about past strain typing. I'd like to talk about, there we go, past strain typing techniques that have been used in the field, and then also a current diagnostic tool that could be used in a similar manner to get the most out of the samples that we're collecting from these bighorn sheep. So coincidentally, about the time this picture was taken 15 years ago, there was a drastic increase in strain typing publications in the bighorn sheep field. And I think a lot of that has to do with Koch's postulates, if you guys are familiar with that. And really what I mean by that is that at the time, we could not find 100% of the pathogens in all diseased individuals from die-offs typically. And then as our surveillance got better, we also detected a lot of these pathogens in healthy herds as well, or healthy individuals. And that really can be seen on this figure of Wyoming and Nebraska. This is pathogens found in the herd at the herd level. So red is Mycoplasma ovidipmoniae. Blue is leukotoxin Mannheimia hemolytica, and then there's all assortment of viruses and parasites as well. And each one of these herds has really a different population response. Several are increasing, several are decreasing rapidly and are of concern, and a lot are stable. So there was really no pattern to what pathogens are found in a population, and then the population performance. And I think a lot of this can be explained by some recent publications done by a couple members in this group about the limits of detection. So for the pastorellas, you have a probability of finding a pastorella from about 40 to 60% of the time. So that means if two individuals have pastorella, a lot of times we only find it in one of them. So that's a huge problem. And then for PCR diagnostic techniques, it's a lot better. So upwards around that 70%. And this also is supported by this kappa value, which just generally is an assessment of labs with duplicate samples. One means a complete agreement of test result between two labs, and 0 is chance. So we're getting close to chance on our diagnostic test results on duplicate samples from different labs. So what I want to stress here is that pathogens are found in, the same pathogens are found in diseased herds as well as healthy herds. And I think it's potentially not likely to find 100% of the pathogens in all the diseased individuals. So this was really our impetus to go back and revisit strain typing techniques on all the pathogens that have been concerned for bighorn sheep in the past 10 or 15 years, and take more of a landscape scale approach. What we mean by this is getting reference isolates from different die-off events and also in healthy herds, and compare past and new strain typing techniques to see if there was a pattern. And we did this with two goals in mind. And one is to improve the diagnostics at the Wyoming State Vet Lab, and then also to look at these strain typing techniques to see if there was an association with disease. So for this talk, to make it manageable, I am going to talk about Mannheimia hemolytica, or Mannheimia in general. So in this talk, we're going to talk specifically about 62 isolates. And that can include species that have been identified as Mannheimia hemolytica, glucosida, ruminalis, or verigina. 4 specifically are from a Laramie peak die-off that occurred in 2019 and 2020. And then the strain typing methods that I'm going to look at specifically for this talk are whether there's leukotoxin present, the 16S gene coding, some MLST typing, and then also I would like to introduce biotype. And the difference with biotype is that it's a functional assessment where all of the ones previous are genetic. So In the past, we've looked for a leukotoxin gene, and typically we only were concerned with whether it was present or not. And I think it's really important to stress here that just because the leukotoxin gene was present in some of these pathogens doesn't mean that it's being produced. A lot of times there's other signals that turn on genes. So just because it's present doesn't mean that it's actually producing any leukotoxin proteins. The 16S gene is used a lot to classify species to proper ID. It is a core gene. It's found in all bacteria. It's about 1,500 nucleotides long and consists of variable regions and conserved regions. And I have it indicated as just one gene here, but there's multiple copies throughout the genome. And then also there's multi-locus strain typing or sequence typing, sorry. It's often referred to as strain type. This usually consists of 4 to 7 housekeeping genes that are found in all of the isolates of a certain species, but these genes can vary, or the MLST genes that are used vary between species. So it's really not comparable across species. And they're typically not virulence genes because it needs to be core genes that are used in this. And the strain typing method that I'd really like to introduce is biotyping, which is commonly used in most diagnostic labs. It's called mass spec or MALDI-TOF. This can be, to explain it real quick on this figure, I wish I could go over there and point it out, but these peaks, each peak is a protein. The peak height is essentially how many of those proteins are being produced. And then on the x-axis, it is the size of the protein essentially. So that's how it differentiates different proteins. So it can look at the cell and determine what proteins are being produced and then how many. And that is used in a big database to compare to known isolates to ID species. And then it also can be used for subspecies classification. So what we did for this talk at the Wyoming or at the University of Wyoming and Wyoming State Vet Lab, we use whole genome sequencing. So instead of getting just parts and pieces of the genome out, we take the whole gene coding, which is like 2 million nucleotides long. And then from that, we can pull out the genes of interest. This is done through a bioinformatic pipeline that we can talk about later. For the phenotype methods, we can use these diagnostic assessments in replicate and merge them to create these profiles called MSPs. And that can be added to the database of known isolates to ID future unknown samples, and then also model protein expression between different MSP profiles. So if you have profiles from diseased individuals and healthy individuals, you can compare which proteins are being expressed. So for the results, this is a phylogenetic tree modeling evolution on the 16S gene of these 61 Mannheimia isolates. Now, I told you guys that there's 4 species here. I'd just like to see a show of hands. Who sees 4 distinct groups? A couple? Maybe? Like 3, 4 hands? Okay, oh, before I forget, all of these, except for this long line at the bottom, all of these had the leukotoxin gene, by the way. So let's add some information. This first bar here is based on the 16S gene identity to known isolates. So blue is Mannheimia hemolytica, green is Mannheimia glucosida, and the other ones are ruminalis and berigina. This should be the first troubling thing that we see today, is that based on the 16S gene, which is used to identify these species, they don't break out into distinct groups, and that there's blue and greens kind of scattered throughout this entire chart. This is really problematic because when we're getting test results back that say Mannheimia hemolytica or Mannheimia glucosida, I'm saying that we're really not confident on what we can call species correctly. Let's go into this a little bit further. I don't want to get into the weeds with this MLST typing right now, So the colors are just assigned randomly as MLST types that are known. And basically the only known types are located in the top of this tree. So let's zoom into that. So I mentioned that there's 4 from Bighorn sheep die-off. They're located right here in the top. It's pretty interesting to note with this herd, we were able to sample the sheep prior to the die-off and then post die-off. The isolates that we recovered prior to the die-off are located down here in this grouping. And then after we stopped seeing diseased individuals, we also recovered that lower grouping of Mannheimia or hemolytica or glucosa, whatever you want to call it. And then all the diseased individuals had this identical strain at the top. So it's pretty interesting. It also matches with 3 other isolates that I've recovered. And we'll look at those. So interestingly, one identical isolate is from the 2008 cattle associated die-off in Colorado. Another one is from 1993 in Shell Canyon, Wyoming, which is a failed reintroduction effort. Those sheep came from whiskey right after the whiskey die-off. And then there was also a domestic sheep grazing in the area that had an identical strain, according to 16S. So let's talk about a similar graph or figure using MALDI biotyping. We're going to go a little bit faster through this one. So again, first bar is the species ID based on 16S. So there is a little bit better grouping, I would argue. There's more greens at the bottom. Again, all of the blues are clustered in the top clade. And then there's a mix of the middle ones. Whether we called them appropriately or not is still debatable. We'll zoom in to the top again and add in our epidemiological information. Laramie Peak die-off, again, all clustered together with the animal with pneumonia from Shell Canyon, and then a representative isolate of the Colorado cattle associated die-off as well. So again, really, really interesting, and it's pretty fascinating that we can use MALDI to identify these potentially virulent biotypes. Two-second delay, right? And then again, the animals that we recovered or isolated Mannheimia before and after the die-off are located in a clade lower down. So the outcomes from this so far, there is strong evidence to suggest that there is a virulent biotype that we are detecting with MALDI. I'm not so sure that we can reliably differentiate the Mannheimia species with MALDI. I think there needs to be a lot more work done, especially on the genetic coding to differentiate these species and call them appropriately. But I do think that MALDI is a better tool, and it's more clear these virulent biotypes from representative die-offs throughout the West. And in addition to that, we are also, all these are profiles in our database now that we can use to reference unknown samples in the future. So looking forward, so this is a small portion of what I'm doing. We're also doing it on Pasteurella multocida. These are some sinus tumor individuals collected throughout the past couple of years. It is interesting to note that every individual in this lower clade either had a sinus tumor. If it was not diagnosed with a sinus tumor, it had chronic pneumonia, and that included lamb mortalities. All in this lower clade. These upper clades had just healthy individuals as far as we could tell, apparently healthy. So there is definitely a trend here that we're looking into farther. We are also doing this with Biberstein chalosi and developing methods in order to do this with Mycoplasma ovipneumonia. The problem with MALDI-TOF is that you have to culture it, and Mycoplasma ovipneumonia is extremely difficult to culture. I mentioned before, we also have the whole genome sequencing. So we're going to look into that a little bit farther, use some proper techniques to make sure that we're classifying these organisms right. and then reevaluate how the species break out in each one of these figures, and then potentially find genetic rationale for virulence. So the take home is 100% detection of a pathogen in a die-off is not necessarily probable. And I think Molotov is useful for IDing pathogenic strains, specifically in the Mannheimias, or not Mannheimias, but Pasteurella family. And that we need to do more work to make sure we're properly IDing these isolates. So that this is a huge coordinated effort, and I really appreciate everybody that's helped out along the way, and several funding folks have been great as well. So do we have any questions?
Thank you. That was super interesting. And one thing that makes me curious about is this idea of co-infection. And I'm wondering if you could speak to whether you had found other pathogens, and whether any of the ones that they seem to group together in terms of the virulent biotypes, but were they infected with other things?
Yeah, they certainly, some of them were, especially in the Laramie Peak die-off. Some were co-infected with Mycoplasma pneumonia, some weren't. So it's hard to say there. Some of those samples were collected in '90s, so we weren't looking for some pathogens then. And that's a big part of another part of my research, is looking at microbiome. So assessing the proportions of bacteria in each one of these samples. So that's also in the works.
Great job, Casey. I wanted to ask you if you could you talk a little bit about the possible interaction between the genotype data, which you have, and your phenotype data, and the idea that phenotypic plasticity could actually change, how to phrase this. Do you think you could predict virulent types before they're actually virulent? Or is that going to switch over time?
I think that's a hard one, and I think that is different for each pathogen that we talk about. What I found most interesting about this is that those Mannheimias that were associated with pneumonia in animals, acute pneumonia, they all had identical 16S genes, which is crazy because there's hypervariable regions that should mutate a lot, especially over 20 some years. You know, the one was collected in 1990, the other ones were collected as recently as 2020. So I think there could be ways to detect. It looks, there might not be that much genetic mutation in Mannheimia hemolytica or this pathogenic strain that might be detectable using genetic methods rather than looking at phenotype. Does that answer your question? Okay.
Excuse me. Have you thought about doing a phylogenetic tree of pastorellas from bighorn sheep, domestic sheep, and cattle and goats to sort of see if there's any actual bighorn sheep types?
Yeah, phylogenetic tree on like the 16S gene or
an MLST.
or an MLST. So included in that, in those 61 samples are mountain goats. There's domestic sheep, there's bighorn sheep. There did not seem to be host specific sequence types.
I know, yeah, you're probably aware that there's papers showing that there are host specific types between domestic sheep and cattle.
Yeah.
And so maybe with a larger sample size, particularly of domestic animals, you'd be able to.
Right. I think certain strains or certain sequences are pathogenic in certain hosts, but I think they could be recovered in multiple hosts without causing disease. That's what I've read.
Yeah, I think that isn't that sort of the theory with domestic sheep and bighorn sheep. Yeah.
Right. Our next speaker is going to be Karen Fox. She's a veterinary pathologist with Colorado Division of Parks and Wildlife. She completed her Doctor of Veterinary Medicine and PhD in pathology residency at Colorado State University and is a diplomat of the American College of Veterinary Pathologists. She's been with Colorado Parks and Wildlife in Fort Collins for 9 years, and she focuses on necropsy and diagnostics, including work on bighorn sheep respiratory disease.
Thanks, Jessica. I am so grateful for the opportunity to be here today. And I'm really excited to talk to you about this diagnostic test that we've developed for bighorn sheep respiratory disease pathogens. But first, I want to make sure I acknowledge my co-authors. Kevin Blecka is a biologist with Colorado Parks and Wildlife, and some of his collar data is what inspired creating this diagnostic test. I want to thank Kevin for his patience with me as I worked on this. And I also want to thank him for allowing me to share his data with you to help you understand the results of this assay in the context of his project. Chris McGlover, who you just heard from Wyoming State, and Mark Stingline at Colorado State are people who have really massively helped me with the bioinformatics for this project, and I literally could not have done this without them. I also want to acknowledge that this project was fully funded by Colorado's Parks and Wildlife's auction and raffle funds. So thank you for that funding source. So as I mentioned, this project really stemmed from some data that Kevin Blecka presented at the sheep and goat meeting in 2020. And this is his data where he's showing that he has some collar positions that are clustering around a flock of domestic sheep. This is a small hobby flock in a small pen. And you can see his collar data are showing locations within 200 meters of the pen. These locations are collected every 4 hours. There's plenty of opportunity here to have some level of contact between the domestic and the bighorn sheep. But I think what really made him want me to look at this is this data, and this is an animation. I'll start in just a second. And if you watch RAM number 20, which is the yellow blur, and again, this is Kevin's slide, you'll see, go ahead and start it. You'll see that this ram has an attraction to this hobby flock. And over a 3-month period, it makes these trips back and forth until at the end of this animation, you'll see that ram, maybe it's not gonna happen, but it shows up dead next to this domestic sheep flock. Oh, it's going again. There he is, he's dead. Sorry that worked differently than in the practice. So Kevin really wanted to look at pathogens between the bighorns and the domestics. And so I told him if we were going to do this, we were going to need samples so that we could compare pathogens between the bighorns and the domestics and also within the two flocks or the herd and the flock. So he went to this producer and he was able to collect samples from this domestic sheep flock. He was able to have one of those animals euthanized. There was a ram that had some chronic upper respiratory disease. So we have necropsy samples from that animal. And then he collected swabs, tonsil and nasal swabs from the other 2 ewes that were in this little flock. So we took those samples and we looked at them with this assay that I'm going to present to you. We also did the gold standard, which I think of as being the Wyoming Game and Fish culture and PCR, especially for the Pasteuracea. And we used both of those methods to look at these samples. And the way that we decided to approach strain typing was through an MLST, which Chris was just talking about. I think most of you are probably familiar with MLST because this is the approach that's been used to look at strains of Mycoplasma open-pneumoniae for several years. And what we really wanted to do was expand that ability to look at some of the Pasteuracea. And the pathogens that we wanted to include in this assay were Mycoplasma open-pneumoniae, as well as Mannheimia hemolytica, Biebersteinia treholosi, and Pasteurella multocida. In addition to those targets, I also have primers in this multiplex PCR for the leukotoxin gene of Pasteurellacea, as well as the 16S ribosomal RNA gene. So I put all of the primers for all of these targets into a single PCR reaction. And I am not going to go into a lot of details about the pre-processing or the sequencing or the bioinformatics, because Kevin Blackett told me those slides were eye glazing. But if you are interested in these things, I promise you, I would be incredibly happy to talk to you about it afterwards, because I think it's very exciting and fun. But so we have to process those PCR products for sequencing, do the sequencing. And the only thing I'm going to say about the bioinformatics is that I'm using the Genius software package and I'm creating a workflow. And by creating that workflow, it allows the analysis to be automated. I am going to spend some time going over the results from Kevin's project, just to give you some context for what you can do with this assay in the context of this project that I think is meaningful to a lot of you. So looking at these samples with both the culture and PCR done at Wyoming Game and Fish, as well as with our multiplex assay, we found the same thing, that these groups of samples had these pathogens present. And there is evidence here. If you look at that bighorn ram that died, he has Pasteurella multasta and Mycoplasma ovipneumoniae, which his healthy herd mates that died that same year did not have. So there is some evidence that something was introduced and that could have been responsible for why you saw that sudden pneumonia event. And we do see in those domestic samples that multasta and ovipneumonia are in those domestic sheep. So potentially that could be the source of those pathogens. But without strain typing, you really can't say whether those are shared strains. I'm going to start with Mannheimia hemolytica. It was only found in the domestic sheep samples, so I don't have a comparison with the bighorns. But just to give you a look at what these phylogenetic trees look like, because these are very small sample sizes, they might look a little bit different to you than other trees, but all of the components are the same. And if you can read the labels, anytime you see CODS, that's a domestic sheep sample, and then the COBHS are going to be bighorn sheep samples. And in this case, like I said, they're all domestic sheep samples. They are all actually identical. So this is a single strain in that flock. If we look at Biebersteinia trehalosii, we found that present in all 4 groups of animals: the sick and healthy bighorns, the sick and healthy domestics. When we look at those with the phylogenetic tree, we can see that there is 1 strain shared among the bighorn sheep samples and 1 strain shared among the domestic sheep samples, but we do not have a shared strain between domestic and bighorn sheep. If we look at Mycoplasma ovipneumoniae, that was present in the sick bighorn, the sick domestic, and 1 of his healthy herd mates. The phylogenetic tree shows that the samples from the sick bighorn have a single strain of MOV. The samples from the domestic sheep have a single strain of MOV, but there is no shared strain of MOV between the bighorns and the domestics. Finally, Pasteurella multasta, here, let me just back up. Pasteurella multasta was found only in the sick animals, so only in the sick bighorn that died from pneumonia and the domestic sheep with upper respiratory disease. If we look at the phylogenetic tree, the bighorn samples have a single strain of Pasteurella multasta. The domestic sheep probably have 2 strains, 1 in the sinus tissues, 1 in the tonsil tissue, but those strains are unique from the strain in the bighorn sheep. So again, no evidence that those are shared strains between the bighorns and the domestic sheep. I mentioned that we have the leukotoxin gene in this multiplex, and this is what the results look like. We do not use leukotoxin as a strain typing gene, and Chris talked about that a little bit already. But what we can do is I'm using a neighbor joining tree just to help you see when you have a leukotoxin sequence, does that sequence cluster with a Biebersteinia trehalosii, like down in the right-hand corner, or a Mannheimia hemolytica, as you see in the upper left-hand corner, so you know which of those bugs that leukotoxin gene likely came from. And although we don't use a leukotoxin gene for strain typing, I think it's interesting to look at these Biebersteinia trehalosii down on the bottom right. Sorry, I pushed the wrong button. Down on the bottom right. that even just looking at the Bieberstein trait halluci leukotoxin gene, these domestic sheep samples are still forming a little unique group. So more evidence that we don't see strain sharing between those bighorn and domestic sheep. And so even though we didn't find shared strains, I thought this was a really unique opportunity to take some high resolution movement data, collar data, and pair it with the strain typing data. And if this is something that could be repeated in different situations, it could help us understand under what circumstances is there adequate contact to transfer pathogens, and that could help assess risks. I want to quickly talk about, you may have remembered from the slide where I talked about what primers were included. It was really important for me to include 16S rRNA primers in this assay because if this is going to move away from cultures, it's also going to be moving away from that non-biased approach to looking at the bacterial community in a sample, which is what culture can do for you. You put it on a plate and you see what grows. Versus PCR, you're guessing oh, it's there and trying to see if it's there or not. So by doing the 16S analysis, you can almost do like a surrogate for the culture where you're taking this non-biased approach using primers that pick up essentially all bacteria and seeing that bacterial community. When I do this with the Genius 16S biodiversity tool, the result is actually a hyperlink, and you click on that hyperlink, it takes you to a website, and you have a pie graph that's interactive. So you can click on different sectors of that pie graph and expand and contract to look at more details. I couldn't get that to work for the presentation, but I'm happy to show anyone what those hyperlinks are like. But just an example, this is the 16S data from the lung of that bighorn sheep that died. A couple of things that jump out is there's a ton of anaerobic bacteria here. More than half of what's here is anaerobic bacteria. That probably shouldn't surprise me because I'm pretty sure Tom Besser has described this phenomenon before, but I still was pretty floored by how much anaerobic bacteria are here. The things you expect to be here are here. Here's your Pasteuracea, mycoplasmas right here. And then it's nice because you can expand and contract these areas and see things like Moraxella, Histophilus, things that you might just want to keep an eye on and see where they are. Are they in the upper, the lower respiratory tract? So I think this is a nice tool to have as part of a standard diagnostic workup. The other thing that I like to do with the 16S is you can compare samples from different tissues from the same animal. So this is that dead bighorn sheep, and we're looking at the lung, tonsil, and sinus lining all have essentially the same bacterial community. That's in comparison with this domestic sheep that only had upper respiratory disease. And the sinus exudate looks very similar to those tissues from the bighorn sheep with that more homogeneous type of community. And the lung and tonsil are a much more diverse community. You would probably consider that a healthier community. And I think this is interesting from an academic standpoint, but I also think you can use these kinds of data to help you guide your diagnostics. So if you're trying to answer a particular question, where you take your sample, whether it be from the nasal swab or your tonsil swab, is going to help you answer your question more effectively. Because I couldn't show you that interactive portion of this tool, I just wanted to show this is where I've expanded the Pasteuracea sector for the bighorn sheep and domestic sheep sinus tissues, and the kind of things you might do this for is to see, while both of them have Pasteurella multasta as the predominant species in the Pasteuracea community, in the bighorn sheep, the next biggest is Biebersteinia, whereas that domestic sheep, the next biggest is Mannheimia. That's pretty consistent with what I think a lot of people tend to see, which is that bighorns have a lot of issues with Biebersteinia, whereas our colleagues that work with livestock really are looking at a lot more like Mannheimia hemolytica. In closing, I guess I just want to say that I presented these data in the context of a small sample size from a specific project, but this assay is designed to be used, and I think it can be used in much broader situations. And so I'm envisioning a phylogenetic tree of all of our herds that has all of our strains and helps us see patterns and understand connectivity and movements. And hopefully you can kind of envision that too, even though these trees I presented are really limited. And if you do have any interest in using this as a test, I would like to approach some labs about bringing this on to make it commercially available. But one thing they're really going to want to know is what's the desire to have this test? What can they expect for a caseload? I'm not looking for anyone to make any commitments, but would love to have some conversations if you are interested. Thank you for your attention so much. And if I have time, questions? Yeah, so when I run it, it is under $200 per sample. I don't know what a lab is going to charge. There were 3. Mike.
I'll use it. So what is your theory of not picking up the same strains of those bacteria from the dead ram and the 3 domestics of what killed that ram? Did we miss something or did something else contribute?
So I think it would be helpful, and Kevin and I would need to see what we have for other samples. If we could find any other sheep from that herd that had Multocida and MOV historically, it would be nice to see if those were the same strains that we have in that ram. If that's the case, it might be a case of a chronic respiratory disease herd that just happened to have a pneumonia, a ram that died from pneumonia. It could have been that that ram picked up some bugs from somewhere else that wasn't those domestic sheep. And it could be, this is bacteria. There are still a lot of other pieces to the respiratory disease puzzle, including viruses and parasites, and those haven't been looked at all in the context of this project. So I think it's important to remember that there's a lot of factors involved, and we have to do these kinds of things and not make assumptions based on what we're seeing with the movement data.
So are some of those callers of Kevin's tracking still alive online and collecting data?
Yeah, I think he still has some S52 callers out. He might be online listening.
Hey, Karen, Mike's question brought up a question I had, which was typically doing the MLST for MOB, you can't only detect one strain. You just detect the dominant strain. Have you been able to resolve that?
No, the way I have this set up, I can only detect the dominant strain.
Next up is Sydney Brinkus. Sydney graduated from Iowa State University with a bachelor's degree in animal ecology and is currently finishing her 2nd year as a master's student at the University of Memphis in Tennessee, which is focused on bighorn sheep in Montana.
The end of it? Oh, I see. Okay. Can you guys hear me? Awesome. All right. Hi, everyone. I'm Sydney Brankus from University of Memphis. So a lot of you are calling me Memphis or Tennessee at this point. And I am studying sheep in Montana, which is why I am relevant at this conference today. And I'm going to be talking to you guys about bighorn sheep respiratory disease via animal behavior and community science. Maybe. So as humans continue to alter the environment around them in many different ways, such as urbanization, which is pushing animals into small pockets and increasing stress, climate change, which is increasing encounters to different diseases, and domestic animals, which is increasing spillover from those domestic animals into wildlife, we're seeing an increased importance on wildlife diseases and the study of them. So I studied bighorn sheep respiratory disease. And as a lot of you guys already know at this conference, that comes from spillover from domestic animals, environmental pressures, and airborne transmission from wild sheep to wild sheep as well. And as the people before me were talking about, this is a multi-pathogen infection. And so some of the causative agents of this disease are Mycoplasma ovodomoniae, Pasteurelliaceae species, and a couple others that are being looked at. Once these pathogens do get into populations, we do see large die-offs of all ages, and then we do see chronic reduced recruitment of lambs for up to decades afterwards. Another thing that you guys probably already know if you're here is that bighorn sheep are really iconic species and important to study. And this can be either because of viewing or for hunting reasons. And then while there was extirpations of a lot of ungulates in the early 1900s, bighorn sheep really struggled to get their numbers back due to this disease. Right now, we have a lot of testing for bighorn sheep respiratory disease in different ways, such as PCRs, culture tests, even antibody tests. And this can be really stressful to the animal, whether that's the capture process or the actual testing process. And stress increases susceptibility to disease. And this can also be really logistically and financially difficult for managers. So what my project is looking to do is add additional tools for looking for the prevalence of this disease. So the two objectives that I have during my project is one, which is developing a behavior-based surveillance program. And then second, using this to see if community scientists are viable for collecting this data, because we don't want to add a lot of time or money to looking for the disease. So the way that I did this is I had a couple different study areas along a gradient of disease prevalence. So on one end, I have a herd that has never been exposed to the disease. On the other, I have a high prevalence area, which isn't where they're sick and dying, but just that they have some of the pathogens in their systems. And then we wanted to look at a couple of other areas along this gradient. So we're looking at a recovered herd and a recovering herd as well. And so, like I said, I am studying in Montana. So a couple of my study sites are Glacier National Park, which is our no disease detected because bighorn sheep respiratory disease hasn't been detected within park boundaries. On the other end, I have a high disease prevalence. And so I'm also working at the Little Belts. And so there's going to be a talk later about Little Belts just to explain a little more about that. But diving in, some of you guys know that sheep were translocated to restart a population in this area. So about 83 sheep were moved in January and December of 2021. And these animals came from the Missouri Breaks area. The important thing is that all of these sheep have collars and they were all tested for different causative agents of bighorn sheep respiratory disease. And so looking through that data, I found that 29 out of 50 of the examples I was given tested positive for Pasteurelliaceae species. And so this is going to be a really powerful metric for the behavior side because I can link exactly who tested positive to each individual. So when looking at behavior, that's just a very powerful metric, especially compared to some of these other herds I'm looking at. So just to describe a couple of my other areas, I am in the Sun River Range as well. And so my herd that I'm calling recovered is Castle Reef. And then I have another area in a hunting district right next to it, still in the Sun River area, which is Fairview, and we're calling that recovering. And while these herds are super close together, it's important to note that there is a river as a barrier that separates the two. So I am looking at these as two different herds. The reason that I picked these areas to look at, as some of you might know, is that with looking at the lamb recruitment, so the lamb ewe ratio, they had a relatively stable recruitment rate going on up until 2010. And so that's when we saw a bighorn sheep respiratory disease outbreak in that area. And while my Castle Reef herd, the recruitment levels went back up to relatively stable numbers. Our Fairview herd did not, and it's still going pretty up and down from there. So just to bring you back to my research objectives, I'm going to focus on my first project right now, which is developing a behavior-based surveillance program. And so in order to do this in the field season of 2021, so last summer from June to mid-August, we hiked and backpacked all of these areas, so Glacier, Castle Reef, Fairview, and the Little Belts, for 4 to 5 days at a time looking for our sheep. And so these are pictures of all the different areas that we were in. While we were doing this, we were locating sheep either using historical numbers and GPS locations or current GPS locations, and then also VHF when we were actually on the ground looking for them. Once we did find them, we were doing behavior observations through a scope. So kind of like the FWP technician, Zach, that we borrowed every here and there. We were setting up that scope and watching them from pretty far away. And I do want to note that we were pretty far away. So even though there is a dog in this picture, we don't believe that was doing anything with the behavior. And he only came out once or twice. So what our behavioral protocol looked like when we did get observations on these animals is that we were first classifying and counting all these herds. So determining who was a ram, who was a ewe, who was a lamb, and then later in the season, who also was a yearling to help with some of those recruitment numbers. And once we did that, we would do behavior focals. So this is when we look at an individual animal for 20 minutes straight, noting all of their behaviors. And so when we were doing this, we were looking at ewes, but we were also having a main focus on those lambs. And the reason we were looking at lambs is because they're the most indicative of bighorn sheep respiratory disease signs and symptoms, with some of the signs and symptoms being up here. So coughing, head shaking, drooping ears, things like that. And for an extreme example, some of you guys may have seen this from the 2020 conference, but this is one of the lambs that was in the Wild Sheep Foundation's documentary on wild and wool. And so you could see this lamb has respiratory distress, so it's panting. And so you can see this in some of the lambs that are sick. Once I did collect all my behavior data, what I did was I categorized it into 2 different categories, so either active or inactive. And so on the board is a list of all my active behaviors that I saw. So feeding, interacting, moving, playing, anything that meant they were not being inactive, essentially. I did want to note that standing doesn't really seem like a very active behavior, but when we ran standing in active or inactive, we didn't see a huge statistical difference or any statistical difference. So we felt comfortable putting that in the active category. With inactive being laying down, because that's very inactive of them. And I do want to note, we saw a couple clinical signs that you would expect, so coughing and panting, but we didn't see them often enough to do any kind of statistical analysis on them. And my goal was to see if this varied between the gradients, so all the different disease status that I was looking at. So when I graphed this, just really quick to orient you to how I did it, was I have on the x-axis all the different disease prevalence levels. And then on the y-axis, it's the proportion of time inactive. So that's actually the amount of time within those 20-minute focals that the lambs are spending inactive. And just for some of you who are more geographically oriented, I did include the map as a key. And then all of my bars are going to be the same color as what showed up on the map. So as you can see, for my first 3 herds, no disease, prevalence, and then recovered and recovering, there was some variation, but there was no statistical difference between the average inactivity level among those lambs. With my third area, which is the Little Belts, we did see a really high level of inactivity, and that was statistically different from the rest. And so that's kind of what we're continuing to look into. And so we found that inactive behavior does correlate with disease prevalence. However, I've only done 1 field season. This is all preliminary data. And I am going back next summer to continue collecting data about this part. For my second project, I'm going to use what I found in my first project to test the viability of community science collecting behavior data. And so just to orient you to community science, it's the same thing as citizen science. And Tabitha Graves gave a good talk on it on Tuesday. And so that's just the collaboration between the general public and scientists. So that could be in data collection, data interpretation, even data analysis in some examples. And so my example is Jamie Belt's GOAT project, which a lot of you have heard about. And that was where we saw community science work in a very successful way, where they were able to collect viable data. So what this looks like for my project is I'm going to be working with Glacier National Park on recruiting community scientists. Then I'm going to be doing a training program with them. So it's going to be a full day of hands-on learning where we're inside and outside, teaching them how to use equipment and look at behavior with all the scientists they're going to be working with. Once they're fully trained, we're going to take them out into the field and we're going to do mixed biologist teams. So we're going to have a volunteer with a single biologist to do focals. And so once we have all those focals that match each other, they're watching the same animal and they're not allowed to communicate about what behaviors they're picking up on. We're going to compare the length of behaviors recorded between the volunteer and the biologist. So in 2021, I did collect a little bit of preliminary data on this, and I found that biologists and volunteers do fall within 5% of each other when recording these behaviors. But also keep in mind, this is only 7 paired observations. And so what I'm looking to do is repeat this in 2022 with a bigger sample size. So I did run a power analysis of 80% to find that I need exactly 28 volunteers in order to see if there really is a pattern going on with that viability. So in conclusion, my first project, which is developing a behavior-based surveillance program, we did find that inactive behavior correlates with disease prevalence. And then with my second project, while we have done a little bit of preliminary data collection, so we have some trials, we are going to want a bigger data set for next year. And I'm going to be working with Glacier National Park in order to recruit volunteers and collect this data in 2022. So while we currently have different ways to already test for this disease, like the pictures up here, I do think it could look like this, where we do have an extra tool in our belts for managers and parks. I have a lot of people to thank for this. A lot of people have collaborated with us. So yeah, just my lab, USGS has helped us with funding, with some data collection. FWP has been an awesome help, especially Brent Lohner. So yeah, thank everybody for helping me out with this. If anybody has any questions, and if anyone has questions and doesn't want to speak, you can email me as well.
Did you have much how much interest did you have in the community for doing those surveys? And do you think that you'll be able to keep the community interested, like to do the surveys over time if you do see that it could lead to some useful data?
Yeah, so with my preliminary data, that was just random volunteers that wanted to help us. But we're working a lot with Glaciers Park, and they have an ongoing community science project already, working with loons and goats. And they've been pretty successful in adding volunteers. You could ask Tabitha Graves if you have more questions about that. But it seems like there's enough interest where we can get volunteers year after year.
Yeah. Oh, interesting talk. Thank you. I was wondering, when you look at these animals, you said you were out from June to August or so. Did you, in your analysis, look for confounding variables such as, there could be loads like temperature or even the day within, so, just to make sure that the thing that we pick up, the difference is really a difference and not due to some other factors which you did not control.
Yeah, definitely. That's a good question. So I did look at a couple of different things, and I can look at more as I go, but I looked at temperature to see if that had an impact on inactivity for lamb level. And we didn't see any kind of trend line or correlation between temperature and inactivity. And I also looked over time to see maybe if that was a period of time that the lambs were more inactive for some reason. And there was also no trend line going on with that. But if anyone has good ideas for me to test moving forward, I did collect a lot of data, so I can look at more things if, yeah, people have questions.
Okay, Emily Almberg asked, how do you distinguish between pathogen presence and disease status?
So the way that me and my advisors have gone about that is that pathogen prevalence is just when the pathogen is coming up as positive on some of these PCRs and these cultures. Whereas when a disease is within the population, you expect to see sick animals that are having different signs and symptoms, and maybe some dying off from it too. So that's what we used.
Yeah. Then you also had another potential collaborator here, Paul Roghair from the National Parks over in Badlands, said that they could get more data because they have a lot of easily viewed sheep and people like to view them. So
cool. Awesome. I'll grab your information after this.
perfect.
All right. For your herd with the high disease prevalence and the low high inactivity, do you have numbers for lamb recruitment and how many of those lambs ended up dying?
Yeah, so for the little belts, which is our high disease prevalence area and low activity area, we did have 13 out of 22 of the lambs die over the summer. So yeah.
Hi, Sydney. Nice talk. I think this is really interesting and potentially useful. And I know there are other folks who are interested in trying to determine something about disease status based on observational data alone. I was curious, do you have super tight definitions on the different symptom categories? And do you do anything more granular with coughing beyond just like coughing?
So we didn't do anything other than just record coughing. And when I say we didn't find enough for statistics, we saw coughing once or twice from animals over the summer. And so with those symptoms, it's kind of hard to have like strict boundaries when we're not even seeing clinical signs.
Okay, yeah, thanks.
Yeah.
I'm curious, you saw this association with lamb inactivity, but I'm curious if you tested whether or not lamb ratios at some time after lambing is actually more predictive.
I don't think I fully understand your question.
So I'm curious if lamb inactivity, which you showed can predict disease prevalence, as you were talking about, is that a better predictor than just good old-fashioned recruitment ratios?
So inactivity versus the ratios themselves?
Yeah.
I think that's a really good question. And I mean, this is just a really short period of me looking at this so far. So that might be something that I can look into later and just see how much it correlates with those recruitment levels. Yeah.
That would be awesome.
Yeah.
Good job.
Thank you. Thank you.
Our next presentation is coming from British Columbia remotely. And I will apologize from the get-go that I'm probably going to mess up the pronunciation of this name. But our presenter is Amelia Matthew, and she is a zoo and wildlife veterinarian based in British Columbia. And she completed her zoological medicine residency at the Columbus Zoo and the Wilds, and a master's degree in conservation medicine at the Ohio State University in 2018. Currently working for the government of British Columbia and support the Wildlife Health Program. Also co-founded the Caribou Conservation Breeding Foundation in 2020 to support the use of ex situ conservation tools for caribou.
Thank you so much. Can you hear me?
Yes, you certainly can.
Excellent. Thank you for the introduction. I'm very excited to be here from the comfort of my home office in the southeastern British Columbia. Today, I'd like to describe to you I'd say, unusual mortality event that occurred in southeastern British Columbia. Bighorn sheep in the late winter and spring of 2021, associated with the giant liver fluke, Fasciolides magna. Before I go into the nitty gritty, and I'm just going to warn you, there's quite a few necropsy pictures in here. I just want to acknowledge and thank my many co-authors, Dr. Kylie Thacker, Irene Teskey, Dr. Emily Jenkins, Brett Wagner, Dr. Brian Macbeth, Dr. Stephen Raverty, and Margot Pybus for their collaboration on this work. This was written into a manuscript, and it recently was accepted for publication by the Journal of Wildlife Diseases. So if you don't mind, I'm going to turn off my camera for now because it's a little bit distracting, but I'll be back for the question section. So first of all, who is Fasciolides magna? We've talked a lot about respiratory disease in bighorn sheep. So this is a bit different. We're dipping our toe into the parasitology world with this talk. So Fasciolides magna is also known as the giant liver fluke, or the American liver fluke, or the deer fluke. It is a large trematode that is endemic to several regions of North America, and can be found in several species of ungulates. So a very quick overview of the life cycle of this parasite to start with. So the definitive host of the giant liver fluke is deer, so wapiti or elk, caribou, and white-tailed deer. So basically these animals are infected by the adult fluke that is able to breed and produce eggs within the liver. The eggs are then carried by the bile into the digestive system and excreted into the environment through the feces. So eggs are laid down, and if they happen to be in contact with the aquatic environment, they will evolve into miracidium, which will basically migrate into aquatic snails of various species, but primarily Limnaea, where they will further develop and be released into the water as cercariae. These little friends of ours will then migrate up aquatic or periaquatic vegetation and become encysted on blades of grass into metacercariae. And then the life cycle is completed when the definitive host grazes on that vegetation that hosts metacercariae. So in definitive host, the disease is more often than not asymptomatic. They have basically evolved with it, and it causes limited clinical signs and basically minimally affect their health. However, the liver fluke can be ingested by other hosts that we refer to as dead-end hosts, because the parasite cannot complete its normal life cycle and basically gets into the liver, insists into thick-walled cysts, and eggs cannot make their way out to the environment. In general, infection of dead end host is also marginally important from a clinical point of view, except in moose, in which it has recently been demonstrated to be able to have significant health impacts. A third group of hosts, and the one of interest today, is the aberrant hosts. So here we're talking about sheep, goats, and obviously bighorn sheep. In these hosts, the parasite is very confused. it's unable to find basically the liver as a place for reproduction, and can wander excessively throughout the body cavity, so both the abdomen and the thorax, in search for a mate. And through its path of devastation will cause organ damage, which can be significant and lead to the death of the animal. In aberrant host, the giant liver fluke does not mature and will be significantly smaller. So only non-gravid immature flukes will be found. So you can see on these two pictures the difference in sizes. So an adult will be 10 centimeters in length and more, and a immature one can be as small as 1 to 3 centimeters long. So the pathology caused by the liver fluke varies based on the host type. So in, just like I mentioned before, in definitive host, it will basically find other fluke, make a happy home in the liver, do its thing, and basically have limited impact on the rest of the liver parenchyma, on the health of the animals. In dead end hosts, such as cattle and moose, it can have more severe impact, leading to hepatitis. And then in moose, we can have major lesions, just like you can see on this picture here, associated with necrosis, big foci of purulent material, and basically destruction of the liver parenchyma. Similarly, in sheep, and we'll go through plenty of pictures today, we can have significant destruction of the liver as well. But we won't find, such as in elk here in definitive host, well encapsulated basically cysts where the flukes can be found. The flukes will be found throughout the parenchyma without being associated with the cyst. So I mentioned the giant liver fluke is endemic to several regions of North America. And you can see here is the area we're talking about today. So this little star is in the East Kootenay Trench of southeastern British Columbia. So just at the foot of the Canadian Rocky Mountains, and it is home of the radium stodart bighorn sheep herd. So this is basically their range. So their range extends from the Kootenay National Park, the western portion of it, where they can be seen on higher elevation ground, and into the town of Radium Hot Springs, where they can see basically all over the place. So in 2021, from February to May, we had 4 bighorn sheep ram die of infection with the liver fluke. This was unusual. It was, I guess, the second time it was reported. The first time it was noted was in the early 2000s, where there was one lamb or a yearling, a young animal. We don't have much details on this mortality event, that was reported to die of liver fluke infection. But to see it on this scale, to have 4 animals was unusual for us, and first time in basically 20 years. So as I said, these pictures always make me giggle. The radium stoddard bighorn sheep herd can be found within Radium Hot Spring in a variety of environment. This herd used to be one of the largest herds found in the East Kootenay Trench, with a population that went up to about 250 animals. It was the largest, like I said, the largest herd in the area, and was even used several times as a source population for herd reinforcement effort elsewhere in the area. So over time, however, the population trend has fluctuated quite a bit, going from 250 to about 20 animals on a bit of a cyclical pattern. The herd faces, like many herds, several threats, including habitat loss and degradation and fragmentation. You can see there's an entire village that overlaps with their range, and that village is rapidly expanding and developing. They also suffer from severe winter condition. They struggle with access to forage at time when the ice layer is too thick and firm. Cougar predation, a huge one, a huge threat that is very relevant to the current news, is vehicular collision. As you can see on the bottom left there, they love to hang out on the highway, especially when there's salt. So we have quite a few animals that get hit on the highway every year. So the population currently numbers at about 120 to 130 animals. It's considered stable at the moment, but it's unsure whether or not this herd can sustain additional mortality or additional pressure from different threats. So at the bottom right here, you can see the herd. On that day, they were hanging out on the local golf course, which is directly adjacent to the Columbia wetlands. I'm sure you can see me coming here. We talked about habitat requirements for completion of the life cycle of the giant liver fluke. And one main requirement is water for the eggs to develop into infectious, basically particles required for infection of ungulates. On the top here, you can see them hang out on the golf course, and then on the left, they're just in the middle of the village adjacent to the highway. This herd, the radium stoddard bighorn sheep herd, is also very, very important to the local economy of the Radium Hot Spring village. It attracts tourists year round. They even have a festival celebrating the bighorn sheep every year. And this picture is the newly installed art piece that is in the center of the roundabout, right in the middle of town. So let's get into the nitty-gritty details of what does liver fluke infection in bighorn sheep look like. So in 1996, Foray et al. Published a paper looking at the experimental infection of bighorn sheep with giant liver fluke. And this is the only report of this infection in this species. And he showed that within 100 to 200 days of infection, bighorn sheep die. And they die of massive internal damage by wandering flukes. So we know that bighorn sheep are susceptible to infection. We just never had seen it in the wild beyond that one case in the early 2000s in the same herd. So basically, in February 2021, we start to see these rams. So on the left here, well, both sides, you can see how skinny this animal is. There's a generalized muscle wasting. There's evidence of that on the scapula, on the entire rump. This animal was weak. would spend a lot of time down. It was lethargic. It was hard for him to get up. They wouldn't ruminate very much. So we saw four animals were sick. 2 were found dead and 2 were seen with clinical signs. And by that point, we knew that we had an ongoing problem with giant liver fluke infections. And we elected to euthanize the 2 live animals to, one, stop their suffering, but also make sure that we had access to samples and that we could confirm that this disease was an ongoing process in the herd, as well as collect data and try to describe it a bit better, so that we could hopefully change our management and protect the herd better. So I said some of them were found dead. This is one that was found within the Kootenai National Park by Brian Macbeth. So necropsy picture time. So on overall, the lesions found are basically identical to those described in domestic sheep. And this is not surprising by any means. Both species are aberrant hosts for the giant liver fluke, and this was to be expected. So on the left here, just to situate you, we are standing in the abdomen, looking under the lifted rib cage into the chest. So the bottom here, you have the bluish area, that's the liver. You might be able to tell that that's not a normal liver. And then there's the diaphragm, and then what I wanted to point out on this picture were the severe pleural adhesions. So normally when you lift up a rib cage, there should be none of these stringy, tough bands linking the side of the chest wall to the lungs and the pericardial sac. And then on the right side here, we are looking down into the abdomen. On the right side, you have the rib cage, and the rump is towards the left. And as you can see, there's severe adhesions between the abdominal wall and all organs. I think a good way to describe it is it was like a cocoon. Everything was basically glued together by these tough fibrous adhesions. And it was immediately abnormal upon opening the body cavities. So here we're standing on top of the animal. On the right, you have the chest cavity. On the left, you have the abdomen. In the middle here, you can see a very enlarged liver. And it looks messy. Not because I'm a messy necropsier, but it's because there were severe adhesions between the liver and the diaphragm. So in the middle here, you can see that the diaphragm is basically stuck to the liver, and there was no way of peeling it off without, using a knife. On the right, you can see there's a lot of blood in the chest. This is not from gunshot or anything. This is a huge hematoma that was present within the lung tissue that was associated with migration of immature flukes. So this is what our lovely livers all look like. A normal liver should look like the portion right in the middle there. So smooth, pretty meaty in texture, brown. What you would expect to find in a healthy animal is not at all what was found here. So you can see there were severe adhesions on the surface of the liver that were adhering to everything that's around the liver. So it was adhered to the diaphragm, but also sometimes to the omentum or the abdominal wall itself. And then there were also large areas of abscessation, as well as hemorrhage and necrosis. Hold on to your hat for the next picture. It is not a pretty one. So on the left here, this is what the liver looked on cut section. So normally, again, liver smooth, firm, brown. This was a liquid, gooey liver, very abnormal. And then on the right here, you can see a nice collection of immature flukes that were found within the liver. These were hard to get out. Again, they don't form cysts in aberrant host, so they're just within the parenchyma. So we had to basically wash the liver and massage it to express the immature liver flukes. So we found about 15 to 20 flukes, more or less, per liver. So here, the goo. So on histology, there was marked cholangiohepatitis with, here you can see ovas. So they're degenerate, they're ovas, they're basically eggs from the trematode. Normally, you wouldn't see that, obviously. We also saw ductular ectasia as well as fibrosis. A hallmark of fluke infestation is this black tenacious pigment that was dispersed throughout the omentum and other tissues, including the liver, but also the spleen, some arterial walls, lymph nodes, basically a little bit everywhere. You have this black pigment that if you rub it, it won't rub off. You can't wash it off. It is within the tissue itself. And this is basically produced by the flukes as a waste byproduct. Our friend here, this is an immature liver fluke that was found just lying, I think, just on the surface of an abdominal organ. So we only found 2 to 3 migrating flukes per animal. They were either found like here, just sitting on the surface of an organ or within the big cloth. I have pictures later, but basically don't have in mind that it takes an army of flukes to kill a sheep. A sheep can die of the damage caused by as few as 1 fluke. They literally perforate organs and bulldoze through, and nothing stops them. So here on the left, this is an example of a huge hemorrhage. So on the top here, you might recognize who that is. That's a kidney, but it was associated with a massive clot just adjacent to it. And on the right side here, you have again a huge hemorrhage and bleeding in the thoracic cavity. And you can see on the left, the bluish area is where the liver is. It's totally obliterated by adhesions, and it's hard to find. So as I said, those gross pathology findings are identical to those described in sheep. So same thing here, the fine tenacious black pigment within multiple organs, as well as large hemorrhage in this case in the thoracic cavity, and then destruction of the liver parenchyma, and then finding multiple juvenile flukes in the liver, but also free floating in body cavities. So what happened? Why is it that all of a sudden we had these sheep dying of liver fluke when they've lived in this area forever? Well, I'd like to give you certainty, but I don't have it quite yet. This is ongoing work. We're trying to elucidate a little bit more what happened, but there's a few things that are worth thinking about. So first of all, the source of infection, it's not confirmed yet, but the most likely source of infection is the Columbia wetland. We know that there is abundant snails in there of various species. We know that F. magna is endemic to the region, even though it's never been described in bighorn sheep. It has been described for decades in cattle in the area, in elk, as well as horses. So we know it's here. So it's no surprise that it eventually spilled over into sheep, but the precise reason as to why it did and why it did in 2021 is yet to be determined. There's also I talked about the golf course that's right adjacent to the Columbia wetland. The golf course has ponds on it. It has quite a few. They have snails as well and periaquatic vegetation, and they could potentially act as a source of infection as well. So on the top here, this is a panoramic picture of the Columbia wetland. Ignore the forest fire in the background that was eventually taken under control. On the left here, you can see just the snails are everywhere. This is from the wetland. And then at the bottom middle, this is one of the ponds in the golf course that has the vegetation. It has snails and it has basically all the conditions to harbor F. magna metacercariae. One thing that I forgot to mention is that bighorn sheep share this habitat with elk, with mule deer, and with white-tailed deer. So there's plenty, if you ignore mule deer, there's plenty of definitive hosts walking around that can sustain the infection, basically, of the environment. So also, there's concerns around changes in migration patterns. So normally animals go up the mountain when it's cool, and they come down into town when it's when it's cooler, and that's sort of their regular pattern. However, recently there's a group of rams that has sort of lost this innate migration pattern, and they've become more sedentary to the village of Radium Hot Springs. So they're basically seen in town year round. And at this point, we don't know exactly why. There's a lot of hypotheses. There's a lot of pressure on the environment, on the habitat. There's a lot of rapid changes in this region of the world. So to be continued. The other thing that is worth thinking about is that it was only males that were found to be infected. We perform necropsies on every animal that is found dead, including females, obviously, and only males were found to be infected. I wouldn't look too much into that because our sample size is pretty small, and there is no saying that there's females that might have been infected, might have died of the infection, that died somewhere where their carcass was not noticed and could not be retrieved. And then same thing for only non-migratory animals being affected. It all sort of ties up together. We don't know that there's an actual association between the change in migration pattern or not, but it's interesting to think about, and it will, hopefully we can gain a better understanding of these dynamics in the future. And then based on the paper published by Fort in 1996, we know that animals die within 100 to 200 days of infection. So based on that, we estimated that they would have gotten infected somewhere between mid-August 2021 and August 2020 and early January 2021. Again, these are just hypotheses. We don't know for sure. This is what would make sense based on what was observed experimentally. But it gives us some food for thought. So what are we doing when it comes to management? So there's a lot of techniques that were developed for the management of giant liver fluke infection in domestic flocks that can, to some degree, apply to bighorn sheep. So the first one is using dewormers. So I guess this is the one tool that least apply to the management of wildlife for multiple reasons. Some of them being the limited drug accessibility and efficacy. So in Canada, triclabendazole, which is basically one of the few medications that has some proven efficacy against both immature and mature liver fluke is only available on an emergency order. So it takes real commitment to get this drug in Canada. It's feasible. We just decided we didn't want to for other reasons. So there's the main reason that we decided not to treat was the limited ease or the difficulties associated with repeated oral drug administration in free-ranging wildlife, and the concern for the creation of resistance against traclabendazole. Even though the herd is found in an area where wildlife viewing opportunities are great, committing to treat this herd on a bi-yearly basis with an oral drench is beyond challenging and is unlikely to really fix the problem if we don't remove the source of infection. So the other tool is the tool of grazing management. So what I mean by that, if we look at this tool with a wildlife lens, it would mean restricting the access of the bighorn sheep to contaminated areas. So let's say we're further down the line in this project, we have confirmed that the Columbia wetland and potentially the ponds at the golf course are infectious. Then we could take measures such as fencing to limit the access of the bighorn sheep to the area. However, this is not as straightforward as this seems, because without access to either these areas, sheep don't have a place to drink. So it would have to be done.
Emily,
yes.
I don't know if you can hear me. We need you to wrap up here. We're running over time now.
I'm sorry, I can't see my clock. Okay. Anyway, so fencing, attracting bighorn sheep to less contaminated areas by restoring nearby restoration areas. Oh, and oh, can you see my screen? Okay. And then the last one is snail control through either cutting paraquatic vegetation, burning the wetland, or using molluscicide. So again, not great, but these are the ideas. So the next steps are to do more research to gain a better understanding of this issue, confirm the source of exposure, characterize the seasonal movement of the herd, gain a better understanding of the determinants of herd health in general, as well as identify action points for disease mitigation. I'd like to thank my many co-authors as well as their institutions for this work. And if you're interested in supporting the recovery of this herd, I invite you to visit these two websites and become more familiar with the Slow Your Roll Save the Sheep initiative, which is a fundraising campaign to fund a wildlife overpass in Raging Hot Spring to decrease road collisions. Thank you so much.
Thank you, Emily. And we do not have time for questions right now, but if anyone has any questions, I do have her email address. And if you need that to get in touch with her, just let me know. So our last presentation of the morning, fifth and last, is going to move us out of the disease realm. Our presenter is Greta Wilson-Henjum, a master's student in the Man Love Disease Ecology Lab at Utah State University. She studies bighorn sheep in the Mojave Desert surrounding Las Vegas, Nevada. And prior to her master's, she spent 2 years working as a research technician with the USGS Western Ecological Research Center.
All right. Hi, everyone. My name is Greta Wilson-Henjum, and I'll be talking to you all today about evaluating ability of camera traps to accurately estimate vital rates for desert bighorn sheep. So again, I work in the Mojave Desert and 4 ranges surrounding Las Vegas, Nevada. Sorry, I don't think the slideshow is in slideshow mode. So there's Las Vegas in the Mojave Desert. So to give you guys some background on this system, desert bighorn sheep in the Mojave, quite like other or all bighorn herds, face recurrent threat from Mycoplasma pneumoniae. And so we have mixed statuses of disease in the different populations around Las Vegas. So we have some that are entirely naive to MOV and some that have faced, different levels of population decline as a result of MOV outbreaks. But in general, if you are concerned about disease in wildlife populations, population monitoring is important for detecting disease as well as managing outbreaks that are currently happening. Historically in this area, population monitoring is done via aerial survey, which as many of you know, comes with a series of challenges. It's dangerous, difficult, costly. And so as a result of these, in our populations, the aerial survey population estimates are done on an irregular temporal basis. As far as I know, our populations are typically surveyed on a 2-year basis. And so this is problematic when you're dealing with disease outbreak because you're going to want higher temporal resolution population estimates to know if you're dealing with a new disease outbreak or knowing how your populations are faring with the current disease outbreak. So in this system, we need higher temporal resolution population estimates. And so a unique feature of the ecology for desert bighorn in the Mojave is they're really dependent on freestanding water. And so many years ago, I think sometime around the 50s, a lot of the Mojave states began developing these water guzzlers to supplement freestanding water because a lot of the natural springs have been developed, extirpated, et cetera. And so this is what the guzzlers look like. They're sort of these metal troughs with a concrete skirt around them. They have water catchments that are sort of set off from the guzzler that funnel water into these guzzlers. And so starting around 2011, the Nevada Department of Wildlife and the USGS began camera trapping at these sites to monitor various wildlife use of these water guzzlers, as well as different, wanting to know different things about sheep activity at the guzzlers. And so given that there's already this camera trap set up here and we know that essentially all the sheep in these populations are going to come to water at some point throughout the summer. We think that there's potential for these cameras to provide opportunity for high resolution population monitoring. And specifically, we're hoping that we can get at monitoring lamb-to-ewe ratios throughout the year with these cameras. So my research objectives are to answer what information is needed to use water guzzler cameras in population monitoring. And so specifically, what is the detection probability of these cameras and what bias are we getting for counts from cameras? And then using the detection probability and bias parameters to ask or get at a model of predicting lamb-to-ewe ratios. So to give you all an idea of what I do in the field, again, I work in 4 ranges surrounding Las Vegas. Each of these blue dots is a different guzzler where we've had camera trapping efforts. There are more guzzlers than this and more cameras that have been set up than this, but these are the 7 subset of cameras that I would visit throughout the summers. And so these cameras are set up in the sheep range, the Muddy Mountains, which is our naive population to MOV, the River Mountains, and the McCullough Mountains. And so this is a photo of one of our sites in the River Mountains. This photo is taken from my point of view where I sit on a daily basis. And so I'll look down at a basin where we have one of the water developments. Sheep will start to come in. And so once sheep are coming down to water, I will keep a time-stamped running total of all the sheep that are coming down to water. And so a group will come in, I'll record the number of sheep, the time that they're there. And then typically multiple groups will start to come in. You'll have mixing of multiple groups. But anytime there's a new addition of a new group, I'll update our running total of sheep at the basin. And then they'll start to peel off, but just to drive the point home, I'll keep a count of how many sheep are coming and going. And then typically, you'll have kind of continuous movement in and out of the water basin, but there will be periods where the count of sheep goes back down to 0. And so I don't know if you can see the blue star in this image here. This is another one of our sites in the Muddy Mountains. And so our cameras are typically, the only thing I want to show you all from this is that our cameras are set up about 20 feet back from our water guzzlers typically. But then also there's not really any trees or much, tall structure in the Mojave Desert. It's mostly barren rock. So the infrastructure for setting up cameras is kind of dependent on previous infrastructure that's been put in the form of T-posts. So I don't have a ton of lateral movement for moving the cameras, but they were all generally north facing to get as little washout from the sun as possible. And so our first objective is, again, to analyze detection probability. We did this at a few different scales. The first scale that we did this at, and our most core scale, was we called it a bout of sheep. And so if you remember from that schematic I showed you, there would be kind of a continuous period where sheep were present regardless of discrete social groups coming in. So our first analysis was looking at detection probability of these continuous periods of bouts of sheep. And so again, since this was like a historic data set that we were coming into, there were certain camera programmings that we wanted to stay in line with. And so previously, the cameras had been set up to only trigger every minute. So they're motion triggered cameras, but there's a 1 minute waiting period. So, but for a period that sheep are present, a camera could trigger every minute that they were present. So we did 2 different analyses. Were any pictures of bighorn captured while bighorn were present? And then over the course of the duration that they were present, what proportion of time did cameras pick up their presence? And so then we also looked at detection probability as a function of environmental covariates. Our first one was the sheep minutes that they were present. So for each minute of the bout, there could be varying numbers of sheep depending on groups coming and going. So if there was 1 sheep present in a bout for 5 minutes, that would constitute 5 sheep minutes. However, if there were 5 sheep present for 10 minutes, that would constitute 50 sheep minutes. And so we summed this over the duration of their presence to get a sum of sheep minutes within a continuous period. And so we predicted that as sheep minutes increased during a bout of sheep, that the detection probability would also increase. And then these cameras are pretty sensitive to heat. And so in the Mojave Desert, in the summer, most days are over 100 degrees. They're often around the 110 degree temperature range. And so the internal temperature of these cameras can jump up to 130, 140 degrees, which can severely impact their capacity to take pictures. You start to get some camera failure at those temperatures. And if you're wondering why I didn't just put them in the shade, there was no shade. So we predicted that as the maximum temperature increased within these bouts, that the detection probability would decrease. And then finally, when we started this project, we were using cameras that had already been set up and were about 10 years old. And anecdotally, I've heard that camera function really starts to decline after about 6 years. I don't work for Bushnell, so I'm not trying to say that that is that number is law, but that's what I've been told. So in the second year, we updated the cameras from the Bushnell trophy model to Bushnell low glow model, which was just the latest model of camera trap from Bushnell. And so we predicted with the newer models that detection probability should increase. And so again, our first analysis was just how often when sheep are present, do cameras at least get 1 photo of bighorn, just very basic detection. And we found that detection probability ranged from 44% to 90% in 2020 with the older model cameras. And then in 2021, this increased from 67% to 100% with the newer cameras. And then we took a finer grain approach and looked at the proportion of time that sheep were present, how often were cameras detecting bighorn. And we found that in 2020, between 3% and 49% of bout minutes were detected by cameras. And then in 2021, this increased from 18 to 84% of bout minutes. And so then we looked at how detection probability would change with our different environmental covariates. And we found that across the different sites in different years, detection probability consistently increased with increased sheep minutes. So the more sheep that are present for longer, your detection probability should increase. And so each of these lines represents a different site within a different year, but the trend held across different sites and different years. Our next prediction was that detection probability should decrease as the maximum temperature while they're present increases. And again, we found that this held across all sites and all years. So our prediction was correct. And then finally, we predicted that with the newer camera models, detection probability should again increase compared to the older camera models. And so these x-axes are the same as the last two plots. So what I just wanted to show you is that detection probability with the newer camera increases and the same patterns hold with sheep minutes and temperature. So the next resolution of detection probability that we looked at was the detection probability of entering groups within these bouts. So again, bout was just a continuous period of sheep presence, but entering groups were the various discrete social groups that might enter and mix within a bout of presence. And so we looked at will a camera trigger within 3 minutes of sheep entering the basin? And this is just based on the time it takes for them typically to enter and move into water. And so if a photo of bighorn was captured within 3 minutes of them entering, we considered that group detected. If it was not, a photo of bighorn was not picked up, they were considered not detected. And so again, we looked at detection probability as a function of environmental covariates. Our first one being that as the size of the group entering increased, detection probability would also increase, but we thought this might vary with different demographic group compositions. Again, as temperature increased, we thought detection probability would decrease. And finally, using the newer camera models, detection probability should increase. And so what we found was that in 2020, detection probability of entering groups ranged from 13% to 70%. And then in 2021, this increased from 33% to 100%. And so we didn't find that total group size had an effect. And the only demographic group that had a significant effect on detection probability was the composition of yearling ewes in the entering group. So with no entering yearling ewes, detection probability was at about 40%, and this increased to about 50% with any number of entering yearling ewes being present. And then there was a weekly insignificant decrease in detection probability with entering yearling rams. And I do want to note that these confidence intervals are generated from the raw data. And then finally, as we predicted, detection probability with the newer camera models also increased. And so finally, we wanted to look at the detection probability of group size that were instantaneously at the water sources. So the group size was just an instantaneous count of how many sheep are present, regardless of social group. And so we wanted to know if you're seeing one sheep on in a photo from these cameras, how many were actually present. And so we predicted that this would change as a function of group size, and again, as a function of the camera model. And so what we are hoping to give managers was a model that they could run with their own data based on site and demographic groups. So we ran individual logistic regression models for each demographic group, and those were total sheep, ewes, rams, and lambs. And so what you can see here is we have the observer counts on the x-axis and then the photo counts on the y-axis. And so the observers are regularly counting more sheep than what comes up in the cameras. And so this was our model for total sheep, no demography being broken out. And so the output of this is that we created a model that when you put in the number of sheep that you are picking up from the photos with your different site parameters, you can adjust your counts to get closer to the observer data. And so we also included a random effect for the different bouts of sheep just to show the variability across just in time camera function. But using our model, we were able to adjust camera counts to get them closer to observer counts. And so that'll be our most important takeaway for managers. And just to show you, we created another model for ewes, rams, and lambs. But what the main takeaway is that managers will be able to use this model to get at more accurate, true counts based on camera counts. And so our takeaways from this is that bout detection probabilities were higher in 2021 than in 2020. Same with entering group detection probabilities. And that we have a formula for managers to adjust camera counts to get closer to true counts of sheep. And then finally, our objective for this was to make this usable for disease surveillance. And so we wanted to know from cameras, can we predict true lamb to ewe ratios? And so here on the x-axis, we have the camera generated lamb to ewe ratios, and on the y-axis, the observer lamb to ewe ratios. And while the camera counts were pretty accurate on the, this dotted line here shows like a 1-to-1 cameras regularly picking up the true lamb to ewe ratios. So our modeled line was pretty close to 1-to-1. However, there was not a lot of precision, so there's quite a bit of variability around that 1-to-1 line that cameras are generating. But we did come up with a multiplier value that managers could use when you're seeing, you know, this lamb to ewe ratio on cameras, multiply it by this value to get at the true count of lamb to ewe ratios. But this number is not very far from 1. You would only multiply by 1.015. And so with that, I'd like to thank our funders. And I'll take any questions.
I have one right here. So excellent, thank you. And this kind of relates to both your presentation and Sydney's. And I'm wondering if you guys have thought about a rate of decline in lamb ewe ratios to look at a metric of herd health. So instead of like a snapshot at the beginning and a snapshot at the end to see how the lamb ewe ratios changed, actually a rate as lambs die if there's disease in the herd. Just a thought that after putting your two together, it made me think about that.
Yeah. So I was out at these sites on a weekly basis typically. And so we did look at observer lamb to ewe ratios, just wondering, you know, that exact thing. Is there some sort of rate of decline throughout the summer? And with Mojave Bighorn, it seems to be a little bit difficult to capture because their birth pulse is so diffuse. And I do regularly see, you know, very young lambs throughout the summer. So we didn't pick up any consistent trends throughout the summer or any consistent rates throughout the different populations. What we did find was there was a lot of variation across the different sites. So it's not a super satisfying answer, but it warrants more looking into. It's frustrating.
All right, we've got a question online here. How did your lamb ewe ratio compare to the aerial survey?
So I actually do not have access to the aerial survey. Aerial survey counts. So that is not because I think anyone's withholding them from me, just because it hasn't really come up yet. But we would like to validate against that eventually. That is in the next steps.
Hey, Greta, great talk. I was wondering, maybe I missed it. Could you clarify for me how you defined presence at a guzzler? Are you using some kind of distance threshold from a guzzler to say if the sheep are present or not?
Yeah. So these guzzlers were interesting because they're surrounded by escape terrain, but then themselves are down in sort of these flat basins. And so there was not a specific like distance parameter from the guzzler, but it was more once they were off the escape terrain and down in the basin surrounding these. And that tended to be a pretty discrete basin. It wasn't like a continuous landscape. So that was the main defining parameter, but that was defined in the beginning and held constant throughout the season. And so we would also keep counts of sheep on the escape terrain continuously with the counts of sheep in the basin.
Oh, okay. Sorry if I missed this at the beginning of your talk, but how did you like aggregate the raw camera data to come up with like a LAMU ratio?
Yeah. So that was a little bit complicated. We had the running total, but so I would mark, or like we had the discrete demographic breakdowns for each social group coming in. So we knew that there were, so many entering groups with a count of lambs. And so we would only total the number of lambs from the entering group count, not like through, summing them over the total of the bout. I could sit down with you and show you the data more specifically if that would be helpful.
Yeah, thank you. Similar to the questions before, I was wondering when you look at animals and strictly at, that they're taking water, is there any probability that, for example, younger individuals or lambs do not tend to go to those water holes because they're excluded from the others, or they're just not that interested in it?
So anecdotally, I did see lambs go and try to drink water, and sometimes they were bullied off of water, depending on how hectic it was that day and where in the rut season they were at. End of July, the rams would be pretty aggressive about, keeping them off of water. There is some behavior of parking lambs away from the water that we know happens. But so that was why we wanted to run individual models for group size bias, because we did feel like each individual demographic group was going to have varying capacity to be detected by the cameras or varying capacity to even come in front of the cameras. So I'm sorry, I don't have a good like beta coefficient to give you that would be like different for the lambs from the ewes, but that is why we gave them their own model.
Hey, Greta. Awesome talk. I was just wondering if you guys thought about building a shading mechanism for your cameras at all.
They had boxes. Beyond that, not really, just in the interest of not having a ton of stuff out there. That and things tend to get damaged and stolen quite often. So I didn't, but that could be a possibility.
Yeah, I just want to make one additional push on the proceedings. If you're interested in a hard copy of the 2020 proceedings from the Alberta virtual, and we've got those. I did bring hard copies of the 2016 and 2018, if you're interested in those. And then in the break room, we've got display copies along with the display copy of a Greater Yellowstone Bighorn Sheep and Mountain Goat book. Tony, did you sell any of those? You got 2 left? Okay, those are really high quality documents. But if you want to take one of the blank order forms, go home, check what's on your library shelf, which volumes you might be missing, then get a hold of me and we'll get those shipped out. But if you're interested in the 16, 18, or 20s, let's get them to you today, and it's just something less I'll have to take home. So, all right, 10:30 sharp.
Yeah. All righty, please find your seats. We'll do our best to stay on schedule here. All right, that scares me. All right, we're ready for our next session of the morning. And what can I say about my friend Hank Edwards, our lab supervisor at the Wildlife Health Lab for the Wyoming Game and Fish Department? But he's just been a person that's been involved with everything disease related in Wyoming for many, many years, be it brucellosis, be it CWD, which are full-time jobs in and of themselves. But amongst all of that, Hank is and his team have made time to do all of this bighorn sheep work we've done. So definitely one of the highlights of my career is working with my buddy Hank. But Hank is going to moderate this next session. So Hank Edwards.
Thanks, everybody. I guess maybe you could say, since I've been involved, maybe some of this is my fault, maybe. So anyway, we have 3 talks today, and then we're going to roll the last part over to Allie. She's going to talk about a film in the last 10 minutes. So stick around for that, please. So the very first presentation will be given by Vanna Bacadori. Vanna is a Butte area wildlife biologist for Montana Fish, Wildlife and Parks. She earned her master's degree from Montana State University, studying the pronghorn population in Yellowstone National Park. She spent the last 17 years as a Butte biologist, trying her best to maintain a balance between wildlife, ranchers, sportsmen and women, and bureaucracy, with the first 3 being the easy ones. Through numerous partnerships, she's involved with several projects ranging from collared bighorn, sage grouse, and bighorn sheep, to banding songbirds, to managing a Superfund site at Mount Haglin Wildlife Management Area. So take it away.
All right. I know Kevin's right in front of me and he's monitoring the volume. So how is that? All right. All right. Well, thank you, everybody. And thanks for the introduction. I very much appreciate having the opportunity to present here today. And I want to take this opportunity also to thank Doug and his committee for putting this conference together. It's been really informative, time well spent, and also just great to kind of reemerge from all of our cocoons. So appreciate that. Some of you may find parts of this talk familiar if you participated in the virtual test and removal workshop that Mike Cox and his crew put together last summer. I presented the Highlands bighorn sheep as a case study for a potential test and removal project. Since that workshop, we did move forward with putting together a 5-year study plan to actually conduct a test and removal project in the Highlands. And we did we initiated it this winter with our first round of captures and sampling. So what I'm going to present here today is the project, very preliminary results, but really I'm going to be selfish and take advantage of this room full of expertise. Some of you I've already tapped into. But I really would like a lot of feedback if you're so inclined, because we are early on in this project and we really want to make it a success. So with that, I also want to acknowledge my partners in this project, Dr. Kelly Prophet, who's a research biologist with Fish, Wildlife and Parks, and Dr. Dan Walsh, who I know some of you know from other arenas, and he is now the unit leader for the Montana Co-op Wildlife Research Unit at the University of Montana. So I really feel privileged to have an A team on this project. So to get you oriented, the Highlands Bighorn sheep is in hunting district 340, which is here in southwest Montana. It's one of 35 sheep hunting districts in the state of Montana. It is home to what has been dubbed the king, not by me, but by other people. The king was found in the Highlands population. It was actually found in the Pioneer Mountains, which is part of the Highland herd range. And this was found in 1992 by Jack Atchison Jr., who some of you may know as a board member for the Wild Sheep Foundation. The king, to everyone's guess, probably died of natural causes. This was before we had a big die-off in the Highlands population. At the time it was found, it's unknown how many years it laid out there, but it did score 203 5/8 and aged 7.5 years. And for a little bit less than a year, it was the #1 ram, Rocky Mountain Bighorn sheep ram in the world. So again, to kind of get you oriented geographically, the Bighorn sheep population, the Highlands population is kind of centered in the middle of the hunting district. Butte, Dillon, that's about 60 miles in between there. The bulk of the herd is in the center area, and you can tell from the color coordination on the map, the yellow is BLM, white is private, and then the green is the Forest Service land. So most of these sheep are on either BLM or on private land. Oh, it helps if I press the advance button and not the laser in this poor man's eye over here. Let me go back 1 or 2 or 3. Okay, another attempt to kind of get you organized with the topography of their range. You have the Big Hole River, which comes around through the range right through here. I should have mentioned on the previous slide, I-15 comes right down through the Bighorn Range as well. And sometimes, all right. Okay, all right. So anyway, we do have I-15, which follows most of this boundary right through here. And that is a feature on the landscape that the sheep have to navigate. We have the Big Hole River, which flows through this way. And then we have the upper elevation summer range for ram groups and a few of the ewe lambs. These are the East Pioneers, the Highland Peaks, and then McCarty Mountain down here to the south. The terrain is probably pretty similar habitat-wise to what you all see in your areas. We have these large, did you lose me on the, can you still hear me? Okay. So you have large sagebrush expanses. We have these talus slopes where the bighorn rams like to summer, steep rocky terrain that's dominated by mountain mahogany. And then throughout the area, we do have a few irrigated pivot fields that definitely do draw in sheep, especially late in the summer when everything else dries up in the hills. So the history of this herd, similar to what a lot of you see in your herds, it was extirpated in the 1900s from over exploitation, market hunting, meat hunting. Sorry about that. It doesn't help that I'm partially Italian and I use my hands a lot. So, and then the herd was reestablished in the late 60s with 2 transplants that came in from source herds in Montana. And there were no further augmentations over the next 30 years. That herd grew to about 300 to 400 animals by the early 90s. The herd was a healthy size with a lot of rams in it. And so in 1994, there were 35 ewe permits and 35 either sex permits with pretty much a 100% success rate. And those either sex permits were primarily filled with rams that were 3/4 curl plus. Again, the story is similar to what a lot of you all have dealt with. 94, 95, that winter, there was an all-age pneumonia die-off, about 90% mortality, and it left less than 100 sheep in that population. The management history following that die-off was just kind of a natural regulation, just let the herd recover on its own or do whatever it's going to do. We followed that approach until 2000. And then we began a series of augmentations because that was kind of what we did at that time in the early 2000s. And so there were several augmentations. All the source herds came from Montana. We were not attuned to Mycoplasma pneumonia at that time or looking for pathogen profiles. And so we were moving sheep. And again, the source herds were from Montana, and there was about 140, I think, if I added that up correctly, sheep that we moved into the highlands to try to move them past this bottleneck. We did not see, like we weren't seeing good recruitment or population growth during that time. From a hunting perspective, we did close that season after the die-off, kept it closed for the next 7 years. In 2002, we opened that season up, kept it open for a few years, kind of watching the ram age classes and recruitment. 2007 through 13, we closed it back down. Didn't look like there was much opportunity for hunters during that time period. And then we've opened it back up since 2014. We've had 1 to 2 tags in there during that whole time. And here's a photo of some of the rams that have come out, including this one right here was the governor tag holder in, I think that was 2014 or 15. And that year it was some prince from Arabia or something like that. But he got a really nice ram. So I also was able to kind of piece together some tracking efforts with every one of those transplants. We did put radio collars on as many sheep as we could, primarily ewes. And we also put neck bands, individualized neck bands on some as well. So just kind of a, shoestring budget. I was able to get some tracking effort from 2007 through 2017. And I want to point out the highlighted, it was VHF tracking. It was not as rigorous. We never get as much sampling and tracking as we'd like. This is definitely was not as rigorous as I would have liked. It was not year-round continuous for 10 years straight. It was more kind of as I was able to get help out in the field. But the data nonetheless was still useful. From that 10 years of variable tracking efforts with VHF, we were able to come up with some sense of what the herd structure is looking like. And it looked like at that, from that tracking information that we had a meta population out there, 5 subherds, and they seem to have distinct home ranges. We did not detect much movement between the subherds with the collared animals that we had. There likely was movement between them all with the rams, of course. The production seemed to be good in all subherds. The tracking that I did have, we definitely put more effort into it from spring through fall to try to get production and lamb mortality. we did have evidence of lamb pneumonia in some of these herds. So I'm going to speed up because I just got the 5-minute warning. Okay, and there is possible commingling with the notch bottom herd down here in the south. So this just kind of gives a tabular form to give you an idea. We did some minimal testing for Mycoplasma pneumoniae, both serum and PCR in 2016. We weren't able to get representatives from all the herds, but you could see we did have positives, both serum and PCR from the foothills herd. And at that time, lamb survival was less than 20. And then the notch bottom herd, I want to just direct you to those two herds. The notch bottom herd did have some exposure to mycoplasma through the serum, but negative for PCR. And at that time in 2016, we were seeing really good lamb survival in there. If you jump ahead 5 years, I had a ground survey effort, pretty expansive in December prior to us going out to capture. And it seems like those two herds have switched, and we do have presence, and I'll show you the lab results too. That notch bottom herd seems to be kind of the sink herd now that has pneumonia and is not retaining lambs. We did many of you probably know, we did a full eradication of the tendoy herds in southeastern or southwestern Montana. That was one management action to try to deal with these herds that have chronic pneumonia and lack of recovery from it. So we did a complete removal using hunters primarily, and then brought sheep back in just last year. I wanted to do something different in the highlands because I felt like this substructure allowed us to have a more surgical approach. There's been, as you all know, you're the body of evidence here, a lot of test and remove studies that have been done and lessons learned in the field. Montana did a bighorn sheep study. A lot of, there's a few folks in the room here who contributed to that. So taking this body of knowledge, we came up with the Highlands Bighorn Sheep Project as we have it right now, where we wanted to look at a couple of management tools that might get us past this chronic pneumonia and help this herd recover. So we set up a before-after control design. Years 1 and 2, we'll monitor the disease exposure of the individuals in the herd, look at the lamb survival, and how connected are those subherds. So again, we had VHF information that led to the idea of the subherds, but now we have GPS collars out there. Do we still have that subherd structure? And then years 3 and 4 apply treatments, and years 5 and 6 monitor results. The treatments that we initially wanted to test was using that subherd structure, takes 2 subherds, do test and removal, take 2 other subherds, do selenium supplementation, and take a 5th subherd as our control. And then the desired response, of course, is to see if we can get rid of Mycoplasma pneumoniae and get better lamb recruitment and increase population growth. Winter helicopter captures, we did that in January. We are probably going to supplement that with summer capture. We are trying to capture all individuals in the herd. We didn't accomplish that, so we're probably going to back that up with some additional summer capture. We will go out there and in the next month and a half and try to do lamb captures and get collars on them so we could do cause-specific mortality. And then, of course, testing for the pathogens. So this is a preliminary summary of what we have from our lab results. Just walk you through this table in the last couple minutes. We have approximate group size for each of the subherds, the ewe lamb subgroups. It does not include all the rams that are out in the population, only the rams that are associated with those ewe lamb subgroups. And then the third column in from the left, the number of ewes and lambs that we sampled for Mycoplasma pneumoniae PCR. So you could kind of compare our sampled animals to the approximate group size and see that we did better in some and not so good in others. And then if you look at the results for the MOV detection in PCR and serum, you could see from the serum, they've all been exposed. They all have exposure to MOV. And then with the PCR, ironically, the one that foothill subherd where we had a lot of pneumonia over the years, it was not detected in the samples there, and we sampled nine U's in that bunch. The other two that I have highlighted here, the Sheep Mountain and the LaMarche, those positives were one lamb in each of the sampled groups. So out of the 12 animals sampled in Sheep Mountain, 11 of them were, the results were not detected, and then one tested positive for it. Same thing with the LaMarche group. I do want to draw your attention again to this lamb survival. So in the foothills, we now are seeing pretty good lamb survival, where back in 2016 we weren't. I think part of the answer to that, this is a ewe that we brought in 2008, and I'm not saying I'm not pointing to her specifically, but during this capture, we did capture several ewes that had old neck bands and collars on there. And I could go back to the trap and transplant records. Two of those ewes that we collared this year, or 2000, they were captured in 2008, and I could find their birth year from the age. They're 18 years old. And so I think what has happened in that foothills herd, which is predominantly members from the 2008 transplant, is we had a bunch of carriers, and now those carriers are starting to die off. So, but anecdotally, I thought that was very interesting. So if somebody asks, well, how long does a ewe live out there under natural causes? We have evidence of at least 18 years. So, okay, so if you remember our design in that first year, years 1 and 2, adjust the sample design as required. Well, it's required. The fact that all the herds have been exposed to MOV tells us that we have mixing, even though our movement data hasn't shown it yet. You know, we did not get all the individuals in each of the subherds, so we obviously have missed some of the carriers, and there's probably more movement than I suspected from VHF information. So that Kelly and Dan and I just talked before this conference that we probably are going to scrub the selenium treatment and just focus on test and removal, using the whole herd as our sample size, basically, and do 2 years of testing. In years 3 and 4, we will do those removals, removing the positives, and just see where we go from there. So anyway, the 2022 to-do list is just kind of put out there what I have been thinking needs to, some of the pieces to make this a complete puzzle. I'm going to stop there because I know I'm probably out of time, over time. But I really, again, I'm just kind of appealing to this room of experts. We are early on in this project. Some of you know I've been beating on this drum for 10 to 17 years. And so I really would love to make sure that we have dotted the I's and crossed the T's with this project. And so at any time, I welcome input from anybody with your thoughts. So thank you.
Comes from Terry Myers, asks, were steps taken to mitigate the risk of future interaction with domestic sheep prior to the initiation of the project?
No, but that is coming, because we do have potential for mixing, especially with that notch bottom herd. And that's where we have, 5 years ago, we started to see pneumonia there. So that is coming. And then I don't know if Justin Goody wants to address the separation study, to partial answer for that.
Yeah, I guess a quick synopsis is that we have situations like this where that notch bottom herd, I mean, they live a quarter mile from domestic sheep on private land. We have situations like that all over the state. And so one of the things we've been doing is working closely with Wild Sheep Foundation, wool growers, and animal science researchers as well. We've designed a study to just estimate how frequently contact occurs between domestics and wild sheep as a base rate around the state. And then obviously what we're getting at there is what is the risk of pneumonia epizootics, and is it correlated with how often that happens, given that it just happens everywhere, not just in the highlands. I mean, that's just a frequent thing. So that's forthcoming. And one of the big things just to focus on there is that is being done in collaboration with, domestic and wild sheep interests, because that's what that's going to take. Learning and doing something about it is not wild sheep advocates, wild sheep researchers. We can't do that on our own. So.
Okay, there's actually two. We'll start with the first one. Are the Highlands going to be part of the 5-year commingling project?
I think Justin just answered
Okay,
it. Yeah.
I wasn't paying
Yeah.
attention because I was reading the
So
questions.
that's okay.
Why the thought to supplement selenium?
That was just one of the initial thoughts leading into this project based on the 2016 testing. The foothill or the LaMarche and the Sheep Mountain herds it did not appear that they had MOVI exposure. And so we thought that they would be good candidate herds for doing selenium to try to improve individual performance that would lead to overall population performance. So.
Your largest sub-herd, you had like 20 sampled. What was the prevalence rates on the PCR?
I don't have that in front of me, but I do have that. So can I find you after this? Yeah. Yeah. It was individuals as adults as well as lambs. Yeah. So.
Thank you very much.
All right. Thank you.
The next presentation is by Brent Lawner from the Montana Fish, Wildlife and Park. He's a Montana Fish, Wildlife and Parks area wildlife biologist based out of Fairfield, Augusta area since 2006. And that is a nice short bio. So I appreciate that. Have that.
I don't know if I might just carry this around. I have a hard time staying still. I do want to second what Vanna said for the organizing committee as far as all the work you guys have done putting this. It is good to get everybody back together. And I'm going to have some stuff here similar to Vanna's approach. It's more kind of doom and gloom stuff with sheep. I was talking to somebody earlier. It'd be nice to have a talk where we could not talk about all this stuff someday and all the positive things, but hopefully we're getting closer to reaching some solutions. So for bighorn sheep in the Sun River area, this is two things. Like Vanna mentioned, I do have a selfish perspective on this that I'd be interested to hear people, any thoughts if I have time, related to what we're doing in Sun River area based on what's happened most recently. And part of this too is just an update from a talk I gave 12 years ago when we were in Oregon. At the time, I was fairly new to this position, and I felt pretty proud of the fact of what Sun River sheep are and what they've done and the history in Montana. And I'll get into it in a sec. Pretty quick after that, I was humbled, and now we're dealing with a die-off there. So this here is a photo. The photo up here. Which one's this photo of that old ram up there is from the 1930s. So that was one of the, as far as I can tell, early 1930s, one of the die-off events we were having in this area. Of course, decimation to restoration and back again. And hopefully we can change that in time. Here's a little perspective on sheep in Montana. I think folks are fairly well, aware of what sheep distribution has done in a North American level. 130 years ago, this was the approximate distribution. 50 years later in Montana, this was approximately what it was. 30 years later after that, with all the restoration efforts that went on, this was the estimated distribution, and this is roughly currently where we're at. The only population that's not in here is Little Belts. Sonia Anderson's going to be speaking to that later today. There is a new population right in here of Little Belts that we're working on, but otherwise these are the core populations of sheep in Montana. And the consistency for this in time has been Sun River. These sheep, historically a native group of sheep that have always been in this area. They're still there, but they've been able to survive a lot of these different events that we've had throughout Montana, obviously, and other places in Western North America. You helping me out over there? Thanks. So here's a similar photo just for reference. This is where the Little Belt area is that we did a new transplant. Otherwise, these are the areas in Montana where we have sheep distribution. So a little bit of an overview of some of the stuff I'm going to go over fairly quickly. Some of that background information, some of the die-off history that we've had with Sun River sheep. Big picture pathogen details. How we've dealt with those over time and where we're at least currently going from here. So the big thing, this is kind of a messy graph, but the big thing I wanted to point out here, this is fall survey day. A lot of it's ground survey day. We do a lot of our counts via ground surveys in this area throughout the course of the year. That black line is the one to pay attention to. It's not that we had these big die-off and low numbers. It's more the quality of the survey at the time, which really varies depending on the year, but general consistency for numbers in Sun River area. This area has been a pretty important area for hunting opportunity since the 1950s. 1950s is when we started, this area is opened back up for hunting. Close to about 1,800 RAM licenses and 1,200 U licenses provided. Of course, the U tags and the RAM licenses have been really reduced the last few years. We don't have U hunting in there. We haven't had U hunting in there for probably 8 or 9 years after the die-off hit. One of the biggest things for Sun River sheep has been the translocations that we've done out of here. So as on Vanna's talk, you saw Sun River was one of those places that was used as a source for the highlands, among other places we've taken them. So about a third or a little over a third of all the transplants we've done in Montana came out of Sun River area. So that one of those photos or the figures you saw earlier from the 1940-ish to 1970 period to reestablish a lot of these populations in Montana was Sun River sheep. Right or wrong, we might have been moving pathogens around at that time. We never even realized it, but it did help restore numbers to where we're at now. This is roughly what it looks like on a map. All the places where we've taken sheep throughout Montana from Sun River area. But the place I want to focus on here, the last part of my talk here, is these last 20 years and what's happened with Sun River sheep. Before I get into that, though, we do have known die-off events that have occurred close to a century ago. Don't know a lot of details of what caused it, what the details were, what's correlated to domestics or correlated to winter conditions, correlated to competition with other species, elk being a big one in those areas. 83, 84, we had a die off, the typical pneumonia lungworm complex with some of the pathogens that were detected at the time. Talking to John McCarthy, who was the biologist there at the time, he estimated probably about a 20 to 30% decline in sheep at that time. And then 2010, there's, as area biologists for fish, wildlife, and parks, there's about 30 of us scattered around Montana. We deal, like Manna mentioned, I think maybe it was her bio, we deal with a lot of different things besides one species. This sheep for me has been the most frustrating thing that I've dealt with since I've been here, and for a lot of people. So 2009, a little bit of timeline here. 2009, 2010, we had that die-off hit in a lot of places in Montana, Western Montana, multiple populations at the same time. In April 2010, I had over 900 sheep that I could find. These are mainly ground surveys, so obviously we're not counting. So there was a lot of sheep in that area. In June 2010, it was literally in Hood River, I got a call from our warden saying he just found a sick ram and put it down. This is that ram. And that was the first sick sheep we heard of. And then over the course of the summer period, extending into the next fall, winter, that's when a lot of these sheep started getting sick. So it wasn't a winter period, it was a summer period when it really started. Sheep looked healthy when I was doing surveys in April of that spring. And by the following year, we were down to about a little under 600 sheep. Over the next 4 to 5 years, it kept dropping, and now we're close to around 300 head with all these different areas. The encouraging thing is this, so it's not all doom and gloom. The encouraging thing, well, here's the fall survey data here, about 800 sheep we were seeing. Then the die-off hit, the sicknesses came in. So that dotted line is where we're at now, right around the 300-ish range. But you see lamb recruitment. We had 2 years of really poor lamb recruitment in the single digits. And every year it's been growing. And we've done nothing. I'll get to it in a sec, pretty hands-off approach so far. So we were doing some removals of sheep out of this area in addition to hunting for population control. So from 2007 to 9, the 65 sheep, the one that was in Vanna's slide, that was one of the transplants into the highlands. This is just kind of perspective on some of the stuff we were detecting. And I don't have the percentages as far as what percentage of these sheep were testing positive, but just what was showing up within the herd as a whole. 2011, we went and sampled, ground darted a few sheep. A lot of it's fairly similar as far as the detections that we had for these various pathogens. And I want to say on the previous stuff, we did go back and have bank serum from 2007, 2009. We did detect a MOVI in those sheep. Our wildlife vet, Jennifer, said they went back and took a few of those and they detected it. And then most recently with the statewide Bighorn Sheep Project in Montana, we've been doing more and more. You can see the amount of pathogens these sheep carry, same in a lot of places. So as much as we struggle with these sheep and their goal in life to die, we do have, to me, what some of this shows is they actually do have the ability to survive some of this stuff. So maybe we don't give them enough credit as we could, even though they are susceptible to a lot of things. And one of the things that's bugged me the most is if we potentially created our own problem here. So in 2008 and 2005, we've moved in 25 mountain goats from 2 different sources into this area. We don't have the disease data to really specify if there's commingling and what the results of that could be. And we'll never know that. But it really makes you wonder if we actually created this potentially, or if it was just something else. So that was a lesson learned. This was the last goat transplant we did in Montana. We haven't transplanted goats anywhere else in Montana since then. Not necessarily because of disease. That's certainly one thing, but there's other reasons that play into that too. I think we're still considering options though for other places. So this is a slide similar. We've seen a lot of this. It just shows you the diversity of what we see in all these different pathogens, and where we have some places carrying things, same broad pathogens are being carried in some places similar to other areas, but yet you have die-offs and some places you don't. So the biggest challenge, this is where like Manna was saying, biggest challenge I have is if our hands-off approach right now is the right approach or not. That lamb recruitment is improving, which hopefully is going to continue, and we don't have to do anything, but time will tell. I've thought about a lot of ideas of test and removal options. Is it something that we could learn as we're learning more and more about these sheep from like the statewide project, learning about their distribution, learning about their movement patterns, subherds within these areas, different hunting districts, correlation to private lands. There isn't really a domestic issue here. Correlation to wilderness, Bob Marshall wilderness areas. Summer range, winter range, transition areas, we're learning a lot of things about these sheep among, like everybody is, but is it going to be enough to actually pull the trigger someday? And if we need to do a test and removal strategy, and how applicable would it actually be? Do we translocate sheep into this area? We've only put 22 sheep in here. 1999, we put 22 sheep into the Sun River area from Western Montana. Do we do within range options? Are we going to be moving bugs around and creating more problems? I've been reluctant to do any of that so far, at least from my end in this area. So ultimately, this is where I've landed anyway. Is a fairly hands-off approach. But again, I'd like to get perspective from folks. So when do you actually decide, okay, that's time, we need to do something or not? How long do you let it ride and hope that things recover versus things actually get worse and worse in time? That's kind of the $1 million question that everybody struggles with. So maybe in another decade, if I'm still here, if things improve, I can switch that title and numbers will come back and we'll be back to where we were 15, 20 years ago, but we'll see. So with that, I want to acknowledge a lot of people that have been here a lot before me. There's a ton of people that worked in this area that have done a lot of good management and research on sheep, among others, and all the local people that have a real sincere interest in these sheep and here along the Rocky Mountain front. I probably don't have time for questions, but I'm happy to stick around later too if you have stuff. So thank you.
Hello, I'm Ian McLeod from Northwest Territories in Canada, and I don't never heard any of you guys talk about predation pressure. And I know 2010 was just the start of when your wolf reintroduction was taken off, and I'm just wondering if there's much observation of because you don't need very many wolves to start pressuring a herd. And you had, before that, you had a more or less sedentary sheep population that didn't have much worries. And then with wolf and lynx reintroduction And then when you have wolf kills, you have increased survival of eagle chicks. And so I'm just wondering if maybe with the increased pressure and less grazing time, maybe the diseases that were there all the time would be able to have picked up and have more effect on the herd than what they were before.
Because of the predation presence creating that disease or expressing it. Is that what you're asking? It's actually
That I know the wolves we have in the Northwest Territories, they'll go in and they'll test the herd, and most of the time they won't get anything, but they'll move the herd off an area and into a different area, and sometimes they'll drive them into deeper snow where it's easier to get them. So
yep. And I don't think so the wolf presence, there's certainly wolf presence. It's the full gamut of predators there. Lions, black bears, grizzly bears, wolves, coyotes, eagles, it's everything. I don't think at least I haven't detected that being an issue that would have potentially caused the problems with disease expression and creating issues with a die-off type event. I think probably the biggest thing related to predation there now is the additive predation, what it is on a smaller group of sheep. So if you're not able to control those predator numbers to some degree, obviously it's going to have more impact on what's actually currently there. Probably the biggest predator that I notice on these sheep is lions. And that's a never-ending balance of lion and lion hunting opportunity, maintaining that opportunity versus lions that keeping numbers in check. We don't see a lot of mortality, at least I don't, from a wolf end of things. Kevin.
Okay. Hey, Brent, you mentioned those 2 mountain goat transplants from Western Montana. Knowing what we know now and not knowing what you didn't know then, but those 2 sources of goats, is there any chance of going back and collecting some samples from those goat populations with the inference that maybe, are you guys back calculating that at all?
Yeah, and I've been working with Jennifer to get the results from those transplants. Matt Becker, our tech there, did send me some of the information, but it's pretty limited. So I think we're going to try and dig up some more of that stuff from, to backtrack that, but then also to actually go in and collect anything now. I don't know if that would, it could add to that subset of information. The interesting thing about those goat transplants, this area is a big source herd for goats years and years ago. And the transplants came from the Crazy Mountains and Round Butte, east of Great Falls. Those were areas that were initially goats that came from the Sun River area. So those goats actually were coming, their offspring or their lineage decades down the road were coming back to that area. So genetically, there's some similarities, but whether or not the pathogens are similar over time is hard to say.
So obviously, as we saw in the previous presentation, in this presentation, there was a lot of animals taken out of there alive during a lot of this time period. What kind of a factor do you think that might play in this picture?
As far as the reducing the population by too many animals?
No, just, I mean, it takes a lot of effort to catch that many animals over that time. Are we adding stress level? Or what are we doing? I mean, when we circle through that periodically on whether our capture effects are having these issues here. You're taking ewes out, you're taking the wrong ewes out, the right ewes. I mean, what do you think?
It's hard to say. I mean, that I've thought about that too. What, did we actually create some more issues there by doing some of that in an indirect fashion, whether it's stress levels or particular ewes that were being removed, that not that we're selecting for healthy or unhealthy sheep, but that certainly is something I think down the road if numbers ever got back to the point where we could be transplanting sheep out of there again, if we did that, we'd have to be a little bit more careful about that, because 180 sheep over 3 winters, that's a lot of sheep moving out of that area. And that, that's something we might have to be more selective about. And maybe hunting is a better opportunity instead of moving them. Thank you, guys.
The last presentation in this session will be Daryl Lutz. He's going to talk about the Whiskey Mountain Bighorn sheep pulling a management lever. So unlike Brent, Daryl wrote a short autobiography about everything that he's done, and I've taken the liberty to edit it just slightly. So Daryl has been dedicated to the Wyoming wildlife and to the Wyoming Game and Fish Department for 34 years. He currently serves as the Lander Wildlife biological coordinator. Let's see, he currently spends most of his time working on preserving migrations and minimizing wildlife vehicle collisions, and working to restore the infamous Whiskey Mountain Bighorn sheep herd. He is co-chairman of the department's Bighorn Sheep Working Group and recently volunteered as an executive committee member for the Northern Wild Sheep and Goat Council. While away from the office, he enjoys nearly anything out of doors. So take it away, Daryl.
Yeah, thank you, Hank. Can everybody hear me? Because my audio on my end audio is pretty poor. So thumbs up from some of you in the front. Yep, good. Thank you. Well, to start with, I would just want to say that doggone, I wished I was there, but I'm getting the opportunity to experience the joys of an angry sciatic nerve. So travel over the Tetons or over the Toby Pass to Jackson wasn't in the cards for me this week. But regardless, I'm grateful the opportunity to be able to join you at least in this fashion. So I wanted to talk to you a little bit about the Whiskey Mountain Bighorn sheep herd, which I'm sure many of you are familiar with, as it's been a source herd for transplants all over the West over the past several decades. But Brent, I did want to just mention real quickly before I get started that we've been kind of doing a hands-off approach for more or less in this herd after its all-age die-off for over 30 years. And so we're at the point now where we're ready to pull on a management lever. Let me just acknowledge that there are a lot of folks involved in this project listed in that you can see on the bottom of that slide: Pat Nillica with the Fish and Wildlife Service, Brittany Wagler, Rachel Siley, Kevin Monteith with the Hobbs School at the University of Wyoming, Jennifer Malmberg at the State Vet Lab, Hank Edwards, who you all know, and Art Lawson, who is the Wind River Reservation Tribal Game Warden Director. So a lot of folks involved in this work as it is everywhere else. So for a few highlights, as I said, this has been recognized, or this herd is certainly familiar to most and probably infamous for many because it's been recognized for years as the largest congregation of bighorn sheep in the country. That's no longer the case. As I said, it was a source herd. Over 1,900 sheep have been captured and translocated from this herd from the mid-1900s through 1995. It still does to some extent provide exceptional opportunity for hunters and photographers. And it's the focus of particularly intense research the past 6 years. And I just really want to stress a thank you to Rachel, Brittany, and Kevin for the incredibly hard work that they've done to get us to the point where we're at today. So a little bit of herd history. It was estimated there were about 500 sheep in the Wind River Mountains in the mid-1900s. Population grew to what we estimate to be about 2,000 sheep in the late 1980s. We had an all-age die-off caused by pneumonia in the winter of 90 and 91. Today, we estimate the herd to be less than 500 sheep, probably closer to 4. Lamb recruitment, at least we believe lamb recruitment underpins the population decline due in part to chronic pneumonia based on Rachel, Brittany and Kevin's research. And while these sheep, at least compared to other popular condition, overall, they do maintain body condition well through winter, but they experience nutritional stress during the summer, which is a bit counterintuitive. And then recent observed lamb ratios dramatically declined in 2015 and were the lowest ever observed in 2017. As you can see in this graph, while you set that, there's a trend in those data that shows some increase in lamb ratios we're observing. It's pretty apparent that while we're counting lambs in December and January, that they just simply aren't being recruited into the population. And again, that in large part is due to pneumonia. And that, of course, results in this kind of population trend, which is pretty steep decline. Okay, I've seized up here. So I just wanted to make the point, what we do and have been doing for several decades, winter range veg production transects, and don't get hung up on the height of those bars, but just know that forage production on key winter range sites for this herd have been really productive and provide adequate forage. The amount of utilization that we see then as a result of the number of animals on those landscapes and that we've got, that in most years it's all below that 60% utilization rate that we at least deem to be the limit. So that at least in part explains why the sheep are doing, or at least in terms of their body condition, seem to do well during winter. But again, it's puzzling that they don't do very well on their summer range, which most of us assume should be unlimited, more productive, and highly nutritious, but that apparently is not the case. So I went through a long and extensive collaborative process with the public in 2018 and 2019. Some of you in the room participated with us in that process as we brought in a panel of experts to help us better understand what we were looking at, what we were dealing with, and maybe some opportunities or management tools that we might consider to, at minimum, arrest this population decline and hopefully reverse it. We really don't have any, as the others have said, there really isn't any silver bullet, but at least this plan does outline a number of actions that were of interest to the public and we think that are worthy of doing. The important point to make here is that we felt that the public needed to be more intimately involved in this process, just given the trajectory of this herd. The whiskey herd is a intertwined herd in the Du Bois community. That's where the National Bighorn Sheep Center is. And it's just a herd important, both locally and in the state. And I think arguably, as I said earlier, just because it was a source herd for so many years, even throughout the West. So whatever the result is in continued population change, we wanted the public to be a part of that planning process and have ownership in where we go. One of the strategies that we outlined in 2019 was remove clinically ill bighorn sheep. And so it was interesting to listen to the talk earlier about using citizen science and observations to be able to perhaps identify what clinically ill sheep look like, because I think that could be hugely valuable. But the point I wanted to make was with this slide is at the time, we really didn't know, at least in Wyoming, much about the test and remove work that had been started primarily in Idaho with Francis Cazier's work. And it was literally 2 years ago this September at the last Wild Sheep and Goat Council conference that we had the opportunity to listen to some of the work that she had done and the results that they were seeing. And that culminated then in the workshop that was done last June on test and remove, because I think there needed to be opportunity for increased information exchange and better understanding if this tool might be something that could be used in other herds, including this whiskey herd. So what we need to add to the plan is the strategy that you see below, and that is to conduct test and remove and to evaluate the efficacy of doing so. So what we've done in whiskey herd is we've taken a baby step towards test and remove. Come on. So we have started doing test and remove, and of course, one of the first steps in doing that is defining those sheep that would, and I hate using the word qualify, but I can't think of a better one. But with that would qualify as a chronic carrier, and so what we did then is define those that chronic carriers as those that were positive for mycoplasma and pneumonia twice within a 12-month window, March to March. That allowed for 3 testing events with a minimum of 2 months between testing events. And we included or are including males and females, and only adults would be considered for removal. And the chart that you see in the middle of that frame then is the result of Rachel and Brittany's work. And those orange dots that you see are positive tests for movie. That last column on the right then are those sheep then that would quote unquote qualify as chronic carriers. So this past spring, last month, essentially last month, we did accidentally remove one in December. 4 more were removed in March, but we did remove 5 ewes that were identified as chronic carriers of MOVI. We did that in the Red Creek subherd, which is on the Wind River Reservation. And just so you know, Wind River Reservation are the Eastern Shoshone and Northern Arapaho tribes. They are keenly interested in trying to figure out a way to increase the corn sheep throughout the reservation, but in particular, this Red Creek herd subherd has declined the past 5 years from about 120 sheep to an estimated or counted about 40 the past 2 years. So again, we removed 5 ewes that were identified as chronic carriers given our definition. And those are nasty, neat, frothy photos of a couple of the sheep. And just know that based on postmortem examination, they were all pneumonic and 3 had paranasal sinus tumor. So we think we're on the right track. Well, Tony, I could use my 5 minutes left if this thing would work. Yeah, the thing is pretty seized up. Well, that's okay, because we're basically at the end of my PowerPoint presentation. I just wanted to give everybody an update where we're at, that we are going to be up towards test and removal. We are going to continue with the longitudinal study that Rachel and Brittany have started in the Red Creek herd as we go down the route of continued test and remove. We are going to get our hands on with either helicopter captures or drop nets all the sheep in the Red Creek herd, or as many as we can, test them, and continue to do test and remove over the next several years. So just as Brent said, I wonder if we're on the right road. I guess time will tell. So with that, there we are. Are we on the right path? I guess we'll find out. With that, I think there's some time for questions if you have any.
Real quickly, Daryl, could you also pull the lever on some habitat improvement projects? Could you elaborate on those?
Yeah, thanks, Steve, for bringing that up. Yeah, at the same time, we've recognized that there are opportunities to improve habitats, primarily on those primary winter ranges for the whiskey herd. We've done a little bit of work in it with a meadow complex that is used extensively by a different subherd of this population on one of our habitat management areas. And then maybe most importantly is that we have been working with the Forest Service. The Shoshone National Forest has gotten dispensation, maybe might be the right word, to do work in the glacier addition to the Fitzpatrick Wilderness, to use chainsaws to allow us then the opportunity to hopefully carry a prescribed burn up Whiskey Mountain itself, which will increase the amount of winter range available to these sheep. And then finally, we are developing a long-term, more comprehensive management plan for more primarily conifer removal to open up more, again, primarily more habitats, primarily in those lower elevation winter ranges. But as I said, it appears the challenge here, or at least one of the challenges here, is summer range, and it's all within wilderness. It's all, just as you would expect any kind of, above timberline alpine tundra. It's going to be difficult, maybe impossible, to improve habitats in that environment. So we are doing what we can with habitat at lower elevations.
Daryl, Steve again, could you quickly comment on, I think your collaborative public process, quite elaborate over a year of having 100 people or so in the room, was definitely provided you the opportunity to pull these levers because you communicated so well with the public. If you want to elaborate on that, that's fine, but thank you for doing that.
Yeah, the collaborative process of, there's a lot of value in getting folks in the room, not just because there's a ton of perspectives, as you all know. But in addition to that, it also provides agencies, whether it's our Game and Fish Department or the Forest Service or the BLM, the social license, to do some of the things that might otherwise be more difficult, like doing mechanical work within the glacier addition. So the collaborative process not only gets ownership of the public and what you're doing and what the outcome, and maybe much more important point here is what the outcome is going to be, but it also then allows us the opportunity to try some things that might not otherwise been possible. And Tony, I am trying to stop sharing, but it just says to cancel. So I'm canceling.
Well, let's have a round of applause for this round of speakers. Before you take off, Allie would like to talk about a movie.
So we have a little bit of a change to the schedule here last minute. Lunch isn't going to be ready until about noon, so we have about 20 extra minutes. So we'd like to show that Denizens of the Steep film that I was talking about a couple of days ago. It's about 9 to 10 minute film. So if anyone wants to, take a break, feel free and leave, but we're going to show that right now. And while we're getting that queued up, Steve Kilpatrick has a couple announcements too.
Thank you, Allie. I'm Steve Kilpatrick, and right now on the board of directors for the National Bighorn Sheep Center right over the hill in DuBois. One of the things we've done the last few years, well, let me back up. That area was developed primarily based off the Whiskey Basin herd. And we now have agreements with National Wild Sheep and some other chapters and affiliates across the nation to go more national with our outreach and educational effort. That's our mission, outreach and education among sheep advocates. So one of the things we started at the Sheep Center is a webinar series, and we've had some great ones. We've had Hank Edwards talk on a webinar, Kevin Hurley, Doug McQuarter, Dr. Richard Garrett, and many others. So I'm putting out a plea for you folks so we can better share and educate each other and stay connected, that you offer to give presentations on our webinar series. And I've got a few brochures up here you can look at, but if you just Google National Bighorn Sheep Center, you'll get a person you can email. And if you'd like to present something, we'd love to hear from people across the nation and other countries. Canada's more than welcome. So thank you. And yeah, and this Saturday, since Dr. Francis Kassir is here, we did rope her in. And Saturday at 11 o'clock, she will be giving a Zoom webinar. I'm not sure what all you're going to talk about, Francis, but we'll she's making that up as we go right now. But anyway, and all those webinars are recorded and they're on our website. So please go to the website, ask questions, we'll answer them, and we'd love to have presentations from folks across the country. And we're kind of focusing on the wintertime when everyone's not so busy and a little bit bored. So you can sit back, prop your feet up, get a beer or whiskey, and watch webinar and ask questions at the end of it in the evenings during the winter. So a couple of presentations a month is what we're kind of shooting for. And if you go right now to the website, National Bighorn Sheep Center, you'll see the webinars that are coming up. And the one coming up soon is Francis's. So thank you for your time. And our mission is to keep you guys connected and share and disseminate information about sheep conservation. We need your help. Thank you. One question, Melanie. It's a combination of us. Having worked for State Wildlife Management Agency, my frustration, and I think it's with all of us, is to get the science to the general public and get them to support us and be behind us. Daryl Lutz and his folks went all out with a year and a half's worth of collaborative meetings in one little community to be able to pull the management lever. But I think, so the announcements do go out to all the chapters and affiliates, and National Wild Sheep is putting out the announcements for the webinars too. So it's a mix of scientists and the general public. And I think that's important that we have that nice mix.
Okay, Doug asked me to say a few things about this film before we start. You guys can hear me okay. So yeah, just as a reminder, I briefly talked about this film during my presentation on Tuesday, but this is a film that we put together with some local filmmakers in the community that were interested in helping us tell the story about how do we balance winter recreation in the Tetons with protecting bighorn sheep. So you'll see Sarah and Carson are in this film. From kind of the agency's perspective, but a lot of it is from the skier perspective. And one of the filmmakers was actually here last night at the banquet, Josh Metten. Maybe some of you got the chance to meet him, but we thought it was, it's just a fantastic way to share this information with the public. And I just want to give kudos to the Wyoming Wild Sheep Foundation, because as we were kind of had this kind of crazy idea to make this very expensive film, they were one of the first funders that stepped up and said that they would fund it. So pretty awesome.
When I first started getting into backcountry skiing, it was definitely for the pure fun hog.
sense of life. I just wanted to see more powder. I wanted the exhilaration of seeing these beautiful lines. I really enjoyed the beauty of the places, but I was focused more on the objectives themselves. I like figuring out terrain, figuring out routes up peaks, figuring out routes down peaks, taking on things that people hadn't done before. I think we gain a lot as humans, getting that feeling of being a pioneer. It's very rewarding. It creates a lot of confidence and self-worth. I'm definitely drawn to places where there's less traffic. I like spending time where there's no people. It's just you, the mountains, the rivers, the birds, and the surrounding wildlife. And it's a very special, unique thing to be that deep and that quiet and in that protected environment. And it's irreplaceable. I think as humans, we're walking a fine line, because as backcountry skiing grows in popularity and expands to greater reaches of the skiing public, it's the fastest growing segment of skiing right now, and has been for the last 10 years, that everyone is seeking this beauty, this freedom of the hills. If more people are going to go into the wilderness and we want it to stay pristine, we need to be very, very aware of our environment.
You know, as a person looking up at the Teton range in the wintertime, who would think that there's, 100 or so animals in and out up there? We've lost the majority of the winter range that bighorn sheep in this mountain range used to take advantage of, and we're left with just a fraction. For me, it's hard to think about cherishing a place and being enthusiastic about it without the health of the wildlife there being a key part of that. And I imagine most people who are recreating the Teton Range share my perspective on that.
What really inspires me about bighorn sheep is, look at the places that they live in. And in studying the sheep, we get to visit these places and see the sheep doing the things that sheep do on a day in and day out basis. And they live in some of the most little rugged pockets of the Teton Range, and particularly in the wintertime, I find what they do just incredible. I mean, these sheep have lost their migrations to low elevation winter ranges. And as a result, they winter now at high elevation. And the conditions on those high elevation winter ranges are pretty extreme.
They're at 10,000 feet, 9,000 feet, sometimes actually up to 12,000 feet. We all know it's a lot colder up there. The crest of the Teton is a windy place, and that's actually what they need to survive.
They are just trying to move as little as possible, and whatever we can do to help them out in that regard will go a long way to helping them persist in the future.
2008 to 2010, I believe, there was field work done by a master's student at the University of Wyoming. They GPS collared bighorn sheep and got permission from backcountry recreationists, mostly skiers, to carry GPS units. The findings from that thesis indicated that the sheep are being displaced by skiers and recreationists. The fact is we're viewed as a predator whether or not we want to be.
wild sheep from that. And so is protecting and conserving the wild places in which we roam.
Skiers need to consider not just the weather and the snowpack, the avalanche hazard, but the impact that we have as humans traveling through a wild ecosystem. And that includes being aware of what animals exist and where, and how to give them the space that they need.
I think the way we have to look at it is just that we're not hunters, we're not skiers, we're not climbers, we're not dirt bikers, we're outdoor enthusiasts. And we need to be a team and do everything we can to protect this environment that we're going. Because it's not about our sports, it's about our connection to the mountains while we're out there. And it's up to us to protect them.
And that, I think that's key. You don't just want to be a user of the back country, but a steward. So this place exists, including all the animals.
We all want to ski in these remarkable places. So by no means is my approach that we shouldn't go skiing, because I love skiing. It's my job, it's my life, it's my family's life. I know for sure that I want my child to be able to see all these animals. I want her to know that it's important that wildlife is respected and allowed to thrive and coexist with humans. But I also want her to enjoy the natural environment and go skiing and backcountry skiing and get the joy and the reward and the freedom that traveling in the mountains gives you.
The way for backcountry skiers to become good stewards of the places they visit is to be just open to the fact that you might be interrupting a herd, open to the fact that maybe you ought to go somewhere else or come back. I don't think there's any right way to be a steward, but if you're open and willing to evolve, then you're on the right track.
Well, sorry that was a little choppy with the Zoom, but you can go on, it's on heathonsheep.org if anyone wants to take a second look. But thanks for watching.
folks are gonna drift away. But I think everybody, before they start drifting away.
I don't want to preempt that. That they give them that suggestion or that idea. So maybe it's next. All right. If everybody can find a seat, we'll get going with our afternoon session here. So please find your seat. All right, for the first session of this afternoon, your moderator is going to be Ben Wise. Ben Wise is a regional disease biologist for the Wyoming Game and Fish here in the Jackson region. And for those of you that went out on the National Elk Refuge yesterday, he was one of your guides there. So Ben, I'll let you take over.
All right, our first presenter this afternoon is going to be Katie Hugler. Katie completed her bachelor's at the University of Idaho and a master's at the University of Wyoming, where she focused on predator-prey dynamics between mule deer, mountain lions, and coyotes in Southwest Wyoming. She is currently a PhD student at the University of Idaho with a goal to link forage availability to demographics of bighorn sheep populations in Idaho, Washington, and Wyoming.
This thing is super heavy. I think I'm gonna do the walking around move too. Cool, thanks, Ben. My primary goal here is to keep people from napping after lunch. So with my humor and also some exciting results from some of the stuff we've learned about how sheep respond to fire. So rapid global climate change has increased both the frequency and severity of natural disturbance events. And we're starting to see more frequent flooding, prolonged drought, destructive tropical storms, as well as unprecedented wildfires. And the ability of animals to survive these stochastic events and navigate these altered landscapes really depends on their ability to rapidly adjust behavior after disturbance occurs. And the ability to adapt to altered landscapes is really relevant when we think about modern wildfires, where increasing drought has caused wildfires that are unprecedented in both size as well as severity. And sometimes when we think about wildfire, in a lot of cases, we think of it as this beneficial process, especially in systems where wildfire opens up canopy cover and promotes vegetation growth in the understory. But what we're starting to see more and more often is systems like this, where prolonged drought is causing fires to be more and more frequent. And instead of allowing native vegetation to recover, what we see is these more fire adapted species such as cheatgrass overtaking these systems. And this is causing a complete shift in forage composition on the landscape and really changing the forage base for critters that rely on these food bases such as ungulates. And with changing fire conditions, it's going to be even more pressing for us to get a better understanding of how fire shifts the forage base, how that affects ungulate behavior, and ultimately how that scales up to demographics. But addressing these questions is admittedly really difficult. Normally, we don't have the data to address these questions because we have to have a disturbance occur. But we also would like to have data in the form of perhaps GPS data before the disturbance occurs, as well as afterwards. But if you haven't already put it together by now, we had the unique opportunity to address just that. So for my PhD work, as Ben mentioned, I'm broadly focused on linking on-the-ground measures of nutrition to demographics of sheep populations in several areas. And one of them includes the Asotin Creek herd, which is one of the northernmost herds in the Hells Canyon meta population. And as part of my dissertation work, we have GPS collars on female bighorn sheep throughout the study area. And while we were doing field work in 2021, on July 7th, a lovely lightning storm came through and ignited multiple wildfires that created this fire complex system known as the Lick Creek Fire. And that Lick Creek fire burned really hot and fast and burned about 80,000 acres in 3 days. But it also burned about 80 to 85% of the sheep home range. And so we took this unique opportunity and perhaps this unexpected event. We wanted to capitalize on this and get a better understanding of how these sheep might adapt to this drastic reduction in forage on the landscape. And so we evaluated changes in home range size, as well as displacement, whether or not sheep were displaced by the fire, as well as changes in movement patterns. And did I just lose the mic? Oh, okay. Changes in movement patterns that may be reflective of the change in forage availability, as well as shifts in resource selection. But it's important to also consider how individuals may respond to fire instead of just at the population level, because individuals with varying resource requirements may respond to disturbance differently. For example, females who are lactating or have the responsibility of caring for not only themselves, but also their neonate, have the responsibility of provisioning a neonate, so gaining access to high quality forage, but also reducing the risk of predation. And so females who have a lamb at heel may be more risk averse compared to a female who's not lactating and may be more plastic in her movements. She doesn't have the responsibility of provisioning a lamb and instead is just trying to put on fat over the summer. And so we also considered not only or at the individual level, so comparing lactating versus non-lactating females in changes in behavior. So we expected that sheep should definitely be displaced by the fire. So we expected that home range overlap of individuals before and after the fire would be relatively low. But we also expected that before the fire, let's see, which is this the pointer? So before the fire, food's still out there, it's still relatively available. We expected that individuals wouldn't have to move very much to gain access to food, and home ranges would be much smaller than after the fire, when essentially all of the food was gone. Animals are having to move much farther to gain access to resources. But we also expected to see differences in movement patterns before and after the fire. And we expected that movement patterns would be, after the fire, would be a reflection of the essentially depletion of forage across the entire sheep's home range. But as I mentioned, we were also really interested in how in shifts in resource selection. And in particular, we were super interested in the relationship between selection for escape terrain. I don't know if you guys know this, but sheep really like escape terrain. And the trade-off with escape terrain and forage availability on the landscape, and how that changed as a function of before and after the fire, but also with lactation status. For example, before the fire, as you can see, food is much more available. And so we expected that individuals would be super tied to escape terrain and wouldn't have to venture far to get access to food. But after the fire, food is gone. We expected that animals would have to sort of go farther from escape terrain to gain access to food. But because lactating females are still having to avoid the risk of predation, we expected that lactating females would still remain much closer to escape terrain than non-lactating females. And to address these objectives, we used GPS data from 18 female bighorn sheep collared throughout the study area. And we subset the GPS data into a pre-fire period defined as the 25 days before the fire occurred, and a post-fire period defined as the 25 days after the last day that the fire had rolled through the study area. And to calculate home range size as well as home range overlap, we calculated home ranges for each individual in the pre and post-fire periods to get at home range size. And then we also calculated home range overlap for each individual before and after the fire. We also calculated a number of movement metrics before and after the fire that we expected would be sort of indicative of foraging behavior. And those included residence time, or how long an individual would spend in a particular area, time to return, or once they left a patch, how long did it take them to return to a particular patch? And also revisitation rate, or how often they revisited a certain patch. Yeah, so getting to the most exciting part of the talk. Contrary to our expectations, sheep were not displaced by the fire. And instead of a figure, so home range overlap was super high for individuals before and after the fire. And rather than a figure, I think there's a really good image that sort of depicts exactly what sheep were doing. So this is essentially what happened. These sheep moved a little bit, but essentially returned right to where they were at. The fire essentially burned right around these sheep. So yeah, sheep were not displaced by the fire like we expected. However, home ranges were relatively large. If we don't include lactation status, they were relatively large and variable before and after the fire. But lactating females had much smaller home ranges, regardless of whether it was before or after the fire. And in fact, they were about half the size of non-lactating females. But lactating females before the fire spent about 4 more hours in a particular patch than non-lactating females. And after the fire, they didn't change their behavior at all. But non-lactating females increased the amount of time they spent in a patch by about 2 hours. And my interpretation of this is essentially that lactating females are spending more time in a patch to essentially capitalize on those resources before they move on. And non-lactating females are perhaps moving on just a little bit quicker. And before the fire, lactating females and non-lactating females were are taking about the same amount of time to return to a patch once they left. Lactating females didn't change their behavior after the fire, but non-lactating females doubled the amount of time they took to return to a particular patch. And this, I think, maybe suggests that non-lactating females are perhaps a little bit more plastic. And after the fire had rolled through, they were spending more time perhaps exploring other patches, and non-lactating females weren't necessarily doing that. And so before the fire, if we don't include lactation status, individuals were revisiting a patch about 32 times compared to about 20 times after the fire occurred. But lactating females were still revisiting patches regardless of whether or not it was before or after the fire. They were revisiting patches much more often than non-lactating females. And again, I think this just goes to show that perhaps lactating females are revisiting these more familiar patches more often. Non-lactating females are sort of moving around to new patches. This is the 2-second wait they were talking about. Oh, no, it's okay. Okay, yeah, so moving on to some of the results from the resource selection functions. And again, just to remind you guys, we were really interested in the trade-off between selection for escape terrain and forage availability. And in this case, it's just indexed by NDVI. And what we found before the fire, again, food is still relatively available, is that non-lactating females were willing to go farther from escape terrain when food in their local environment was bad, or I guess more brown in this case. And lactating females were willing to do the same thing. However, the threshold for how far they were willing to go from escape terrain was about half that of non-lactating females. But after the fire, again, forage is essentially gone. Non-lactating females were still willing to go farther from escape terrain, and actually they relaxed that relationship with escape terrain quite significantly, and were willing to go even farther than 800 meters from escape terrain when food at the local scale was bad. But this was different for non-lactating females, and forage greenness wasn't important in determining how far they were willing to go from escape terrain after the fire, and instead they were still really tied to escape terrain after the fire. So my interpretation of this is that females are sort of hedging their bets and realizing that there's essentially no food and the food is no good. So they're sort of trading off staying close to escape terrain for gaining access to food that's no good anyway. So bighorn sheep, I think, are just like many other ungulates that have been adapted to these more small and infrequent fires that have occurred over history. But we still don't know how these animals are going to adjust to these more large-scale, frequent fires that might occur. And despite this drastic reduction in forage, sheep were not displaced by the fire whatsoever, and they still exhibited relatively large and variable home ranges. However, lactating females were constrained to a much smaller home range than non-lactating females. So sheep might be inflexible when it comes to these dimensions, but we did see that there was variable movement strategies, and in particular, non-lactating females were sort of changing their behavior in response to the drastic reduction in forage. But we did also see shifts in selection in response to the fire, and again, non-lactating females were able to be much more plastic and perhaps risk-prone to gain access to food compared to non-lactating females. And so we demonstrated that there's definitely changes in behavior occurring, but we still don't know really what this means for demographics of sheep populations. And interestingly, in the autumn after the fire, when we caught these females, this was the poorest condition from a nutrition perspective that we had observed the lactating females in, here in pink. And there's a couple of important things to note here. And first, that 2021 was a record-breaking drought, which is likely why the fire happened in the first place. But this fire also occurred right at peak lactation. So when these females needed food the most, it wasn't there. So we still have a lot of work to do to better understand what this means for sheep out here. So in the upcoming year, we'll be working on understanding how the fire has shifted forage biomass, as well as quality and diversity, better understanding how sheep behave in this potential shift in forage composition, and ultimately how that might scale up to both survival and reproduction. And so I think we sort of demonstrated here the sort of short-term responses of bighorn sheep to this drastic reduction in forage, but that lactating females seem to be much less plastic in their ability to adapt. And we're going to continue to work on what this means for sheep, especially in Hells Canyon. But I think this is imperative as we move forward and drought continues and fires become much more frequent to get a better understanding of what this means, but at the forage scale, but also how this scales up to demographics of ungulates. Yeah, and with that, I hope I don't have time for questions, but I probably do. So thanks, everybody.
Nice job, Katie. So for those movement metrics that you talked about, how are patches defined in that context? Is that based on like areas on the landscape that didn't burn or had forage quality come back faster or something along those lines?
Yeah, good question. So essentially it's a moving window approach. And so it's essentially building a circle over each one of the GPS locations along an animal's trajectory and then count or quantifying each one of those movement metrics. So you have to define the radius of the circle. And in this case, it was defined as average step length for sheep, which was about 400 meters. And that was also kind of where the variability in all those movement metrics kind of asymptoted. So that's what we went with for now, but it's definitely sensitive to the radius that you choose for those analyses. And there's also like definitely other ways to do it, and we may consider those in the future.
Katie, I'm just wondering, pre-fire, was there pretty good veg data, you know, in terms of composition? I mean, not by design, but by coincidence, maybe, that so that you guys can make that comparison pre and post in terms of composition, et cetera.
Yeah, so perhaps this is like the coolest part about this is that in 2021, we have been doing extensive veg work out there, as part of my PhD work, but totally not expecting the fire to occur. So we have extensive data on composition, biomass quality from the before the fire occurred, as well as directly after the fire occurred and into this next year. Yeah.
I know you're still working on it, it sounds like, but did you have any lambs die over the course of the summer there?
No, we didn't. We had nobody die. That's not true. We had one lamb die, but it wasn't related to the fire. It just happened to die during the fire. So no mortalities, which is maybe not comparable with some other studies that have looked at sheep after a fire, but yeah, no mortalities thus far.
Good job, Katie. Our next presenter is going to be Rachel Smiley. Rachel received her bachelor's from the University of Connecticut before moving west, where she worked with mule deer, elk, bighorn sheep in Wyoming and Idaho. She's currently a PhD student with Dr. Kevin Monteith in the Monteith shop, studying the interactions between nutrition, disease in bighorn sheep.
Thanks, Ben. Okay, first I want to start off by thanking everyone involved with this particular study, and then also this is part of a much larger project, and there's so many people who have contributed that I probably would take up the whole 15 minutes if I named everyone, but thanks to everyone who's contributed in some way to this project. So the variability of food resources across landscapes and across time can influence species distributions, animal behavior, and importantly, the life history of animals. So nutritional condition in many animals has been directly linked to pregnancy rates, timing of first reproduction, size and number of offspring, and of course, survival. So how an animal acquires food resources and then allocates them is the fundamental link between an animal and its environment. For animals living in temperate environments, there's large seasonal changes in the food quality and amount available to them. So while this deer had a lush meadow during the summer, predictably in the winter, food is going to be much more scarce and of worse quality. So an animal needs some way to deal with these predictable seasonal changes in the food resources they have available to them. And so one way to do that is to accumulate fat reserves. Fat is the primary energetic resource to many animals. And so in the summer, when food resources are really abundant, they can eat almost in excess to acquire extra resources in the form of fat reserves that they can then use later when they know food is going to be much more scarce. But this acquisition of resources during the summer becomes a little more complicated when you factor reproduction in. So many animals in temperate environments give birth in the summer when food resources are the most plentiful. So mother has to be acquiring resources for herself in the form of fat to use later, but she also has to invest a lot of energy into her offspring to make sure they can grow and survive throughout the summer and into the winter. So when there's nutritional limitations, this can present a trade-off between reproduction and survival. So if food resources or energy isn't unlimited, an animal has to decide how to allocate resources both to herself and to her offspring. And so how an animal allocates nutritional resources has been termed risk-sensitive resource allocation, but it's actually a concept that came from the world of economics. So bear with me for a minute while we jump into economics to describe this concept. So let's pretend we're a company that produces trucks. The ultimate risk for the company is bankruptcy. So that's the thing we're trying to avoid. Now we have a certain amount of financial resources to invest in both in ourselves as a company and into our product. So we have to pay employees salaries and also have to produce these trucks and buy materials to make them. But of course, the money we have available to us varies over time, both in maybe predictable or unpredictable ways. So we have to decide how to invest that money relative to how much money we have. So when times are good, we can invest heavily both in ourselves and into the product. So we can maintain the quality of our product and also high salaries for employees because we have a lot of money to spend. But when times are bad, we have to be more conservative or think a little bit harder about how we're investing the money. So we could do that by firing employees or lowering salaries, or we could do that by lowering the quality of our products. So using cheaper materials to make those products. So it's all about avoiding that ultimate risk of bankruptcy and changing how you're allocating resources depending on how much you have. So we'll come back to wildlife, and instead of a company, we're a deer that's trying to produce offspring. And the environment or nutritional resources available to us varies a lot throughout the year, both predictably because of seasonal changes, but also unpredictably because of changing environmental conditions. And so we have to decide how to allocate resources to avoid the ultimate risk of starvation. So when times are good, we can accumulate a lot of fat resources for ourselves and also invest heavily into the growth of our offspring, make sure we can both survive throughout the year. But when times are bad, we have to be more conservative with how we're allocating those nutritional resources. So potentially by gaining less fat or by investing less in your offspring. And for many ungulates, when there's that trade-off between deciding between yourself and your offspring, the selfishness prevails and you generally try to acquire resources for yourself first before investing in your offspring. So the seasonal and predictable nutritional dynamics are exacerbated in alpine environments. So alpine environments have short and cold summers with generally low primary productivity. And there's also late season storms or early fall storms that make it pretty unpredictable on the food resources that you're going to have throughout the summer. So not all animals are adapted to living in these environments, but for those that are, it's likely extremely important to be very risk sensitive in how you allocate those resources to make sure that you can deal with the unpredictability that comes with living in alpine environments. So we wanted to test this with bighorn sheep. Do they allocate their nutritional resources in a risk sensitive manner? Meaning specifically, do bighorn sheep vary how much fat they gain or lose between seasons by how much fat they have, by their reproductive state, so whether or not they recruit a lamb and whether or not they're pregnant, and then also by environmental context or population. So we did this in 3 populations of bighorn sheep in Northwest Wyoming, right here in the Jackson population, in the Upper Shoshone herd, which is part of the larger Absaroka population, and the Whiskey Mountain herd, which Daryl gave a good overview of. And these herds are similar in many ways. They're all relatively large populations of bighorn sheep, and pneumonia is considered endemic in all of these populations. So the pathogens associated with pneumonia have been in the populations for decades. For the most part, there's not a lot of adult mortality due to pneumonia in these. But where we see the herds differ is their population dynamics, which is generally associated with pneumonia in these populations. So the Jackson herd has experienced several crash recovery cycles associated with pneumonia. So they've experienced die-offs, but recovered pretty sufficiently from those. The Upper Shoshone population, it's very large, but there hasn't been a documented die-off associated with pneumonia. We have seen a couple adults dying of pneumonia, so it's certainly there, but hasn't led to a huge crash. And then of course, the Whiskey Mountain herd, which had that die-off in the 90s, and the population has been declining ever since. So that decline is mostly due to low levels of lamb recruitment in that herd, as Daryl was talking about, that's in the single digits in some cases, and really the problem for that herd. So our study designed to see how bighorn sheep allocate their fat resources. We are doing a longitudinal study based on individuals that we capture every December after they've come off of their summer range. And then again, we capture them in March, shortly before they return to their summer range. And we collect a suite of data points on each animal including body mass and age. But importantly, for what I'm going to talk about today, is we measure the amount of body fat these animals are carrying. So we know how much fat they ended the summer with, and then we also know how much fat they're ending the winter with. So first, we want to know what influences how much fat these sheep are gaining over the summer. And of course, we see that in general, sheep do gain fat over the summer, and this is really influenced by whether or not they successfully recruit a lamb. So sheep that can recruit a lamb gain almost 6 percentage points less fat than sheep that either weren't pregnant or their lamb dies at some point throughout the summer. But what we really wanted to know was, how does their nutritional condition before the summer influence how much fat they gain over the summer? And so we can see across the board in all three populations, sheep that are skinny before the summer really have to focus on packing on the fat throughout the summer and make sure they can gain those fat reserves throughout the summer to then use for the winter. But a sheep that starts the summer in pretty good condition, she has plenty of fat already, doesn't have to focus quite as much on gaining fat throughout the summer and can invest that energy to other places, like maybe reproduction. So we see in all three of these herds, how much fat they gain over the summer is largely dependent on how much fat they start the summer with, and also whether or not they recruit a lamb. We wanted to look at the same thing over winter. So how much fat are these sheep losing over the winter? And of course, invariably, sheep are losing fat over the winter. This didn't vary by whether or not they were pregnant, but in all three of these herds, we have really high pregnancy rates. So there were very few sheep in these analyses that weren't pregnant. And again, we wanted to know what influenced how much fat they're losing over the summer. And again, we see that sheep that are pretty skinny starting the winter, sorry, I said summer. Sheep that are skinny starting the winter really have to conserve those fat reserves and save them to make sure that they can get through the whole winter with those fat reserves, while a sheep that starts the winter with a large amount of fat reserves can really burn through those throughout the winter. So again, how much fat they're losing over the winter depends on how much fat they start the winter with, and doesn't seem to vary based on the pregnancy. So all of this supports that bighorn sheep are allocating their nutritional reserves in a risk-sensitive manner. So they're allocating their resources based on what they have and based on their reproductive status. And it supports that these alpine ungulates really need to adapt to their environment and fine-tune their resource allocation based on the level of resources that they have to survive these unpredictable environments. And I want to dig a little deeper into the differences between populations. So as Daryl talked about and alluded to, the Whiskey Mountain herd, you can see those lines are shifted down a bit because they're just not gaining as much fat as the other two herds are throughout the winter. And then in particular, that yellow line is the sheep that recruit lambs. There's not many of them, but when they recruit a lamb, on average, they're losing fat throughout the summer. And so this supports that we're seeing a nutritional limitation on summer range in the whiskey herd. And we're also talking about the herd with really low rates of recruitment. And when you think about a sheep that has to lose fat to successfully raise a lamb over the summer is also going to lose fat over the winter, it becomes unfeasible for them to raise lambs year after year because they can't just lose fat year after year. So while these lambs, generally the problem is pneumonia with them, it brings us to that perhaps pneumonia and nutrition, instead of two separate stressors, are ones that have added effects on each other. So while we often treat them as two separate things, we're going to dig further with this project to see how nutrition and pneumonia might influence each other to especially make nutritionally stressed herds vulnerable. So again, I want to thank everyone who's funded or collaborated on this project. And I'll take any questions. Steve.
You got any of your veg nutritional quality data back yet to show that there might be nutritional limitations? Because quantity seems to be out there, but I don't know anything about quality.
So in this analysis, I didn't talk about it, but we did include biomass. So just a remotely sensed measure of how much food is in their home ranges. And the whiskey sheep do have lower biomass. So there is actually less available to them out there, but this didn't actually relate to how much fat they're gaining on an individual level. And then you're going to have to ask Brittany about the rest.
And a quick follow-up question while the mic's here. We see those sheep on Arrow Mountain consistently eating horse manure off the trail, and I mean, quite attracted to it. What might be in that? Do we know that they need or want?
That's a great question. I have no idea if that indicates like nutritional stress and they're trying to get the good food that the horses didn't digest. Yeah, we've seen that, I think, on Arrow, like a very sick looking sheep eating poop. I have no guesses, but
I'm just wondering, just to follow up on what Steve said, whether they could actually be trying to get salts out of the
that's a good point. Yeah.
horse manure. But my question is, and I've thought about this, being somewhat familiar with your system, is how do you think that habitat used to support so many more animals? And do you think that, like, what do you think the importance of summer range versus winter range was at that time? And it sounds like maybe there's some changes in forest composition as well that could be altering the habitat.
Yeah, I guess this is just speculating now, but I wonder if 2,500 sheep was too much for that summer range and it became overgrazed. I have no evidence for that, but just wonder if we can't support 500 sheep now, how did it support that many? So that's a good question, but it also speaks to the quality of the winter range for those whiskey sheep. And a lot of the habitat work that's going on now is to help improve or maintain winter range, which is really important for these sheep, because if they had a bad winter range and a bad summer range, that just wouldn't work for them.
Good presentation. So I'm curious, for the animals that they're starting the winter with less fat and then they're losing less fat, how are they doing that? Are they catabolizing other kinds of tissue, or do they have different habitat use or forage?
Yeah, good question. And so there's a whole nother part. This is a paper recently accepted in Ecosphere. And we also looked at lean mass of these sheep. So how much do they weigh minus the weight of their body fat? And if they don't have a lot of fat resources, they're actually catabolizing more protein resources. I also suspect there's something behavioral going on too. So a sheep with less fat can't move around quite as much, but we haven't done those analyses.
Have you looked at any of the historic data to see if there has been a shift in species composition on that summer range at all? Have you looked at any of the Forest Service data for that?
We haven't looked at that data specifically, yeah, to see if there is a change in species composition, but that would be interesting. We've talked to some of the people. John Mayanzinski has done a lot of habitat work even before the outbreak there. And so he had a good feel of what the habitat was like, but I'm not sure if species composition has changed.
Hey, Rachel, great presentation. I have some off the wall question
Cool.
based on Steve's coprophagy observations. So, fasting animals, especially ungulates who are capable of urea recycling and all of that. I mean, we haven't heard much about this, and it's probably not something you guys have looked into. I was just curious if within your lab, you guys have talked about urea recycling and what that looks like in these nutritionally stressed animals. So if that's something anybody's talked about, or you are, especially with that coprophagic behavior, it's like, oh.
Yeah, I don't know anything about that, but something that I just thought of, I'm sorry, not related to that, but is that ungulates need to maintain gut fill. So they need to have their rumens full at all time, which I was just wondering, maybe that's an easy way for them to fill their guts up, but it's not like, there is stuff out there. It's not like horse poop is the only thing they can find, but maybe that explains it.
Really nice talk, Rachel. I'm just wondering what you guys, or what you maybe have discussed about if an animal is sick, perhaps it's eating less because it doesn't feel good.
Mhm.
And so there could be an interaction between the forage and the pathogens,
Mhm.
so that if they didn't have to spend their energy fighting pathogens as well as had more desire to eat, that range would have been fine for 2,500.
Yeah, absolutely. And some of the things we started to work on is if those pathogens do come at some energetic cost. And that's exactly the line of thinking that we have, is maybe carrying these pathogens is explain some of this difference in nutrition.
Interested in your thoughts. There's a very robust elk population there with those sheep. So I don't think we've seen a decrease in calf production recruitment on the elk population. And matter of fact, they keep increasing the harvest because elk keep doing better and better. Yet there's a lot of forage left. So I don't want the audience to know that utilization levels are high, because they were the lowest this last year, I believe, since several years. So a lot of forage is still being left. So it kind of still, sheep have a pathogen complexity in their lives, and the elk don't, and the elk seem to be doing fine, but the sheep don't.
Right.
Just a thought of mine. I don't know what you think about it.
Yeah, absolutely. And I wonder if the elk are somewhat pushing the sheep out from where they used to be. So for example, we used to Greg used to see sheep all the time on Goat Flat, and we just don't see it quite as much anymore. Maybe because those are the ones that are dying or not recruiting lambs. But I also wonder, we definitely see elk up there. So. Okay, thank you.
Thank you, Rachel. Our next presenter is going to be Sonia Anderson. Sonia received her undergrad degree in wildlife, rangeland, and watershed resources from the University of Arizona. She completed her master's degree in wildlife biology from the University of Montana, working on mule deer on the Rocky Mountain front. She's been with the Montana Department of Fish, Wildlife, and Parks since 2011 and is based out of Lewistown in central Montana. She works on a variety of projects, issues, and species, but overseeing a portion of the Missouri Breaks bighorn sheep herd is among her favorite parts of the job. Outside of work, she enjoys hunting, hiking, horseback riding, making art, and helping her 5-year-old daughter, Sage, enjoy these things as well.
Okay, so today I'm going to be talking about the Little Belt's wild sheep restoration effort. This project was headed and completed by Jay Colby. He's our area biologist out of White Sulphur Springs. He did all of the background work to get to see this done. I'm just here presenting it. So these slides have been shown before several times, but sheep used to be pretty widely distributed across available habitat in Montana. This little red circle here is just where the Little Belts are. And by the mid 20th century, sheep were pretty well diminished at historic lows due to unregulated hunting, habitat loss, competition, and the introduction of novel diseases. And they were extirpated from the Little Belts during this time. Montana began its restoration efforts in the 1920s, completing over 130 transplants and moving 2,200 sheep. These efforts ceased in the 1990s. And then our current bighorn sheep distribution, sans the little belts, looks a little bit like that. Sheep are restored to many areas across the state, but nowhere near where their historic distribution fell. And so in 2010, Montana Fish, Wildlife and Parks published the Montana Bighorn Sheep Conservation Strategy as a guiding document for bighorn sheep management in the state. It contains a history of Rocky Mountain bighorn sheep, including the establishment of herds through trap and transplant efforts. And it also provides guidelines on reintroducing sheep. It directed the department to establish 5 new viable and huntable populations over the course of the next 10 years and to augment existing populations where appropriate. Given that timeline, 10 years would have been 2020. And despite this direction, Montana had not established a single new herd since the strategy's publication. And with this, there are numerous limiting factors that have limited our ability to restore sheep. One of the first things that would come to mind would be habitat, but given habitat modeling and the abundance of suitable potential habitat for bighorn sheep in Montana, that does not appear to be the case. Disease is another issue that's pretty quite commonly brought up and as a factor in limiting populations and restoration. Numerous studies have implicated commingling as the primary factor of epizootic outbreaks in bighorn sheep. And so in line with this, the 2010 conservation strategy discouraged reintroduction efforts if bighorn sheep were likely to range within 14 miles of domestic sheep. And this direction effectively limited the little belts from consideration. A lack of source herds is another limitation. There aren't very many herds in Montana where we have a huge surplus of sheep to provide. And also transplanting sheep from one area to another also can transplant those endemic pathogens, putting other nearby herds at risk, or the herds they're being transplanted into. Social tolerance is another big issue. Separation policies designed to protect wild sheep have often made local producers and landowners feel alienated and less inclined to support augmentation efforts. Larger domestic sheep operations and grazing allotments are pretty easy to track, but hobby farms constantly spring up throughout the state. We don't always know where they are. And then a lot of hobby farmers are also just naive to the impacts of interactions between domestic and wild sheep. And then public hunting opportunity is a big factor in Montana's wildlife management. Establishing new populations of bighorn sheep is not realistic in areas where there's little public land or where there's not a commitment from landowners to provide hunting opportunity. Oh, sorry. So basically, there's no perfect place left for us to restore bighorn sheep in Montana. And therefore, in order to do so, we've had to increase our risk tolerance a little bit. The conservation strategy laid out a hard and fast buffer for where we can and cannot put sheep, but with new disease research, we have a nuanced understanding. So we're constantly looking and constantly learning about disease interactions with wild sheep. In Montana, we've sampled over 1,000 sheep for disease pathogens since 2012. Respiratory pathogens are pervasive in nearly all populations, and MOV exposure has been detected in 80% of our populations. However, we have some populations of sheep that have exposure to these pathogens yet thrive. The ultimate cause of epizootics and resultant die-offs are complex, variable, and we still don't fully understand them. And it's likely that many factors interact to produce a disease event. And so this brings us to the Little Belt Mountains. It's an island mountain range of about 950,000 acres, about 50 miles southeast of Great Falls and 70 miles east of Helena. There is no existing bighorn sheep population in the Little Belts. However, since about 2014, we've documented a number of individuals around the Nyhart Monarch area, but we've never seen more than 6 at a given time. These sheep are most likely descended from either the Beartooth WMA area or break sheep in 482. which also founded these bear-tooth WMA sheep. So if basically they're likely to just contain a lot of the same genetic characteristics and a lot of the similar pathogen histories. There are no domestic grazing allotments on the U.S. Forest Service. However, there are bands of domestic sheep within 3 to 5 miles of the release area. And it is primarily public land. So there would be hunting opportunity if the restoration is a success. So for habitat suitability, FWP initially used the MaxScent model to identify suitable habitat, and it predicted, this is an older model, but it predicted a good number of suitable habitat in the Little Belts. Newer models developed in 2021 also indicate the same thing, that there's a high amount of suitable habitat for bighorn sheep in this area. Bighorn sheep were also historically present in the Little Belts. This pictograph was taken actually really close to the release site. And bighorn sheep are mentioned by Charlie Russell on his travels back in the late 1800s. And then this is just a picture of some of the sheep habitat in the Little Belts. This is the South Fork Road right here. We initially released sheep just on the other side of this mountain here. And then our second release area was up here. And then here's another picture of the sheep habitat in the Little Belts. Our source herd is from the Missouri River Breaks in Hunting District 482. And it's one of the most demographically robust populations in Montana with high recruitment and survival. This is despite endemic pathogens, including MOV. FWP has sampled and tested 119 live captured sheep since 2010. And while presence, the prevalence of MOV is low. And there have not been any symptomatic animals or an all age die off in this herd. The herd has also been above objective for the most part since 2008. And while we do have ewe hunting in this district, we've issued between 20 and 40 ewe licenses since about 2010. And we've also removed about 220 sheep from the area since 2003 to augment other herds, but they still do fairly well. So to get this project off the ground, Jay did a lot of outreach with local landowners, local producers, the Montana Wool Growers Association, land management agencies, sporting groups, and potential partners, and got buy-in and support from all of them. And one of the initial commitments to landowners and partners was that all initially released bighorn would be radio collared. And so the national and Montana chapters of Wild Sheep Foundation purchased 62 collars over a 2-year period for us to deploy. These collars are GPS collars with mortality detection, a 13-hour fixed rate. And then there's also a geofence that sends a text alert whenever a sheep enters or leaves a predetermined polygon. So in December 2020, we captured and released 49 sheep, 44 ewes, and 5 young rams. Last December, we released an additional 33 sheep to supplement that initial release, 29 ewes and 4 rams. That totals 82 sheep with 73 ewes and 9 rams. Right off the bat, we did have a lot of mortality. 20 of the 44 ewes died. We didn't lose any rams that first year. 15 were killed by mountain lions, 13 prior to lambing, 2 killed by coyotes. 1 was unknown, 1 was an apparent starvation, and 1 died during a fall. And then since January 2022, we've lost another 10 ewes and 1 ram. 7 were killed by mountain lions, 2 by coyotes, 2 unknown. And just one important thing of note is that 9 of the 11 sheep mortalities this year were all from the second cohort. Necropsies indicated that most of the sheep were in good condition based on marrow and body condition metrics. Predation has been the leading factor in mortality of these released sheep, and it's consistent with what other reintroductions and augmentations suggest when you put naive sheep in an area with predators. However, given that the pattern of mortality is largely affecting the newer sheep over the more experienced cohort, maybe there's greater survival after an X amount of time for these released sheep. And so for this winter, we also increased the quota on lions for the area, and 6 lions were legally harvested near the release area this year. A little bit more about the geofence. This is when we receive the text messages about when sheep enter or leave. We've had kind of intermittent success with this. A lot of times the alerts don't differentiate if there's a really high DOP associated with the waypoint. So there have been a number of false positives. But then we did have a handful of sheep kind of leave and hang out in this bluffy area and traveling back and forth. There has been one ewe, she went about 10 to 20 miles east over to the town of Buffalo and then came back. And then we have one ewe that's currently on the west side of the Big Snowy Mountains south of Lewistown, but there has been no documented commingling with domestic sheep. We've also been monitoring lamb production and began intensive monitoring last May 1st. All the lambs were born between the 10th and 19th of May. And so what we'd do is we'd use collar location data, and when a ewe was really locked into a certain point away from the others, we'd go out, find her, and determine whether or not she had a lamb. And this was repeated throughout the summer to see how many of the lambs possibly survived. 16 of the ewes died prior to lambing, and we confirmed that 22 lambs were born. Pretty much every adult ewe that wasn't a yearling when she was captured produced a lamb. Also with habitat use, we're finding that the sheep are using the habitat as they're predicted to by the models, and that's giving us more confidence in the model's capability to predict sheep habitat. They have not developed any sort of migratory behavior. Winter and summer habitat use are pretty near identical. But again, break sheep were not migratory either. And overall, the mountain range is a fairly lower elevation. So just some kind of closing remarks. Higher numbers of sheep and multiple transplants, we feel will increase our chances of success here. We released 82 sheep over a 2-year period, and mortality of the first cohort decreased after year 1. And then we hope to incorporate the Little Belt Sheep Project into larger statewide efforts, especially looking at potential methods to prevent commingling. And with that, I'll take any questions.
It appeared there was a fair amount of conifer encroachment into some of that habitat that you're transplanting sheep into. I was just wondering if there have been any wildfire histories or any planned habitat disturbances that you might be thinking about there to improve.
I'm not aware of any. Jay probably would be, and I can find that out, or I can get you his contact information if you'd like. But the conifers have definitely, as far as monitoring these sheep in the future, that will be rather difficult, especially as we have fewer and fewer colored animals, just because the timber cover is pretty high.
I've got a question, Sonia, and Kurt and I have talked to Jay a little bit about this, but we all learn every time we take a step. And so looking at that lion predation the first 2 years, I'm just wondering if within FWP, probably any jurisdiction, have you guys talked about doing some advanced or prophylactic type lion work before starting another transplant somewhere else, in a few years, just to give those sheep a leg up?
Right. I'm not aware of any conversations as far for doing that before release. I think there's only been one case of lions being killed to protect bighorn sheep, and that was on Wild Horse Island, and that was done by the tribe. We definitely have the capability to do that, but so far we've just been relying on public hunting to, upping the quotas. Yep.
Well, so to combine what both Kevin and Ryan said, the combination of prophylactic predator control early on, as well as habitat improvement with prescribed burning, having the Forest Service do some of that before your introduction would likely increase your survivability, I would guess.
That would be one, yeah. Yeah.
I'm just looking long-term here. When the geo fence collars no longer work, what are you going to do? Are you going to replace those and continue monitoring with the, I guess, the goal to keep them away from the domestic sheep? They're pretty darn close.
Right. So I don't know of any plans to recollar any of the sheep that were released after the collars are no longer functioning. They have a 5-year battery life right now. But there has been a lot of work with area landowners and neighbors who are pretty keen on just kind of looking out and letting us know if they happen to see any wandering sheep, or the sheep producers happen to see a sheep in with their domestic flock. And if that happens, we would find and remove that sheep.
Just a follow-up question on that. Will you be adding geofencing collars to maybe some of those young rams as that herd grows?
I'm not aware of any future collaring efforts of sheep that of us coming in and capturing sheep after we've already done that, but it's definitely a possibility.
You mentioned that there's a very low prevalence of MOV. I don't know if it's exposure or infection in the Missouri breaks. Did you transplant any sheep that were MOV positive?
Where's Austin? Do you know of any of the disease? Okay. There could have been.
Hey, just because I looked at that data for my stuff, there was no MOVI present in the ones who were transferred your first set. So those first 50.
Thank you. All right. Sounds like I'm off the hook.
Thank you, Sona. Our final presenter for this session is Ryan Amundsen. Ryan is a terrestrial habitat biologist for the Wyoming Game and Fish Department out of Southeast Wyoming. After graduating with a range management degree from the University of Wyoming in 1995, Ryan joined the department and he's worked in the habitat field for the last 27 years. He's worked in a wide variety of habitat types in the state and has developed strong working relationships with federal and state land management agencies, nonprofit conservation groups, and private landowners for the benefit of diverse wildlife species. Ryan also serves on the department's bighorn sheep working group, helping the agency tackle numerous complex issues affecting bighorn sheep in the state. In Ryan's free time, he enjoys leatherworking, riding and training horses, volunteering for the Wyoming Wild Sheep Foundation, and hunting in the mountains of Wyoming on horseback.
Thank you, Ben, and thank you all for coming to our great state and to Planet Jackson. Hope you had fun this week. So I'm going to talk about the Mullen wildfire that happened in 2020 in southeast Wyoming, just west of Laramie, burned 176,000 acres. It was an illegal campfire on the Medicine Bow National Forest. It took about 2.5 months to burn that much country. But it did take some pretty wild runs in late September. It's the third largest wildfire that's happened in our state's history. It's kind of hard to tell by this photo, but the forest is largely dominated by lodgepole pine, which was impacted by beetle kill epidemics that happened in the late 90s and early 2000s. Here's a picture post-fire, June 1st of 2021. You can see the North Platte River in kind of the left part of that photo. Very high fire severity. Probably the most startling thing there is there's not a snowdrift left anywhere. June 1st, we had an extremely dry spring, very, very poor vegetation recovery in the first year post-fire. The map on your left is a map of showing soil burn severity completed by the Forest Service and what they call their burn area emergency response teams. Areas in red, of course, are the highest burn severity, and areas of green are very mild. The map in your upper right is hunt area 18 for this herd, about 75 to 100 animals estimated there. And they spend most of their time in the areas that are circled in red. And it includes 2 wilderness areas, Savage Run, as well as the Platte River wilderness areas. So the Forest Service determined about 10% or 17,000 acres of this wildfire area were at extreme risk for cheatgrass invasion post-wildfire because of burn severity and known populations of those plant communities. This slide is just for the people from Nevada that are not familiar with cheatgrass. Just a little history from Mike Cox and company. It is a winter annual, very shallow rooted plant. We see a lot of soil movements and erosion when that plant is dominant on the landscape. There's a very small window where it's actually palatable to animals. When it first germinates in the fall, it can be palatable, and then again a short window in the spring. But as this photo shows, by early June to mid-June, there is no value left in that plant whatsoever standing there. We can see some animal injury in eyes, nostrils, in the mouth with sharp awns on those plants, and they get stuck in your socks, and I'm sure you don't like that very much. I think the biggest thing with cheatgrass is it does cause kind of a desertification of the landscape. Even in the early spring when things are supposed to be really green and popping, that's a desert landscape at this point in time when it's dominated. And in my career, I've never seen a plant community that's been dominated by cheatgrass revert back to a native perennial plant community by itself. It's taken some act by humans to help that. This is a really good photo showing how important slope and aspect can be in determining where cheatgrass is going to be most prevalent. So in the background, that's a south-facing aspect in the North Platte River drainage, and in the foreground is an east-facing slope that has good native perennial plant recovery this past summer. So we use, looking at slope and aspect, elevation, anything from 7,000 feet to all the way up to 10,000 feet are going to be at risk of cheatgrass invasion in the Snowy Range. So we ventured out to the field last summer and established 150 monitoring plots with help from the Forest Service and USGS folks from Colorado. That included these 20-meter plots. We had 83 of them were considered planned percent cover, 23 were opportunistic plots, and 44 were very detailed floristic plots looking at vegetation diversity. And I made the note there, it was so hot in June, it was melting the soles on our boots of our hiking boots up there. So standing on south-facing aspects on black soils in that rocky terrain, it's just really hot. So that tells you how quick the growing season shut down. There's a heck of a lot on this slide. Sorry about that. But basically what we're trying to do, we're going to the field and getting all this vegetation monitoring data collected, and we're providing it to Colorado State University's Natural Resource Ecology Lab and what's called a NASA Develop team. And they're using that information to try and train these models to make sure what they're seeing from the sky is actually what's going on the ground. So a rock is a rock, a tree is a tree, and bare soil, that kind of stuff. So the floristic plots that we put out, again, very detailed. We're trying to get plant species diversity information. So when we go back to these sites after we've done some upcoming treatments, we can see what kind of impacts those herbicides had on those species. The planned percent cover plots were mainly to determine percent canopy cover of cheatgrass. Again, to train the model so it could actually see how many acres are actually infested by cheatgrass, because we can't get to all 176,000 acres within that fire scar. And then opportunistic plots, as our crews are going out in the field, if they came across a heavy infestation of cheatgrass, they would stop. and collect plot information there, again, to help us train those computer models. And we'll be going back to all of those monitoring sites in field season 2022. It'll be an effort of about 25 to 30 people from the agencies, and we'll take about 3 weeks time. Pretty major effort in the month of June to identify those species during the growing season before some of them really desiccate and blow up and dry away, or dry up and blow away, I guess. So the map on the left is all of our plot locations on the western flanks of that wildfire. And then on the right side of the photo, you can see some areas that have been converted to cheatgrass. It's a little bit tough to tell, but some areas is pretty significant. There's just a few graphs here to kind of show what we saw in year one. The pie graph up on the upper left, the two most disturbing things out of those 150 transects, cheatgrass is prevalent as canopy cover, 14%. And then the other big one to be concerned about is bare soil. 21% of those sites were bare soil, and something's going to try and fill that void. And without some intervention, I've got a really good idea what's going to fill that void there, and it's going to be cheatgrass. The graph in the lower left is showing the percent of bare ground and also percent of rock. And we know we're not going to grow vegetation over rock, but again, the bare soil we need to get covered up with hopefully native perennial plants. The graph on your right talks about species richness. That is encouraging what we're seeing. So on average, over those 150 plots, we saw 26 different native species of grasses and forbs and shrubs. So that means that there is a native perennial component there that will respond to some form of treatment. If we had low species richness, it was likely due to high cheatgrass prevalence, extremely xeric sites, higher burn severity, and then also steepness of slope plays into that too, in really well-drained, shallow granitic soils. So conversely, when you have 45 species in your transect, it probably means there wasn't much cheatgrass there, more mesic sites, lesser burn severity, and maybe a milder slope too. So this past summer, right after we got done doing all these monitoring plots, we opted to spray aerially with a helicopter, a herbicide called Rejuvra. You may not be very familiar with this one. We've been using plateau herbicide for the last 20 years with mixed success, I guess I could say at best mixed success. Rejuvra is made by Bayer Corporation. We feel like it has a lot of promise. It's very expensive. It costs about $40 an acre for the chemical, where plateau was about $6.60 an acre for the chemical. But we do like what we're seeing there. Less collateral damage to non-targeted vegetation. In particular, one of our key winter range shrubs for mule deer and bighorn sheep also is antelope bitterbrush, and it was really taking it hard when we're using plateau treatments. So we're not seeing that happen with Rejuvra. So it has to be applied as a pre-emergent. So you can, the good thing about Rejuvra too, we can apply that from mid-June till end of August. Plateau treatments had to be done pretty much immediately prior to germination. So it's a little bit bigger window when we can get a helicopter on site to do it. We're seeing greater longevity with Rejuvra, 3 to 5 years of control versus with plateau, we're hoping to get through 2 years of decent control. And then we started to see the wheels really fall off by year 3 and 4. And those graphs on the bottom left kind of show that 4 seasons after treatment, you know, we still had single digit percent cheatgrass control with Rejuvra, but it could be up to 50% or greater composition with plateau. So that's just showing that efficacy wearing off on that. So the other cool thing when we're doing cheatgrass treatments, we can see twice the leader production on our winter range shrubs where cheatgrass is controlled in the understory versus areas that are untreated. And that's really cool to see. Some of the open space areas in Colorado, in the Front Range, around Boulder, Fort Collins country, they've been using Rejuvra for a while there, and they've really seen this happen and very excited about what they're seeing there. So the areas in purple there identify the south-facing aspects, mostly that were treated with Rejuva via helicopter this past summer, 10,300 acres in total. Probably the most exciting thing there is we sprayed over 6,000 acres within those wilderness areas. So when the Forest Service tells you we can't spray in wilderness, say, aha, they've done it. They've done it in the Snowy Range. So kudos to the Forest Service for working through the paperwork nightmare that was part of that to make sure that could happen. So this coming summer, we have an additional 4,000 acres that are on the eastern flanks of the Snowy Range that'll be treated again, probably in the month of July. We work with a company called Frontier Helicopters out of Johnstown, Colorado. They do a phenomenal job. They have a really good pilot. As I mentioned, the costs of this kind of work are extremely expensive. Rejuva herbicide at 5-ounce rate, which is the rangeland application rate, is $40 an acre. Helicopter application at $35 an acre. That's likely to go up this summer because of fuel prices, I'm sure. Anyway, this past year, we were able to put together over $1 million from our conservation partners to treat that country. $500,000 came from the Forest Service and their bear funds, emergency response funds, and then the rest of it from Wyoming Wild Sheep. We do have an organization in Wyoming called the Wildlife and Natural Resource Trust Account. That has been our cash cow, and we utilized a lot of their monies to get it completed, but also some secretarial order money also because it is some important mule deer migration route country as well. There has been some sheep collared in this herd unit. In 2019, 9 ewes were collared. So we have 2 years of pre-fire habitat use information there. This herd has been very, very limited by the conifer encroachment that's occurred there. And then here in 2022, just a couple months ago, we collared 19 sheep, which included 5 sheep from 2019 that were recollared again. So we're going to get 3 more years of habitat use data out of those sheep. And that'll be really telling for us to see what kind of new habitats they're going to use now that a lot of them are available. The sad thing is, of course, these sheep are very dirty. 17 of the 19 were sampled for pathogens. Everything under the sun was kind of there. Probably the most alarming thing was MOV was just identified here this year. It was not identified in 2019. So we wonder if there's been some potential contact there. So 12 of 17 sheep did have that pathogen there. So you've seen the map on the left, that's burn severity. So the most severe parts of that in red were likely the most conifer encroached, standing lodgepole dead. The picture on the right is steepness of slope. Areas that are yellow to red are at least 30% steepness, and red would be the steepest stuff, of course. And then the center picture there shows some collar data points, green being the areas pre-fire, and the purple dots are area post-fire. So you can see them starting to foray out a little bit into some new habitats that they weren't using previously. We are seeing quite a bit of sheep use to the north of all those purple dots too, which is in the Savage Run Wilderness, kind of the upper third of that fire scar. So it's going to be pretty exciting to see where those sheep end up here. So next step, where do we go from here? We're going to continue to read these vegetation transects. We want to make sure that the herbicide's effective, that we're not seeing a lot of non-targeted species damage. And we're going to continue to help train those models and try and use that Landsat imagery and their computer modeling programs to map and identify cheatgrass on the landscape. That'll help us inform where we need to do follow-up treatments. If the herbicide efficacy holds for 3-4 years, we're probably not planning on any kind of retreatments till 2024, 2025. And we will treat new infestations as they arise, and whether we're seeing that on the ground or through the computer modeling. We'll continue to monitor herd health, and we're really excited about what new habitats they're going to use because a lot of escape terrain is now open for them to move around. And I picked up this quote from Hank Edwards. We were throwing around some emails after our latest disease sampling efforts. And I think this really sets it up pretty appropriately. This herd will be interesting to watch to see if habitat trumps pathogens or vice versa. So we'll see if this herd continues to thrive and do well, even though they have all these pathogens in place or not. And so hopefully we'll report back in a couple of years to everybody. That's all I have.
Thanks, Jessica. Ryan, good job. Two questions. One simple one about Rejuvra. Is there any limitations on proximity to live water, streams, et cetera? Share that if you would.
Yeah, so the Forest Service put on a 300-foot buffer away from waterways, which was a little concerning. We don't have that kind of spray drift issue. If you're following the label and spraying under 10 miles an hour, wind speeds and stuff. So I'm trying to remember what the label exactly says, but you're not supposed to spray directly over water, but up to water's edge. So I thought that the Forest Service was being extremely conservative with their 300-foot buffers. So as steep as like the North Platte drainage is, I mean, there's cheatgrass right up to the water's edge. So I hate to see that buffer being left. And it's something that we're going to talk about going back in on another entry to take care of. Rejuvra is not approved for use on BLM lands yet, only on Forest Service. We're working very hard to get BLM to get that EIS done so they can do that hopefully next year in 2023.
Okay, thank you. And second half of my question, if I can, Andy, are you still here? Yeah. So with that big fire in northern Colorado, what, 2 years ago, 3 years ago?
Same year.
Same year. Okay. I'm just wondering if there's some compare and contrast stuff that Colorado and Wyoming are working on, so we all learn more.
So I am not aware that they sprayed any acres in Colorado on national forest, in wilderness, outside of wilderness.
I don't know either. I was wondering the same thing.
Ryan, kudos to your habitat monitoring. I was curious if you were monitoring control areas, i.e. non-burned areas, and if you were doing any comparison between habitat quality, i.e. nutritional energy levels, unburned versus unburned, and if you plan to do that in the future.
Yeah, so our plots are looking at areas that were planned for treatment and those that are outside of treatment areas.
Unburned and burned.
Yes, there is unburned and burned also within that. So what was the other part of your question, Steve? Yeah, we are not collecting anything right now.
Yeah, Ryan, given you're applying it in June through August and you said you indicated maybe there's multi-year efficacy. So it's not just a one and done. It can last, it can persist and prevent. Is it prevent germination? Is that what?
That's correct.
Okay.
It's preventing germination. So Plateau was giving us 2 seasons of pretty good control. Rejuvra is now, they're talking 3 to 5 years of control. So even though it's much more costly, we feel like that, it's worth that.
That's fantastic. Maybe we need like one of those giant firefighting jets, like
Yeah.
those, the 747 style, and just hit everything.
Yep.
Ryan, did the Forest Service ever approve herbicides previous to this effort in wilderness?
Like,
Did they allow Plateau to be used previously?
I think this was their first stab at it.
Yeah.
Any herbicide use. So, yep.
I'm not aware of many other public land management agencies that are allowing. So
they had to
you got some good folks on staff.
Yeah, they had to complete what was called a minimum requirements analysis to be able to do it. So, showing a range of alternatives, and it was spraying via backpack, spraying via horses, and that country's huge, no way you could get it done in the limited window we have to do. So, and frankly, the helicopter applications are the most precise application method out there. Very consistent. Exactly. Yep. It's very precedent setting.
So, on the work in the wilderness, how did you get through that? Was it an emergency exclusion or a categorical exclusion or whatever? How did you get in there with the helicopter?
Yeah, so it was the Forest Service botanist and range specialist that did the paperwork for that. And it was, like I said, it was called this minimum requirements analysis that was completed. Showed a range of alternatives, and basically what wilderness values were going to be sacrificed if they did not do something. So they pointed to a lot of previous herbicide application work that we done with plateau herbicide, not even with Rejuvera, but I guess that was promising enough that they opted to do it. We were told it was going to be a one and done. This is your one time to try and do this. But what I'm hoping is through our transects and going back and revisiting these sites more than once is we can show that there's not a lot of impacts to non-targeted vegetation, that it's safe to use, and we should consider retreatments.
Yeah, so I think it's really interesting because a few years prior to this, on the other side of the valley, there's the Beaver Creek fire that was both in Colorado and Wyoming. And we worked with the Forest Service botanist in Wyoming and in Colorado for that same bear analysis, that burn emergency analysis. And they were offered no money for cheatgrass control for a very large burn. So it is very refreshing to see that has turned around. So yeah, no money was given and certainly no access to the wilderness. So I don't know what changed, Melanie, maybe you know, but yeah, it's nice to see that.
Political pressure, maybe.
So the minimum tools, it's called an MRDG. It's what you have to do anything in wilderness. So that's the standard procedure for approval of activity in wilderness, and it's a NEPA analysis, so it's not a categorical exclusion. And it sounds to me like I'm retired, but it sounds to me like this is a forest by forest thing on the Arapahoe Roosevelt versus the Medicine Bow route, and whether or not the forest supervisor was supportive and what the bear team was doing at the time. So I think that's it. And the other piece is that in Wyoming, you have that habitat fund that was coming to the table with money too. So I think that's a part of that whole piece. I think that you have had a perfect storm that does set an example for others to follow.
Got a question online. Any work planned to look at glyphosate levels in sheep grazing the treated areas and potential health impacts?
Okay, so we're applying this herbicide as a pre-emergent. Glyphosate is a contact herbicide. Roundup is a contact herbicide. So if you've missed your treatment window and you're applying it after cheatgrass has already germinated a little bit, you're going to have to add some kind of a tank partner with it, like glyphosate. So as long as you're getting your treatment done at the right time, you don't need to use glyphosate. And I choose not to whenever possible, because that's going to cause some additional impacts to some native perennial plants. Even though we'd put it on at a much diluted rate, talking like a 6 ounce per acre rate, versus if you really wanted to kill your lawn, you'd probably spray it at a 24 ounce per acre rate. So it's very diluted, but you're going to see some yellowing of some vegetation, and it could kill some very sensitive small forbs and things like that. So I don't want any part of glyphosate in my mix if I can help it.
Have you looked at any of that work that's going on in Montana? There's probably people here in the room that know more about it than I do. I've just heard a presentation where that fellow's doing trial plots and putting heavy fertilizer on. Have you guys looked at maybe doing that in combination or something?
We've talked about it. I'm not sure we've identified sites yet, but we've said we'd be willing participants in any kind of studies that way. Yep. Those would be extreme costs. I'm trying to remember what they were per acre. Hundreds of dollars per acre. Yeah. Yep. Yeah, none of these weed treatments are a one and done. I sprayed enough plateau over the last 20 years, sprayed them once and walked away and said, yeah, we took care of cheatgrass. And then 3 years later, it came blowing back up again. So we've had to be very diligent in doing reapplications in those sites. And over time, you know, it might be 50% a couple years later, and then all of a sudden it was 25 and 5%, you know, a few years later, and then we feel like we've got a handle on it. So they're not one and done. Yeah. Thank you. Okay.
Thanks, Ryan. Good job. So we'll take a break. We might make it a little bit more brief since we're a little ahead of schedule. Oh, you got a question? Okay. So we'll take a break here and maybe come back at 10 till, and we'll get out a little bit early too and get people on the road. And I'll talk about Melanie's issue here shortly too. Oh, but now might be a good time if there are people that are leaving. Is anybody wants to sign Eric Rominger's card that hasn't? I'll get it to you. It's going to Kevin first, and then we'll make sure, go see Kevin if you need to sign it, but haven't yet. So see you back here at 10 till. Thanks.
All right, I think we have mostly everybody. So I'm gonna start. I'm Carson Butler. I'm a biologist with Grand Teton National Park, and I'm moderating this session, which I like to call the last best session. But unfortunately, everyone from Montana is gone. So I don't really have any support here. We're gonna kick this off with Brittany Wagler. Brittany is a master's student at the University of Wyoming under Kevin Monteith, and her work focuses on forage quality, nutrition, and disease in bighorn sheep. And she really hopes you enjoy her talk today.
Thank you, Carson. And thanks for everybody for sticking around for the last session of the day. And yeah, I'll just jump right into it. First, I'd like to acknowledge all of the co-authors and everyone that has contributed to both of the projects that I'll be talking about. Yeah, there's been a lot of work, and I've only been here for a short period of time. So thanks to everyone who has participated. So wildlife capture and the data collection associated with it has led to really cool advancements in ecology, many of which have been integral to conservation and management. And specifically, long-term individual-based studies have led to insights into social dynamics, resource allocation, nutrition, and density dependence. And as important as these studies are, they can be challenging to conduct for many reasons. Funding can be hard to maintain and acquire and maintain for such a long period of time. And all the logistics that go into these studies can be challenging. And additionally, a lot of this work requires repeated capture of study animals, which can impose risks such as physiological stress, decreased nutritional condition, capture mortality, and altered behavior of study animals. Helicopter net gunning is one of the most popular methods for capturing large mammals. It's super efficient for targeted recapture of animals. Previous work has shown that it's safer than some methods such as drive netting. And it doesn't require chemical immobilization, which can impose additional risks such as decreased ability to thermoregulate, and especially for bighorn sheep, when you use like chemical immobilization, you can't really choose where the animals go down, and they can get into some pretty gnarly terrain quickly. Whereas with helicopter net gunning, the net gunner can choose where and where not to fire the net onto an animal. But regardless of the method of capture, there's always going to be some level of the risks that I just mentioned. And so I think it's our responsibility as researchers and people who are capturing animals to always be evaluating that to make sure we're using the safest and most effective methodologies. So following a capture event, we can see mortality of all different types, from malnutrition to predation and to capture related mortalities. And a lot of times these can be difficult to disentangle. So a common practice has been to use a predefined censorship window to censor all mortalities that happen after a capture event. And for ungulates, a lot of times that window is from 10 to 35 days. Probably the most common window is 2 weeks. But a problem with these predefined censorship windows is that they assume that post-capture survival should be 100%, whereas in a lot of populations, that's probably a naive assumption. We can see this hypothetical population up here. Daily survival is kind of bouncing around some level of naturally occurring mortality. Some days it's 100%, some days it's lower, but yeah, it's bouncing around some level of background mortality. So we come into this population with a helicopter and capture a bunch of animals, and we would probably see daily survival decrease on the day of capture and be suppressed for some amount of time until the effects of capture wear off. In this period of time where we see depressed survival following a capture event are really the mortalities that we want to be censoring. And if we censor mortalities that occur after this period, then we're probably excluding biologically real mortalities and excluding important data. And as we all know, a whole bunch of things can affect the survival of our study animals, from disease, especially in sheep, to age and nutritional condition. And previous work has shown that a lot of these factors can increase an animal's susceptibility to capture mortality. And additionally, we have a bunch of capture and handling protocols that we've developed to decrease the risk of mortality on animals that we capture, such as limiting chase times and cooling animals that have elevated temperatures. And a lot of these things do work, especially reducing chase times. However, the variables that leave an animal most susceptible to capture mortality are largely unknown, and understanding them will just aid in making sure we're using the safest and most effective techniques. So specifically for bighorn sheep, some of the long-term data that we've collected over the years and all across the country has led to really cool insight into the ecology of bighorn sheep, and has kind of led to some of the most promising conservation actions that we can take right now, such as test and cull, which requires repeatedly capturing an animal, identifying chronic carriers, removing them from the population, in hopes that that will increase lamb survival and subsequently population growth. And then also just surveillance of pathogens across these different herds, and using translocation are some of the most popular management tools that we use, which all require capture. So especially for bighorn sheep, it's important to understand how this is affecting survival. And so we set out to do just that. We use a long-term data set to evaluate how long and to what extent does helicopter net gunning affect survival of bighorn sheep? And additionally, how do biological factors and handling techniques influence the probability of capture mortality? So we captured 736 bighorn sheep in both Wyoming and California. These captures occurred over a wide range of seasons. Our pre-winter captures happened between October and January. Most of them happened in October through December. And then our late winter captures occurred from March through May, and most of them happened in March or April. And additionally, at each capture event, we were capturing both new and recaptured animals. And so for the recaptures, we were able to monitor their survival in a window before the capture event. And additionally, in Wyoming, we were focusing on recaptures. However, we weren't able to capture every animal. And in California, recapture was less of a priority. So at each capture event, we have a sample of animals that were captured, and then a sample of those that were wearing a collar but weren't recaptured during that event. So we could look at post-capture mortality for both captured and uncaptured animals. And an important clarification is that's actually a bighorn sheep under that helicopter and not a snowmobile. So once an animal arrived to us at the staging location, we immediately took its rectal temperature. For animals that were above 104 or 105, we cooled them using water or snow or a cold water enema. In Wyoming, every single capture received a fever reducer and pain reliever. In California, only those that had high temperatures received that. We then took a variety of samples and measurements, including blood, fecal, and hair samples. In Wyoming, we took both tonsil and nasal swabs. We aged them by counting horn annuli and looking at tooth replacement and wear. And we measured their nutritional condition with ultrasonography and palpation. And every animal was then ferried back to the capture location. We did release some animals in Wyoming that were captured less than 3 miles away. Okay, jumping into some results, I'll just start with some summary statistics. We captured 736 animals, 347 of those were recaptures. The mean number of times that an animal was recaptured was 1.9. I think 32 animals were captured at least 5 times, and 1 animal was actually caught 10 times. Our handling times ranged from 5 minutes to 3.5 hours. The mean was 40 minutes. And the age ranged from 1 to 16, and the mean was 6. And our maximum chase time was 360 seconds. That was definitely on the long end. Most of them were between 30 and 90 seconds. And so going back to our first question of how long does net gunning affect survival of sheep, we used the survival data from every animal and we centered it on the day of capture. So day 0 on this graph is the day of capture. Everything before that is pre-capture. Everything after 0 is post-capture. So our mean daily survival for our pre-capture window was 0.9992. And so that's kind of that background level of mortality that I was referring to earlier. We saw survival decrease on the day of capture, and it rebounded to pre-capture levels within 3 days of capture. And we were unable to detect any differences between the captured and uncaptured daily survival within that whole 35-day window. And so moving forward in our analyses, we used that 3-day window in which we saw suppressed survival following captures to define indirect capture mortality. So overall, we saw 1.36% direct capture mortality, just over 0.5% of indirect capture mortality, for a total of 1.9% capture-related mortality. And then we use these mortalities to evaluate how biological factors and handling techniques influence the probability of these mortalities. Specifically, we looked at initial rectal temperature, age at capture, nutritional condition, the number of recaptures, chase time, handling time, and the season of the capture. So for direct mortalities, we had a sample size of 10, and we did not look at initial rectal temperature or handling times because most of the direct mortalities were dead by the time they arrived to us at the staging area. We did not detect effects of any of these variables on direct mortality. For indirect mortality, we had a sample size of 4, and we didn't detect any effects besides those animals that came in warmer had a slightly increased probability of succumbing to capture mortality within 3 days of capture. And I'm sure you've noticed we have a very small sample size here, so that really limits our statistical power and our ability to detect relationships here. However, the fact that we had such low mortality with over 700 captures of sheep kind of speaks to the relatively low risk of mortality that we're imposing when we're capturing animals within the bounds of these variables here. So overall, we observed pretty low risk of mortality in capturing bighorn sheep. We were specifically interested in the effects of repeated capture and data collection associated with longer handling times because of their implications for long-term individual-based research. And this kind of shows that you can do those kind of capture protocols while maintaining relatively low risk of mortality when conducting these captures in a responsible manner. So our capture mortalities occurred mostly randomly. This means that although we observed less than 2% capture-related mortality, sometimes you might see more than that. We had a total of 52 different capture events, and most of those capture events, we actually had 0% capture mortality, and some of those events we had is up to like 10% and anywhere in the middle of that. And we weren't able to find any differences between the capture events where we had 0% mortality or higher rates of mortality. And that's kind of tricky too, because some of these capture events, we were only capturing 20 sheep or so. So 10% mortality can be just a couple animals. And then we did see that relationship between indirect mortality and initial rectal temperature. So that just kind of emphasizes work that has been done previously, and that we should really be prioritizing cooling animals, especially those that are hot at the staging area. And I would like to point out that we only evaluated one metric of risk here. That's the risk of capture mortality. And as I said, there's other risks involved, such as physiological stress and altered behavior of study animals. And though it's a kind of a difficult thing to quantify, we haven't observed any differences in performance between our study animals and non-study animals in the same herd. And previous and ongoing work has showed that ungulates return to their home ranges within a couple days of capture, and their movement behaviors return to normal relatively quickly. And so we've been using pretty conservative post-capture windows to censor mortalities in an effort to avoid the influence of capture on our survival estimates. And this work shows that those windows might be a little too conservative, especially in this situation. If we would have used a 2-week censorship window, we would have been leaving out likely real mortalities, which can actually have the opposite effect and lead to inflated survival estimates, especially during times where there might be elevated mortality, such as after a harsh winter. And the last point I would like to make is that the low mortality that we observed here is related to every part of the capture process, and that all starts with the skill and expertise of the capture crew and goes all the way down to the level of care that an animal receives while they're at the staging area. So it's all tied together and can really impact the mortality that we might observe at a capture event. And I'd like to thank all of the funders of both of these projects. With over 20 years of sheep captures, it's been quite an expensive couple of projects. So thanks to all of our funders and collaborators. And with that, I would take any questions. If we have time, I think we do. Steve.
Real quickly, Brittany, could you address fetal mortality and lamb survival rates with captured versus uncaptured, or what you found there?
As far as fetal mortality, we've had, as far as in this project, every sheep gives birth. So I don't think that that's happening here. I'm not sure of anywhere else that that's been documented, especially in smaller ungulates. I think there actually is some things that show in moose. If you're capturing later on, there could be some effects on the fetus. But as far as captures that are occurring in March and April, I don't think they're really affecting the fetus. And yeah, we have seen relatively similar recruitment rates, because when we capture animals in December, we can see if they're lactating, and the recruitment rates between a lot of our study animals seems to be the same to what people observe, like with the state biologists by doing their flights. So that seems to be the same. Yes.
Hi, thanks for that. That's just useful information. I may have missed this, but just wondering if all the animals included in the study were transported, or were any captured, processed and released on site?
Yeah, in Wyoming, there were some that we just released on site, but most of them were ferried back to their capture location.
Sorry, it may have been confusing there. Were they all transported from their capture location initially?
Yes,
Okay,
yes.
Thank you.
Great talk. I was wondering, very, very small sample size, but is there any correlation between number of times they've been captured? I'm thinking of like after an animal's been exposed to a capture event, they may be a little more cognizant that the helicopter that's chasing them is trying to actually catch them. And so is that extending the chase time and thus potentially increasing the internal body temperature?
Right. That's a great question. So initial rectal temperature wasn't correlated with any of these variables, including the number of times that it had been recaptured and surprisingly chase times. But yeah, I think that's probably true for some sheep, but just anecdotally, it almost seems like it sometimes happens in the opposite direction, whereas they're kind of like, okay, they're here again, and they kind of lay down. But yeah, I think you're right. And I think when it is happening in the way that you said, I think they are just able to get into forest and we can't capture them anyways. Yeah. She's our friend.
Yeah, I agree. This was a great talk. Thank you. And you're what, 750 sample size? That's fantastic. Thanks for doing that. Out of those 750, how many mortalities did you have between that 3-day proposed possible like new censorship period and the 14-day like current suggested period?
I think 16. That close to 16, I think. Yeah, I don't know exactly off the top of my head.
And that seems a little higher than the 0.999 that was your background pre-capture.
Well, so that's 16 mortalities. So it's all of the data is pooled together across everything. So in that pre-capture window, even though mean daily survival was 0.9992, there was still, I think, close to 20 or more mortalities that happened in that whole window. Yeah, I think those exact numbers are in the paper if you wanted to look them up. Thanks.
Yeah, thanks so much.
Again, good talk. And I might have missed it, but I know some of the captures had temperature reducing drugs and another drug. Was there any effect there? And during any of your captures, were there any calming drugs or, the atipazole or whatever that is that's calming? And then how did that affect on the actual release after administration of those
Right. None of these
behavioral modifying drugs?
Mhm. We didn't use any sedation of any kind, even partial sedation in these captures. So I can't speak to that. And then we were not able to evaluate the influence of the fever reducer that we used, just because so many of them received it. And we didn't, yeah. So we weren't able to look at that. And then the initial rectal temperatures that we were recording, those were right when the animal arrived. So even if we did use a fever reducer, it shouldn't have been reflected in that temperature at least.
You didn't have a capture crew take an initial.
No, that was when they arrived at the staging area.
Yeah, so, whoa, it's on. Thank you so much. We were actually just talking about this at lunch, talking about digging into our data because this came up at lunch. I think as a point of clarification, you captured all adults, right?
Yes.
There weren't no lambs. It was,
right.
Okay, thank you.
Thank you. That was an interesting talk. I have a question for you as well. So you looked at those 3 days, the 3-day window. How much do you think capture myopathy? What will be the window so that you can detect some longer term effects that are still from those captures by net gunning?
Right. Yeah, I mean, capture myopathy can definitely happen within a couple of days or a couple of weeks of capture. And that can certainly happen outside of this 3-day window. I guess I would just say prioritizing getting to the mortality and see if you could see if it's capture related or not. as far as like a window of time that you could say that, I think that's why we've used such a conservative censorship window in order to avoid that. However, once you start having potentially capture related mortalities and other mortalities happens, it gets tricky to just have a line to say, okay, it was capture related. So I'm not sure what the best solution would be. I don't think that it would just be saying, here's a line. Yeah, I think, yeah, that's a good question though. Thank you.
You could probably compare those that were recaptured and those that were not recaptured but still had collars, and just look at their their survival probability over a time span. I don't know how long that would be.
Right.
but I don't
Right.
know
So
whether
that's
you've
what
done
we
that.
so that's what we did.
Okay.
I can't go back to the graph, but we had once those points broke into black and gray, it was the black ones were the captured animals, and the captured animals did have lower survival for about 3 days. And after 3 days, the survival was pretty similar to the uncaptured animals that were wearing collars, and we didn't detect any differences thereafter.
Mine is really more of a comment of some things that we've seen in Oregon, and it's kind of the some accidents by the capture crew. One time, a couple of us were watching them hauling sheep and dropped it, of course, killed it, and didn't report it. And another time I found one right after a capture when we were doing a sheep survey. And I'm sure the same thing happened. And I just wondered how much that goes on. We've had some low bed outfits that I've been suspicious of, but those are two I know happened. If anybody else had that experience.
Yeah, I can't really speak to that much. We use a pretty good pilot, David, with Native Range Capture Services in Wyoming. And I think that's kind of what I was getting at in my last slide, in that the capture crew has a big factor in capture mortality. And so using people that we know are good pilots and are responsible and would at least report any incidents that they have is pretty important.
All right, the next presentation is coming remotely via the internet. The speaker is Tanisha Henry, and Tanisha holds a Bachelor of Science in Honors in Zoology with a specialization of Behavioral Ecology, and also a Bachelor of Arts in Archaeology with a specialization in faunal analysis. Tanisha is currently a graduate student at the University of Calgary under the supervision of Dr. Catherine Ruckstuhl. And Tanisha is interested in horn morphology, bighorn sheep mating behavior, as well as science education. And hopefully Tanisha is there and ready.
Yes, thank you for the introduction. Hello, everyone, I guess virtually. Today I'm going to be talking about hornography, which is what I've termed my methods, using photogrammetry and 2D measurement to assess bighorn sheep horn morphology. So I'm going to start with a brief introduction. Most of my research stems out of an interest in male weaponry. So these secondary sexual characteristics are largely the product of sexual selection, either male-male competition or female mate choice or some combination thereof. They're considered to be an honest signal of individual quality, with the idea behind that being that only the most fit individuals can afford the energetic costs of developing and maintaining these features. So I work out of Sheep River Provincial Park in southern Alberta, Canada. I'm part of the University of Calgary, and so I use their RB Miller Field Station while I'm in the field. I'd just like to acknowledge that I do my research on the traditional territories of the Blackfoot Confederacy, which comprises of the Piikani, the Kainai, the Siksika, the Tsuut'ina, and the Stoney Nakoda First Nations, as well as Southern Alberta is also part of Métis Region number 3. It is an honor and a privilege to learn, work, and study on these lands. So on the right here, we have a close-up of the Sheep River area that I work, this dark cutout across the middle of the picture is the Sheep River Valley. The area is dominated by pine and aspen forests, as well as open meadows. It's an area that has got a long-term data set, so lots of data for me to work with. As it's also quite easy to find the sheep here. So I work on Rocky Mountain bighorn sheep. They're an excellent study organism for my interests in horn morphology because they have these big, beautiful horns. I take advantage of their annual growth rings, so those annuli, because they're a reliable age determination technique. And so in the spring and summer, when resources are available, this interannuli space is grown. And then in the winter, when resources become more scarce and energy is being allocated to other functions such as reproduction and general maintenance and whatnot, growth slows down to almost a stop, and when they develop deep, dark grooves around their horn, which can then be used to age them. So this individual in this photo is 4.5 years old as the photo was taken in the winter. So the significance of my study. So there is a lot of interest in horn morphology and more generally the morphology of various different types of weapons and ungulates as it relates to male dominance, reproductive success. We're interested in how various environmental pressures might impact horn growth, such as resource availability or demographics. There's also lots of interest in the genetics of horn growth. So how different aspects of horn shape or size are inherited. What I've found, both in the literature and from my own experience, is that databases that look at horn growth have the potential to have kind of an inherent level of bias. For sure in the Sheep River group, these animals are captured when they're young, and we do take horn measurements when they're captured, but if that's done as when they're a lamb or in the first year or two after that, those measurements aren't necessarily representative of what their adult horn growth is going to look like. Additionally, horn measurements are taken post-mortem for individuals that are legally hunted, or for individuals that are found dead either by parks, people that work with parks, or researchers. However, not all individuals are found dead. And so using capture and postmortem measurements to build this data set isn't necessarily the most representative of a living population. Of course, an alternative to this would be to repeatedly capture individuals throughout their life. If we can avoid that at all, that's ideal. And so that led me to develop the research question of, can the size and shape of bighorn sheep horns be accurately measured using non-invasive photographic techniques? We hypothesized that using 2D measurement and photogrammetry would allow us to get accurate estimates of horn shape and size, which would then enable researchers to comment on the representation of various morphological metrics in living populations. So we had two predictions. The first was that 2D representations would allow us to measure the annual horn growth, so the length of horn that's grown each year. And two, that using photogrammetry, we would be able to develop 3D models which would provide accurate depictions of both length and shape. So this brings me to my methodology. Before I go on, I just want to point out that all the individuals, or more than 90% of the individuals in the Sheep River population, are individually identifiable. They're captured largely as lambs, at which time they're fitted with ear tags, which correspond to a unique ID number. DNA is collected, and we can track these individuals throughout their life. So the first methodology that I was exploring was the 2D measurement of annual growth. And this methodology was adapted from Pat Bergeron's method that was outlined in a 2007 paper that basically involved projecting a known scale onto ibex horns, taking a picture and using that picture to then measure. Of course, ibex horns are much straighter than the bighorn sheep, And so I wanted to know if this was a method that could be applied to curly horns as well as much straighter horns. And so we took photos with a known scale, one of two ways. The first type of known scale that we explored was laser pointers. So green laser pointers that I've drawn over for visibility here that are spaced 4 centimeters apart. The lasers are mounted on a digital camera and projected onto the ram's horns. Another way that I explored, which makes, you don't have to carry lasers around and is a little more accessible, is to actually use their tag, which is a standard shape and size, as a scale. So I was able to use the height of the tag, not the width, because the width, depending on how they're moving their ears, the width can be distorted quite a bit, but the height stays, pretty much the same regardless of what the sheep are doing with their ears. And so this part of the tag is 3.7 centimeters, and again, acts as a known scale on these photos. So we then uploaded the photos into the ImageJ software, which basically allows you to set a scale and measure other things in that photo. So we had laser pointer photos from a number of sheep. I wanted to compare the accuracy of these measurements to manual measurements. So there were a total of 9 sheep that we had both manual measurements that were either taken during capture or postmortem, and also had images that we had laser pointers projected. So 9 sheep, I had a sample size of 65, which includes the annuli from these individual sheep. So I compared the digital measurements that I got using both the lasers and the tags to manual measurements using linear regressions. The second methodology that I was interested in exploring is photogrammetry. And so this is a technique that basically allows you to develop 3D models from a series of photos. And so I tested and calibrated this model with the horn of a deceased ram. Quite obviously, this is no longer attached to a living individual. I took about 200 photos rotating the horn around an axis and from different angles. This photo series was then uploaded into the software Agisoft Metashape, which is a photogrammetric software. Right now, the comparison between the shape of the actual horn and the shapes and whatnot visible in the model are qualitative. I'm working on doing some more quantitative analysis on that. So basically how this works is the photos are uploaded, these black numbers and the blue squares all correspond to an individual photo. And this software takes points from those photos and aligns them to create a point cloud. So this is not a solid model. As you can see, there are points that are kind of just out in space. They're not really part of that horn. This particular model was built off of approximately 200,000 points. So then the next step is to apply a mesh. And basically what that does is it takes this point cloud, it eliminates those outlying points and makes it a solid model. The last step is to texturize the model. And so it adds the color, if those photos are colored, and it adds texture. So you can clearly see various annuli on this. You can see that there is a bit of scarring in this area. And you can also see some of the brooming that has happened here. So in terms of my results, when comparing the digital measurements of annual growth that I got with the lasers to the manual measurements, we had an R-squared value of 0.99. When I force this regression line through the intercept, we have a slope of 0.98. So this indicates that using the lasers in place of manual measurement is a highly accurate, unbiased method of measuring annual growth. Similarly for the tags, not quite as accurate, but very, very close. Same deal. The R-squared value was 0.98. And when we forced the regression through the origin, we had a slope of 0.96. So again, a little bit more variation, especially in this mid-length range, I guess. But again, an accurate and fairly unbiased alternative to manual measurements. for the photogrammetric 3D model, I developed the model, I 3D printed the model. So that's what you're seeing on the right here is that 3D model from 2 different angles. So if I was able to be there in person, I would have passed this around so you could have felt the model, but you can definitely tell what the annuli are. You can feel it even better. The brooming at the end of this horn is quite obvious. And then rotating on the left here is one horn. It's got a mirror image of itself, you know, for visuals basically. But again, it creates a nice representation of what that actual horn looks like. The annuli, damage, aspects of the shape are all represented quite nicely. And of course, when you build these 3D models, I mean, you're building them off of pictures. And so if there's any issues with the model, you can kind of compare it to the photos and decide whether or not, you know, a certain part of the horn has maybe been misrepresented or not accounted for in the angles of those photos or whatnot. So in terms of discussion, there are a few considerations that I encountered. The first is the photographs themselves. So I'm fortunate enough to work with a very large photo database. And I found that especially for the 2D measurements, the angle of those photographs matters. So the most helpful or the best angle if you were to go out and take photos for this purpose would be a profile shot, such as this one on the bottom here. For older rams, measuring those later annuli it's quite a bit easier to do it from a head-on shot. I wouldn't recommend using head-on shots when you're working with annuli that encompass part of that curvature. Additionally, the individual that you're interested in measuring should be the focus of photographs. We had, you know, lots of photos where an individual we were interested in was in a group, but you have to zoom in quite a bit on the computer. So if that individual is already the focus of the photograph, it makes it a lot easier to analyze that photo. It stays much clearer and whatnot. So in terms of the accessibility of the methods, first with the 2D measurement, I thought it was quite an easy method to learn and to calibrate. You know, at least our group takes lots of pictures of the sheep, so we have those photos available already. If they've got tags on, then that's excellent. They've already got a known scale basically attached to their head. I had an undergraduate student help me with some of my data analysis, and he was also able to pick up this method really quite quickly. We were able to process a large number of photos quite quickly, so definitely very doable. The photogrammetry requires a little bit more time in terms of familiarizing yourself with that software and learning how to create those models. It is also a little bit more time consuming in the field for taking those photos because you need to get those photos from enough angles around that sheep that you can create accurate and reliable photogrammetric models. That being said, I used 200 photos for the model that I made. That was overkill. I think you could easily do this with approximately 100 photos. In terms of analyzing the 3D models, this is part of my graduate work. You don't actually even have to print the models, which is what I started out doing, but I now use 3D geometric morphometrics to analyze those models. And so once you've got a digital 3D model of that horn, there's a lot that you can do in terms of analyzing shape and length and whatnot. So some potential sources of error. So both for the 2D representation for both the lasers and the tags, there was a standard error of approximately 2.0. I think the tag or the lasers was slightly under and the tags was slightly over. It's important to remember though with this, that part of that error might actually be in the manual measurement. So for sure, when I was going through the database of manual measurements, there were some that just seemed like kind of fishy, like they were way too big for a sheep of a specific age or whatnot. So definitely some of that error might lay in the manual measurements as well as some in the digital methods. Personally, to within 2 centimeters is, I think, for me, an acceptable amount of error. when the trade-off is not having to capture sheep repeatedly throughout their life. In terms of limitation, using this these methods does require, good quality cameras with high zoom lenses and whatnot in order to get high resolution close-up headshots of the sheep. So definitely a potential investment depending on equipment accessibility. Also, this relies pretty heavily on having tagged individuals or having access to that laser setup that can be mounted to the digital camera. I haven't played around with this, but alternatively, you could probably set a camera up on a tripod and take a picture of, say, this individual on the right here. And if you left that camera on the tripod, you could then probably use this road sign as a metric. If you went and physically measured it, you would then know your digital scale. So still usable, although if they're not tagged or you don't have access to the laser setup, some creativity might be needed. So in conclusion, 2D measurements and photogrammetry are accurate, non-invasive methods for measuring metrics of horn morphology. In terms of application, I'm using these methodologies as part of my graduate research. So I'm interested in looking at how various metrics of horn morphology, specifically horn shape, influence big horn sheep behavior, male dominance hierarchies, and female mate choice, and then ultimately tying that all back into male reproductive success. And thus far, those methods are serving me well. So thank you for listening to my talk. I'd also like to give a quick shout out to Dr. Peter Newhouse, who helps me take the many photos that I need, especially for the photogrammetry that I do, and has given me access to photos going way back before I was watching sheep. I'd also like to thank one of our lab's undergraduate students, Fabien Rude, who helped me with the data collection for this talk. So thank you. And any questions?
Thank you. That was really interesting. I'm wondering if there's an application for individual identification, so you wouldn't even need to capture them, and potentially even be a mark recite methodology. Like, could you quantify the horn measurements enough to call them, to number them, and then that'd be a mark, and then going into the future, you could use mark recite to estimate your population, and then to take that to the next step, would this work on ewes? Yes.
First, definitely analyzing these photos, especially when you look at individuals in a group. Once you've drawn the dots and analyzed enough of the horns, I definitely feel like I can start to recognize the individuals based on their horn shape and locations of different damage and whatnot. So I definitely think that there would be the potential to adapt this and to take it more into an individual identification type thing. Because my research focuses on male reproductive success, I haven't done a whole lot with the female horns. We do have a number of photos with the lasers projected onto the female horns, and I don't see any reason why it wouldn't work on the females as well as the males. If anything, I think that the curvature in the male's horns would make them the more difficult horns to measure.
I'm wondering, in cases where you might have multiple photos of the same individual, if you've looked to see how close of an answer you get from multiple photos.
Yeah, so that's what using this research or this method in my own research, I do use a number of photos and average over them. Looking at them, sometimes the tag is at kind of a weird angle if it's, been flipped up or something. Those I can eliminate. Definitely the more photos, the better. And again, like, we can analyze a large amount of photos really quite quickly once, once you're familiar with looking looking for those annuli. And I mean, especially for individuals that we're familiar with, being able to compare markers on their horns in person is definitely definitely helpful. But yeah.
Got a question online if we have time.
Oh,
We good?
yes.
Okay. Joe, I'm going to allow you to talk here. So Joe, go ahead and unmute and ask your question. Joe, are you out there?
Oh, I think I can actually see his question in the chat. The question is, how are secondary growth grooves identified in the photos? For the most part, using multiple photos, I can kind of figure out which is the actual annuli and which is a secondary groove. If not, there were a handful that I really couldn't figure it out if they were very, very close, and I measure to a midpoint. In terms of most of the annuli, though, especially those older annuli, it becomes pretty obvious when they encircle the whole horn. And again, for individuals that are still living that we're familiar with, watching them, I can kind of cross-reference with the real sheep.
I think we have time for just this last question.
Okay, one more, sorry.
Oh, no problem.
I'm wondering if you saw rams that got, say, 10 plus that no longer grew any more horn as they aged. Like essentially they got, the annuli get smaller and smaller,
yeah,
or closer and closer together. And then at some point, did you see rams that no longer grew horns, or could you always differentiate another annuli?
For the most part, I could differentiate future annuli. There's actually one individual that lived to be 16, which we had manual measurements. He was one of the ones that the measurements, though, were just like, I didn't trust the manual measurements. They were way, way too big. Like they would have been like world record measurements for a 3-year-old ram. But he lived till he was 16, and it does get a little bit more difficult to determine those annuli, and especially if they're underneath the hair, because obviously with photos you can't kind of push their hair down and measure right to the base of the skull.
Okay, well, let's have a round of applause for Tanisha. Thank you.
Yeah, thank you.
All right, next speaker is Tom Stevenson, and I'm getting his biography now. Okay, Tom is the program leader for the Sierra Nevada Bighorn Sheep Recovery Effort and is a wildlife supervisor with the California Department of Fish and Wildlife. He currently has affiliate faculty appointments with the University of Wyoming and Utah State University. He received his bachelor's, master's, and PhD in wildlife biology from Colorado State University, Virginia Tech, and the University of Idaho, respectively. Tom was formerly a research biologist with the Alaska Department of Fish and Game and the director of the Kenai Moose Research Center. His research focus is, the screen went black, it's nutrition, and I'm sure there's more to that as well. Come on up, Tom.
Thanks, Carson. Is that about the right distance? Yeah, I might resort to, that's probably better. Tell me if I'm moving it around too much, but that might be better. So I'm going to be talking about cost distance models for evaluating risk of contact. There's a number of us in here that are part of this risk of contact working group, and obviously it's a subject of interest to a lot of bighorn biologists. We have taken a slightly different approach from Josh, and that effort that was developed, and he's going to be following my presentation, so it'll be interesting to maybe have some discussion about the different methods if there's time afterwards. I just wanted to acknowledge my list of co-authors here. Most of us work for California Department of Fish and Wildlife, and this is an effort that we've been working on for years. I don't even want to tell you how many years we've been working on this, but we finally got it where we wanted it, submitted it for publication. It's been accepted. Kathleen Anderson is the first author on this work, and you'll see a lot of nice graphics in here that she generated. She is here. She's a spatial data analyst, and she really preferred it that I gave the presentation because she was afraid she'd pass out. And given that I'm her husband, I figured I should probably avoid that. But she's here if you have any questions. I do a lot more public speaking, so in front of these professional groups, so I was willing to do it. So Sierra Nevada bighorn, for those of you that aren't familiar with them, are a federally recognized endangered subspecies of bighorn sheep. They're endemic to the Sierra Nevada. They're only found in the Sierra Nevada. I've been with the program about 20 years, and that's about how long they've been listed. And during that time, we've collected quite a bit of data on animal locations. We've collared over 600 bighorn. So we have a pretty robust data set, and that in part led to our interest in developing this somewhat different approach from what Josh is going to talk about, because we had, such a large quantity of data. And that's a portion of the Sierra Nevada in the background, just to give you a sense of what the terrain's like. So over the years, and actually going back, 40 years or so now, because even prior to listing, there was efforts in restoring this animal. We have 14 occupied subpopulations now, and sometimes I'll refer to them as herds. The historic range was about 10,000 square kilometers. It includes substantial portions of Yosemite National Park and Sequoia and Kings Canyon National Parks, just to give you some reference. So there's quite a bit of latitudinal variation and changes in elevation across that recovery area. We have animals that winter as low as 5,000 feet, animals that winter over and some are over 14,000 feet. So you've got about 10,000 feet of relief in portions of the range. So you can have animals that are wintering at low elevation in terrain and conditions somewhat similar to those rams on the right, and at the same time have animals wintering in the alpine. And we've published several papers on this. We recognize several different migratory behaviors, from traditional migrants to alpine residents, as well as what we call vacillating migrants that move back and forth between those high and low elevation ranges. So I thought it might, for groups that aren't really familiar with the Sierra Nevada, I thought I'd give a little bit of a background on the Sierra Nevada. It's, unless you've lived in California or spent time in the range, you might, you've probably heard of it. You've at least heard of the beer. But it's, it's a pretty impressive range. It's actually the largest single mountain range in the lower 48 in terms of, and it's particularly relevant to this type of analysis because there's so much contiguous escape terrain. And I think that's the real key about why we developed this approach and why it's, I think, particularly relevant to our system. May or may not be relevant to other systems, but sort of keep that in mind. And what we did here was projected it over Wyoming, not to diminish the size of Wyoming or anything like that, but it it illustrates, you know, how large of a range it is. And I'm not suggesting that that entire Sierra Nevada polygon there represents all bighorn habitat, but what it does represent is a lot of contiguous escape terrain. And you know, you can look at the polygon in California next to Nevada, and Nevada has a tremendous amount of mountainous habitat, a lot of that sort of basin and range topography, but it doesn't have the continuity that we see in the Sierra Nevada. So one of the two primary reasons for listing Sierra sheep was disease risk from domestic sheep and goats, and the need to manage that in order for the animal to be delisted. This is actually a bunch of domestic goats on some private land at the base of Wheeler Ridge, which is one of our herd units. It illustrates kind of the proximity and the Sierra in the background. But we don't have a huge amount of concern with private land grazing like you do in a lot of areas, because 98% of the Eastern Sierra is public land. So there's not a lot of opportunity for private land grazing. So really the public land grazing was the bigger concern when we developed this, but the techniques used is useful for both. A lot of history of domestic sheep grazing in the Sierra Nevada. You know, it's at one point they were probably grazing millions of domestic sheep in the Sierras, and that's undoubtedly was a major factor in the demise of the decline of the subspecies. So our approach uses a cost distance analysis. Just to give you a quick idea of what that entails is we start with a source raster, which in our case is going to be core home ranges. And then we developed a resistance raster that allows us to project animal movements across the landscape with different pixels having different levels of resistance. So if it's high quality habitat, it's going to have lower resistance. You sum those values across the landscape and end up with that final layer on the right, which is a combination of the linear distance as well as the underlying habitat characteristics. And I'll go through this and hopefully it'll make sense to you. So the first step of the analysis was developing a resource selection function. Our initial RSF we developed using 30,000 locations from 67 RAMs. We developed the RSF using RAM locations because those are commonly considered to be the highest risk of moving into domestic sheep operations. They also use the greatest diversity of habitats. So we really wanted to be liberal in our characterization of habitat. We used the May to December period because that was the period that domestic sheep were on the landscape, at least on the public lands. Our RSF, these are the variables that were identified in our top model. You know, there are a lot of the typical variables that go into particularly bighorn models: elevation, slope, hillshade, distance to escape terrain, vector ruggedness, and also a number of these vegetation variables that were either indicated selection for or against particular veg types. And, you know, and I will say, you know, bighorn, I think I've worked with RSFs on a number of other species. Bighorn are a lot easier to model than a lot of species just because they're so linked to these topographic variables that are relatively easy to acquire and incorporate into these models. So this is a sort of a close-up of a couple of our herds and that RSF. The yellow-green coloration represents the areas that are most selected for, the white the least selected for, and then we just, as an example, overlaid a bunch of those locations, a bunch of the location data and how it fits with that preference. And obviously, when you generate an RSF, you can identify a lot of surrounding habitat that's what we would consider habitat, but not necessarily occupied. So this is that same RSF, a little bit different color scheme. And this is the full scale of the RSF for the area that we modeled. And the yellow-green band, the large green band that's on the left is the Sierra Nevada. And then you've sort of got that purple strip that comes down the middle right here. That's the Owens Valley. And then on the right, you have the White Mountains and the Inyo Mountains. And that's desert bighorn habitat on the right. In 20 years, all these collared animals, we've never had a collared desert bighorn travel across the valley. We've also collared animals on the east side, and they haven't come across. And, you know, early on, I was kind of puzzled by that because, you know, it's not uncommon for bighorn to travel 10, 15 miles. And that's at the closest point, that's about what you're talking in the Owens Valley. But I think the difference here is you have so much contiguous habitat that if a ram wants to travel, he doesn't need to travel across the valley. He can stay, or a ewe for that matter, travel through habitat with escape terrain. So then to create that resistance layer that I talked about for the cost model, we inverted and transformed that RSF. And as you can see, that Owens Valley is that yellow band that runs through there. So there's a lot of resistance to movement for bighorn in this system. Then the second layer that's required for this is the source point layer. So for that, we used a 50% core home range and identified in red there. And those are for all of those 14 herd units. And we used a 50% core because we wanted to identify the point where we needed to identify a starting point and then model movement out away from those core home ranges. So then there's a few more steps after that. On the left there, you know, we have the resistance layer with the core home ranges overlaid on it. Then you produce that cost distance surface, which is the second image from the left. And there you can really start to see how you have that blue polygon, or sort of those two blue polygons that really represent that connectivity up and down the Sierra Nevada. And as you move off to the east, you have that higher cost to travel outside of bighorn habitat. Then we clipped it to 60 kilometers because we had to identify an area that we could work with and develop all these spatial layers for. And we use 60 kilometers because that's the maximum distance that we've seen a ram or a ewe travel from their core home range. And then we rescale and invert it to create a final risk surface. Sometimes that gets a little confusing and I even get confused. So forgive me if I start mixing up numbers here, but when it's a cost surface, there's little cost to moving when you're within that high quality habitat. So it's going to be 0, close to core, and 100 as you move away. By inverting it and creating this risk surface, it's going to be a value of 100 in that core range and 0 as you move out. And you have to think of it in terms, the risk is risk of grazing domestic sheep in that habitat. So if you're grazing domestic sheep right in the middle of bighorn habitat, it's going to have a value of 100. Oh yeah, and so just real quickly, so I mentioned that we transformed it. We evaluated a number of different transformations, and you end up having to transform this data to really to recognize that value of the habitat quality. Because if you just use a constant value, then the linear effect has the linear relationship has more of an effect on cost than the habitat quality. And really what we want, so we ended up using a natural log transformation. So when you come over to this right image, there's far less cost for an animal to move up and down within bighorn habitat than to move outside of it. Here's just another image that sort of illustrates how it works. If you pick a 30-kilometer radius, kind of focus in on that, and there's values associated with that that are pulled from that underlying cost layer, which is actually the risk layer in this particular case. So if you're in bighorn habitat at 30 kilometers from the core, it's a value of 99.1. But then if you go off to the east where you've moved into the Owens Valley, that risk value drops to 45.3. So you could graze domestic sheep in the valley at the same distance as in bighorn habitat and actually have much lower risk to bighorn sheep. So hopefully this is making sense. I'm watching Kevin. He's not looking too confused. So he's my guide right now. So then once we've developed this risk layer, then we needed to, so the agencies, so we've been working closely with the land management agencies in the area, and they wanted us to identify a risk threshold using this. And so to do that, we included all of our GPS data. This is a few years ago, so we actually have quite a bit now. Over 1 million GPS locations, 105 rams, 196 ewes, all seasons. And using this, we were able to establish a risk threshold of 91 by overlaying animal locations on that cost surface. And you'll see there's a few points where we did have some forays where animals came out and came right up close to this polygon. And those are the values that were used to establish this risk polygon, this risk threshold. So when the land management agencies use this model, they're really keyed in on that value. And so essentially everything outside of that black polygon is a value that's below 91. So then a few other things we did just to try to help validate this. We looked at 100% herd minimum convex polygons. So these were individual animal polygon, minimum convex polygons that were merged to create herd polygons. You can see they all fall within. And some folks ask, well, how do you incorporate forays into this? Well, those herd polygons incorporate the forays. So the foray, all of the animal movements are incorporated into this, and it represents all the long distance movements that we've documented. Just one other quick thing. We compared our risk polygon to we just picked the 25-kilometer standard buffer. And the real difference between the standard buffer and this is that the standard buffer over represents risk, say when you're moving off to the east and outside of bighorn habitat, but in many cases it's going to under characterize it within bighorn habitat. And then finally, this is what the situation we're in now is, and we've been working on this process really for almost 20 years. We've identified a lot of grazing allotments within this polygon that have been closed or vacated by federal agencies. Most of the blue that actually falls within that risk polygon is not grazed through various agreements at this point, but there's still some work to be done. You can see that there's still, quite a bit of grazing that occurs outside that polygon. But for the moment, what we've tried to do is identify what we consider to be risky grazing. And with that, just want to acknowledge all the agencies and organizations that help fund this work, as well as many field assistants over the years. And if you're interested in learning more about Sierra Bighorn, you can visit us on the web.
Thanks for the great talk. And I think really an interesting use of cost distance to try to incorporate more of the variation in the landscape into these considerations of risk. One thing I was curious about is, so this is all centered around kind of the bighorn movements out. And I was just wondering how much or what have you thought about, or how have you thought about the other situation in terms of domestic sheep moving in the other direction?
Yeah, well, and so we have an interagency group that has developed a document that looks at trying to manage this risk. And this is actually one component of that. The other component is looking at domestic sheep operations and the factors that either affect the potential for straying or the that can prevent strays from coming in contact. So for example, we look at physical barriers on the landscape. So we are using that in combination with this. And I mean, I tend to be very risk averse when it comes to grazing. Early in my career here, I spent a lot of time observing grazing operations and was told all sorts of things about this never happens, that never happens. Well, it all happens. It's just a question of, over how much time will it take for straying to occur. But as far as that question of how do we address that portion of it, I think we haven't really tried to develop similar models, although you could certainly use a similar approach. For the most part, we've really tried to ensure that there are barriers in place that would. So like, for example, there's a highway that runs up 395 or runs up the middle of the Owens Valley, Highway 395, that's, fenced on both sides. I wouldn't call it an impermeable barrier, but it's a 4-lane highway in a lot of places. And, so things like that, bodies of water are incorporated into that document.
Tom, just a question, and Mike, I'll point this sort of at you both. With that long linear 400-kilometer north-south axis range, then you look at the basin and range country over there. I'm just wondering how you would modify this approach in that basin and range country where there is a lot of squirting across from across the valley floor to the next range. Have you guys tried this in Nevada, and
yeah, how do we
what's it look like?
Well, one thing that we want to do, and we haven't quite gotten around to it, but I think we're getting closer to it, is we've wanted to apply this model, say, in the Mojave Desert, which is a little bit different, but somewhat similar situation where you have these islands of bighorn habitat surrounded by intermountain habitat. I mean, I think it's still going to work because the cost distance model actually does is pretty good at identifying where bighorn will travel, even when it's not completely contiguous, because they tend to jump from feature to feature. And since distance is also a component in this cost distance, but it may be that it's more akin to using a buffer than strictly using habitat. Right. Yeah. So, I mean, at least by applying this, we would at least be able to evaluate what the effect of habitat is on some of those movements in that type of environment. But I mean, we need to try it, and we just haven't gotten to it yet.
So the big difference I saw with the cold risk of contact, the current risk of contact, incorporating foray into the entire move versus us, or home range, and then the added foray. We've got some big polygons that obviously see that
Well, and so I think, you know, what's, and correct me if I'm misinterpreting this, but what we call core home range is 50% core home range. So it really is just the core. And then we're modeling the movements out from that. But then when you look at, you know, like I had those 100% MCPs, you know, we're incorporating everything that's bighorn habitat. I think the core home range that's in Josh's risk of contact model actually encompasses a much greater portion of bighorn habitat, but not necessarily all of those forays. And, you know, as part of that group, we've had some discussions about this, and Josh can speak to this when he gets up here if he needs to, but that model was developed for folks that just don't have this quantity of data, and so don't necessarily have that complete information about foray distances or in particular the directionality of forays. So I think there's probably more information about distances of forays, but not necessarily the directionality. So I think that's, you know, Josh's model is potentially more adept at working with in areas where you just don't have the quantity of data.
Well, a lot of the forays that I've documented are single path, whereas with that into minimum common polygon, it's got force bigger or surface area to count that single line.
And that's one reason why we use the 100% at MCP here, is because it does, you know, it represents, I mean, depending on how much data you have, it may represent more or less area than, say, a kernel home range, but it does encompass 100% of the points.
Nice work, Tom. That's really interesting stuff. And I have a question that's sort of, I think it's sort of about risk tolerance, and it's sort of several parts. Like, I get this feeling that you've basically made the assumption that you've seen everything that's going to happen. And so my question is sort of two parts. It's something about collar coverage. You know, do you just have so many animals collared that you don't have to worry about unobserved points? And it's also sort of related to, I think this thing happens with GPS collars. You can put out a handful of collars on a handful of animals and start collecting points, and in a very short time say, oh, we got 1 million points or 2 million points, and feel like you're really riding high.
Yeah.
But if you only got 10% of the herd collared, what does 1 million points mean, you know? Can you talk about that a little bit?
Yeah, sure. And it's part of this interagency process we have is that we do rerun this model at some regular interval. And, you know, particularly if we have herd range expansions, then we rerun the model. If we, you know, as the population grows in size, we're going to continue to rerun that model. So we definitely don't view it as static. So that's one part of this. The other, shoot, what was the other part of pardon me? Yeah, that was it. Our goal is to try to keep about 30% of our female population collared. And sometimes it's more than that, sometimes it's a little bit less. And that's for a whole host of reasons, including getting mark resite population estimates. In terms of the proportion of rams that we've marked over the years, in general, if it's a small herd or a herd that's in closer proximity to domestic sheep operations, we try to collar a higher percentage of the rams, and we continue to collar rams. But the proportion has varied over time, but yeah, we pretty much collar everything we catch, and we can tell the helicopter crew to catch one thing and end up with all sorts of things, you know, which sometimes works out. I mean, they do a really good job with capture work, but sometimes they always want to collar the biggest catch the biggest rams, so we always get big rams, and I'll send them back out and say, yeah, we need some yearlings and 2-year-old rams. And so we have a pretty good cross section. We've even collared quite a few 6-month-old lambs. We've collared very few neonatal lambs. And we use expandable collars on yearling males in particular. So we do have GPS data on those as well.
All right, we're borrowed time at this point, so I think we got to cut off the questions.
Thank you.
All right, thanks, Tom. Next and final speaker is Josh O'Brien. Josh is a biologist and geospatial analyst living in Corvallis, Oregon. He received a PhD in ecology from the University of California at Davis. He now works as a consultant for nonprofit NGOs, university researchers, state and federal agencies on a variety of topics, including spatially explicit disease modeling, vegetation ecology, and plant distribution modeling, and of course, the bighorn sheep risk of contact model. Come on up, Josh.
Okay, is this working? Okay. Yeah, thanks. So Tom, that was a great talk and is a great intro in some ways to what I'm going to be talking about today. What I'm going to be discussing is assessing the performance of the risk of contact tools core herd home range estimator. And for those of you who are not familiar with it, the risk of contact model is one that we developed for an analysis on the Payette National Forest back in 2009, 2010. And then we have tried to make it portable and bundled it in a tool that can be used to apply the same sort of analysis to bighorn sheep in other areas. And the model takes three takes as inputs 3 different summaries of bighorn sheep spatial use. It starts with this definition of a core herd home range, which is intended to capture most movements of bighorn sheep, about 95%. And then focuses on modeling the forays out beyond that core herd home range. So it's really a model aimed at looking at the risk of contact at a distance from the core herd home range. And so the second summary quantity of spatial use that goes in there is the foray frequencies and distances. And then there's also a very crude model of bighorn sheep habitat preferences outside of the home range. But crucially, everything starts with the core herd home range and then is modeled probabilities of jumping off and traveling beyond that. And so because this model is being applied in many different places than where it was designed, from up in Canada, out in Nebraska, across the West, we thought it was time to sort of revisit some of the assumptions that are in the model and to basically look at each of those pieces in depth and see how well they generalize. So here's a diagram of the herds that we were analyzing when we developed the model, and from which we draw all of the default values, and which people tend to use because it takes a lot of data to estimate foray rates in a local landscape. Just for context, this data set had 1,300 animal years of data. And, some of the longer distance U4A forays, there's only 1 or 2 forays in that whole data set out to that distance. So it takes a lot of data to parameterize the model. And so in addition to having such a large data set, the data that went into that went into our analysis were collected on a sort of biweekly basis. So about a 2-week interval between data collection points, which is very different than any of the data sets that are being put into the model now. So the questions that I'm going to talk about in this talk are how well does the risk of contact tools default home range estimator perform when applied to these modern, much more high frequency and very autocorrelated tracking data? And then also, are there better methods? There have been in the last 15 years, lots of developments in the field of coordinated home range estimation, and would we be better off using an estimator that accounts for autocorrelation in the data? And so I looked at this both at the individual level, so testing how well our default home range estimator works for individual animals, and at the core herd home range level. So starting with home range estimation, the data set that we used to do this assessment is the NDOW telemetry data set. There's actually data from 60 plus herds in there. I restricted myself to data that were from animals that had at least a year of telemetry data on them. So that leaves us with 47 herds, well distributed across the state, 277 animals, 541 animal years, and a mean fix rate of 3.5 observations per day, which is just about exactly 50 times the fix rate of what is in the Hells County Initiative data set. And the 4 home range estimators that we looked at are old standby minimum convex polygon, local convex hull, this independent and identically distributed Gaussian kernel density estimator, which is the one that the model uses as a default, and then what's called the autocorrelated kernel density estimator, which I'll discuss in a minute, but it's a very hot new kernel density estimator that from the ground up models and deals with autocorrelation in tracking data. And so to show these 4 estimators, here's a example data set. This is a U up in the snowstorm range in northern Nevada. And this is about a year and a half of data. And so the minimum convex polygon, which you all are probably familiar with, we draw a trimmed one. So this is basically trying to draw a boundary that contains 95% of the points and is as small as possible. And so it's very easy to use. It doesn't deal very well with sort of holes or gaps in the interior, but still tends to perform pretty well. The second estimator is a local convex hull estimator. We call it, this one KLOCO-H, because there's a variety of different local convex hull estimators, and they are elaborations on minimum convex polygon estimators that work by drawing a bunch of small hulls, one around every point in your data set, and in this case containing K points. And the recommendation is that you use the square root of the number of points, put that into each of the small holes. So you draw all of the holes, combine them, and then selectively drop them in order so that each one that you drop is dropping the largest area. So the sort of the promise of these is that they're very good at identifying holes in the interior. And also if you have a home range that runs up against a hard boundary, a river or a fence or something like that, local convex hulls can pick that out. And so here's the home range that they would draw for this set of points. So they do a great job of, not including that putative hole in the middle of the range. So the IID Gaussian kernel density estimator is the estimator that ships with the model, the default one that's used. And the Gaussian refers to just a two-dimensional Gaussian mound of density or a kernel that's placed over each point on the landscape, over each telemetry point. And those are just sort of added up as if you were dropping sand on top of each point. And then once you've accumulated that density surface, you draw a contour around the density that's as small as possible, but contains 95% of the density. And so the key decision then, when you're using a kernel density estimator, is what shape that mound of density should have. And there's all sorts of different options. You can make it adaptive. You can find the density or find the shape of it by cross-validation. The IID Gaussian kernel density estimator just pretends that all the points are completely independent. So the model it works on is that if you have a set of telemetry points, it's as if the animal is moving randomly between them. And so it tends to draw quite small and very peaked mounds of density over each point. And so here's the home range that it would draw for this U. And then finally, there's the autocorrelated kernel density estimator. And what this one does, and it's actually it's really promising, is it first looks at the autocorrelation structure of the entire data set, and then develops a movement model that would fit that autocorrelation structure. And from that, computes the amount of time that it would take on average for the animal to travel from one edge of its home range to the other, which is called the time to independence. And then, or the range crossing time. And then finally uses that and the length of the data set to say, hey, although we have 10,000 points, this animal's only traversed its home range a handful of times. So we only have actually a handful of independent points. It'll then draw a much larger, broader Gaussian kernel and result in larger home range. And in this recent review that I cite here, this was a review of his application to like 270 different data sets. It does remarkably well. It achieves exactly 95% coverage on average across those data sets, and it does better than any of the alternatives. So we were hopeful that that might improve our estimates of corridor home ranges. Okay, so the way that we chose to measure home range estimator performance was number one, obviously you want to tally the home range size, because a well-performing estimator is going to only include those, areas where the animal is found and exclude areas where they're not. Then the second aspect is that you want a home range estimator that performs well, not just on the data it's trained on, but on other data, because as a range estimator, it's supposed to be ideally extrapolating well to where the animal will be. And so to implement that, we split each of these data sets in half, use the first half to train a core herd home range, and the second half to test the quality of it. So, what proportion of the points that were out of sample are included in the home range. And so for instance, here's with the minimum convex polygon. Here is a 152 square kilometer home range, and it ends up covering 48.6% of the next year and a half of data from that animal. So what had actually happened was this animal started out in the river, drainage up to the northeast, and then towards the end of that first year and a half moves down and continues to explore from there. So KLOCH does a really poor job of generalizing to other locations that the animal travels to. So it ends up with a coverage of 11.3%, and but it does have a very small home range size. Here's our model default. It gets a coverage of 41.9%. And then finally, this AKDE comes a lot closer to its nominal coverage, but it does so by drawing a really large core herd home range. And this home range ends up being this large precisely because the pattern of movements that this animal had was it started in one location, ends up in the other, and only crosses the range one time. So it's treated as if effectively it's only got a couple of data points. Okay, so I applied this, thanks. I applied this to all 377 animals in the data set. And as promised for male animals there on the right, the AKDE gets exactly 95% coverage, so it works really well. It overshoots a little bit for ewes, but the model default, the green there, performs better than minimum convex polygon, but not as well as the AKDE. As far as the areas of the home range that are drawn, both the minimum convex polygon and kernel density estimator, or KDE, are relatively small, and the AKDE is getting its improved performance by drawing a larger home range, but it also has, this is a log scale for area on the y-axis, so it has for some animals it draws sort of ridiculously large home ranges, up to almost 9,000 square miles for that upper point. And so just looking at the same data in tabular form, there's the median coverage of the AKDE, which is good. And the kernel density estimator is a little bit better in terms of being compact, which we would like. I'm going to speed through this. Well, this is just to show that there is a diagnostic, this effective sample size, which you can derive from the data, that will sort of let you predict which home ranges are going to be perhaps unacceptably large. So almost all of the home ranges that are extraordinarily large have real small effective sample sizes. So if you want to use AKDE but are worried about that bad behavior, you would be able to have a diagnostic that lets you avoid those worst case scenarios. And those large home ranges are both males and females, is what this is showing. So to review, the AKDE for home ranges has close to optimal coverage, produces these large home ranges for some animals. The kernel density estimator of the model does all right, but for home range estimation, I would recommend using the AKDE. However, the model itself is using actually a core herd home range estimator, which is actually a home range for a group of animals. And typically that's going to be based on, as Tom or Nathan was asking about too, it's often going to be based on just a few animals in the herd. So these are two of the animals in the data set showing that even though the Santa Rosa's maybe has more collars in it than any of the other data set, a relatively small proportion of the animals there are collared. So when you use these machine methods for estimating home range size or shape, you're still basing that on just a few of the animals. And so the way that I did this evaluation is I selected half of the collared bighorn sheep and estimated collared home ranges based on them, and then tested them on the other half of the collared sheep. And I did this in the same way by computing the collared home range area and the coverage of the out of sample points, and then repeated this 50 times per herd, sampling different ones of the radio collared animals and using them and then testing them with the others. And so quickly the results from that were that the minimum convex polygon KDE and AKDE all had very similar coverage. So the AKDE does not really have an advantage over KDE when you're aggregating the densities from a bunch of different animals. And at the same time, the AKDE still is ending up producing really large home ranges, which isn't desirable if it's not gaining anything in terms of coverage from that. So same thing in tabular form, KDE has basically equivalent coverage of out of sample points to minimum convex polygon and the AKDE. But it fares much better, or it does a much better job of producing compact home ranges than either of these other two. And so my conclusions are that the three methods provide similar coverage, but the KDE produces more compact collared home ranges. The risk of contact tools default choice seems to work as well as any of the methods, which is nice for us. And then I think it's worth highlighting that all three methods will often fail to map significant portions of the landscape that's being used. I mean, they all, when you used half the data points, so between 4 and 16 animals to set up a collared home range and then test it on the other half, they miss about 10% of the points. And typically there's a lot of other uncollared animals that are out there. So it's just a reminder to people who take the lines that are drawn on the map as reality that there's, if the animal doesn't have a collar on it, it could be anywhere. So, and with that, I'll take questions. Yeah.
Hi, thanks. That was really interesting. The home range literature is just, there's just so many methods. So it's interesting to see a comparison of those. So I was just wondering, what is the smoothing approach with the KDE approach that has been being used?
The smoothing approach that we're using is, okay, yeah, the smoothing approach that we're using is probably the original kernel density estimator that uses a Gaussian kernel, and it takes its bandwidth to be a function of the sort of, so it's of the -1/6 power. So it would work well for a bacteria on a Brownian random walk in a petri dish or something like that. It's sort of mathematically optimal for that, but probably not for any real animal, but seems to do a pretty good job also.
Another question.
Thanks. I know we've had a number of discussions about this, or I can't ever seem to get this to be the right volume. But since it seems like using that 95% tends to underestimate the area that the population's using, or even the individuals that you're sampling, do you have any other thoughts on whether we should be using something other than 95%? I know it seems like when you go to 100%, especially with some of the kernel estimators, they're overinflated. But something in between 95 and 100, I was just curious what your thoughts are about that.
Yeah, I think that would be something that would be worth sort of that'd be worth. Okay, yeah, thanks. Yeah, so I think that we could play around with that. Probably don't want to go all the way out to 100, in part because we treat the core home range as, if there's direct overlap, as being kind of certain or highly likely to result in contact. And so if we push it out too much, there's going to be large areas of empty space where we kind of know there's not any bighorn sheep.
Right.
All right.
You have to wait. All right. Hey, what a great day. What a great week, right? Can you give yourselves a hand? What an amazing week. We've learned so much, but we didn't want to leave here because there wasn't a talk that really specifically addressed some of the differences between goats and sheep that we actually see. We alluded to it last night. We want to make sure that as you leave this meeting, you leave knowing that there is a difference between goats and sheep. Now, I believe British Columbia actually created this so that you didn't have to be a biologist to know the difference between a goat and a sheep. So let's see what they put together. Stick around too. We have a distinguished guest that's going to speak to us at the end. So hang tight.
Goats and sheep, really where the mountains must be. Goats and sheep, climbing high, we don't need your ropes, no, no. You make comfortable, really fluffy to a pump. I really cannot help myself, I'm down here with the stove. My head is often smoking, eyes are gold and brown. My rump is white and so my nose, the rest of me is brown. Talk are quite elusive, live at high elevations. If you catch a glimpse of me, it's cause for celebration. My coat is long and thick and white, and helps to keep me warm. My boots are black, my nose is black and brown, my eyes are horns. Sheep and goats, goats and sheep, we live where the mountains must be. Goats and sheep, sheep and goats, climbing high, we don't need your ropes, no, no. We'll run the unicorn, but ramp on much bigger and curl around our heads. We crash into each other in a big forehead butt fight, so that we impress the ladies. Oh golly, what a sight! The Billies and the Nannies, they both are fluffy bodies. The on rods, climbing high, we don't need your ropes, no, no. I like walking hillside, the grass is like a munch. We sheep are very social and like living in a bunch. But predators like wolves and bears want me for a snack. So I give you defenses to help us stay attacked. I'm way up in the alpine, I love to walk these cliffs. And if I see a predator, I try not to let you. My body's slim, hospital, on narrow mountain ledges. Thanks to meals of lichens, grasses, flowers, and the sedges. Sheep and goats, yeah, goats and sheep. They live where the mountains are steep. Goats and sheep. Sheep and goats, climbing high, we don't need your ropes, no, no. I'm a worthless provincial mammal, you may see that's the ball. But some hunters just want trophies, my head upon their wall. But if I make my home inside a park, I have no fears. I may even reach the ripe old age of 14 years. Some folks think they're the same. I'm visiting the parks. We both have horns and hair and hooves. Can you tell us apart? My drunk brown back and curling horns. He's a bighorn sheep, all dressed in white, and way up high, a mountain goat you eat. Sheep and goats, goats and sheep, we live where the mountains are steep. Goats and sheep, sheep and goats, climbing high, we don't need your ropes, no, no. Climbing high, we don't need your ropes, no.
There you go. Now you know the difference. Hey, we couldn't think of a better way to end this conference than by having the words of John Claude Van Ram given to you. Please welcome John Claude Van Ram coming through the door right now.
This is kind of tricky. I don't know if you can hear me or not. I am supposed to be a doll ram, not a domestic ram, even though I have a beef cow muzzle. But anyway, I just wanted to say some closing remarks and thanks to all the speakers and our poster presenters. Let's give every one of them a hand or a hoof in this case. I don't think I can do this anymore. Anyway. That is on loan from our local Jackson Hole Conservation Alliance. So it is Jean-Claude Van Ram. You too can borrow it for a cleaning fee is all. So Amy was going to wear that last night playing the accordion, but it was just too dang hot in there. So anyway, I would just like to say thanks to my colleagues that helped pull this off. They helped even when they weren't asked, and when they were asked, they were given very little notice. I want to thank all the field trip leaders from the Game and Fish, from Grand Teton National Park, the Elk Refuge, University of Wyoming. Definitely a hand to our IT crew, Wayne and Samuel, for making this work. It's not optimal, but I hope you folks out there remotely enjoyed this and were able to participate as well as you could. I want to thank the staff here at Snow King for everything they did, but just the main thing is thank you all for coming to participate and for participating online. It was really good to see everybody in person. This was wonderful. Hope to see even more of you in 2024. And just wish you all safe travels on your way back home, wherever that may be. And just thank you and take care. Kevin?
Yeah. I mentioned the other day that Doug and I have known each other and worked together and hunted together and had fun together for 37 years. And I'm not sure I've ever been as proud of him as I am right now. No, Doug, good job. Thank you to all the Wyoming crew for putting this on. You know, I don't know how many this makes for me, but my first one was 40 years ago, and I'll be back for the next one. So Todd, maybe we'll talk some more about Alaska in 2024, but yeah, thank you all for coming. It's great to see friends and colleagues, make new friends. And so I'm the same with Doug. Safe travels home, and well done, Wyoming.